Oh, I see. About this mice imaging. So you use three different temperatures. So what is the influence between different temperature with the mice metabolite?
Yeah. So in this research, they do the bioluminescence for the what to say, the virus. Then the vaccine itself will be injected, and then will induce the neutralization antibody. Then when they inject with the virus, it will give the signals from the lungs, because the virus will target the mice lungs here. And then how they gonna be to be protected from the vaccination? Yeah, using the thermostable vaccination. So basically, this research more focus on the development on the encapsulated mRNA vaccine. So they want to develop because we know mRNA is very fragile, is unstable, easily to fragmented, and how do we encapsulate the mRNA as the vaccine with the thermostable. So no need to have the low temperature storage, something like that. Just the
judging from this in vivo imaging, the fluorescence in liver is very high. Yeah. Is it really effective to use this strategy? Because you target a lung, but a majority, the accumulation of the fluorescence is in the liver. Did they mention about this?
I think for this research, it only shows that the protections can confer the protection against the COVID. But for the organ itself, I need to check, Ms. Reni. Thanks for the confirmation.
Thank you very much.
Yeah, anything else from the chats? Okay, maybe Elokarsa will play some games at the end. I will stop share, and then my colleague, Ms. Stephanie, will conduct with the door prize. Yeah, we have already several people that already fill the form. Yeah, thank you very much for your participation. Then we can spin here, and we have two prize here. Okay, we will looking for two. Okay, please spin. Who is the winner? We will contact you after this. Okay, Miss Yati. And then the next one. Okay, Elo Rifki Virginia. Okay, thank you very much. We will contact because already submit the email and the phone. Thank you.
Thank you, Mr. Andreas Agustian, for the great speech, and congratulations to the winner. And you can contact Mr. Andrian for further information. Ladies and gentlemen, we have finished principal mechanism of biotechnology product session 1. Now, going to our next agenda, we will hear keynote speech from our keynote speaker, Professor Duk Jo from Asian Federation on Biotechnology, or AFOB, with the topic Enhanced Production of Recombinant Human Interferon Beta through Signal Peptide Modification. Now, allow me to invite Professor Duk Jo. Good morning, Professor.
Good morning.
Good morning, Professor. Before we begin, allow me to read a brief CV of Professor Dugjo, if
Yes.
you allow me, Professor.
Thank you.
Okay, so Professor Dugjo earned his doctoral and master degree in Chemical Engineering Korea Advanced Institute of Science and Technology, and his bachelor degree in Chemical Engineering, Seoul National University, Korea. He is currently a professor in Sejong University, Department of Integrative Bioscience and Biotechnology. Professor Dugjo research topic focus on bioreactor development for cell cultures, therapeutic protein production, including antibodies, cytokine, hormones, and many more. Cell culture process development, adult stem cell expansion, cell culture medium, including serum-free medium development, and gene therapy technology using virus and or non-viral delivery system. Now, without further ado, the time is yours, Professor.
Okay, thank you. Let me share the. Can you see?
Yes, we can see it, Professor.
Okay, thank you. Okay, thank you very much. I'd like to appreciate the organizing committee for giving me a great opportunity today to share our experience on the bioprocess and also cell line development. Okay, I'm Dr. Oh from Sejong University. I work for Asian Federation of Biotechnology also. So I'd like to invite you, all of you, to Asian Federation of Biotechnology. Actually, membership is free, so you can join and enjoy your network with other people in Asian region. Okay. Today, my talk is enhance the production of recombinant human interferon beta through signal peptide modification. Actually, my background is engineering, and I used to have a strong interest in how to enhance the productivity from cell culture by bioreactor development or bioprocess development, media development, that kinds of thing I have a strong interest. But this time, today, I have a little bit different area. Recently, we have interest in this cell line development area, and we got some results. So I like to share this result with the participants in this symposium. Okay. Okay, what is the interferon beta? It is right now it's a biotherapeutics and mostly produced by animal cells, and it can be used for treatment of multiple sclerosis. And still, it has a huge size of a market in the global. And then actually, since it is a kind of biologic product, so it needs to have harms like that. This time, focus on the cell line development, especially for vector engineering. But I'm not a specialist in the vector engineering, but we started with a small project to approach this area, and I'd like to share our experience with all of you. So as I told you, in order to produce a recombinant protein from animal cells, you need almost 3 steps. First one is cell line development that produce your product. And then after you get the cell line, you need to develop the cell culture process, especially for that cell line. You need some specialized bioreactor system, and you need to decide what kind of a culture mode you're gonna use, like a fed batch, perfusion, whatever. And also you need to develop the media to get a high productivity. Maybe it's gonna be serum-free media or chemical defined media. And then after you finish the cell culture, you need to purify your product. So if you optimize and develop the purification process, you can get a good productivity also. So most of my research area is focused on here, and most of my published paper is focused on this area. But this time today, I like to talk about cell line development, especially expression vector development through signal peptide. Okay, what is signal peptide? I think some of you already know that. And here we have a target protein, our target protein, and the signal peptide is a little bit earlier than your target protein, and it has a three region. This blue region is cleavage region. This sequence is you cannot change much because enzyme should cut this side. So if you change something, that enzyme cannot cleave this part by enzymatic reaction. And there is a hydrophobic region in the middle. There are hydrophobic amino acid to form the alpha helical structure. And there is an N region. This is positive charged. So this is very important part for especially the secretional protein. So I will show you one cartoon in textbook. So this is it. So this is a ribosome here, and the signal peptide is always translated first before your target protein. And then there is a signal recognition particle. They bounce together like this, and then they bind to a signal recognition particle receptor protein, and then your target protein can be translated and put into the ER rumen here, and then signal peptide can be removed and cleaved and removed, and finally you get the protein inside the rumen, inside the ER rumen, and then your protein can be followed by the post-translational modification, like cutting and then glycosylation or something like that. So the signal peptide synthesis is a translation is the first step important for secretional protein for secretion. So this is important. And then when you look at the interaction between signal peptide and the signal recognition particle, they have an interaction in M domain and with the signal peptide through alpha helical. This yellow one is alpha helical structure. They interact through this alpha helical structure. So in signal peptide, alpha helical structure is mainly located in the H region. That is a hydrophobic region. So we thought that we can change or increase or decrease the hydrophobicity of the hydrophobic region, that is H region. But in this case, we decide to increase because some paper already show that. So we decide to increase the hydrophobicity of the H region. So when we increase the hydrophobicity, we decide to use leucine instead of other hydrophobic amino acid. And also from the codon usage table like this, we decide to use a CTZ codon for leucine. So this one is the signal human IL-2 signal peptide example. There are 20 amino acid, and this part in the yellow box, this region is hydrophobic region. And if you look at closely, then you will find these blue ones are hydrophobic. Okay, let me close this first. Okay. So the blue one is a hydrophobic amino acid, and the red one is a polar amino acid, and the green one, the green one is basic amino acid. So in hydrophobic region, the H region, you still can find the non-hydrophobic amino acid. For example, here serine and cysteine is here. And isoleucine and alanine is not that strong as like leucine in hydrophobicity. So in a previous study with erythropoietin, not interferon beta, in my lab, we changed the hydrophobicity by changing the amino acid in this hydrophobic region. For example, we replaced the serine to leucine, cysteine with the leucine, and both together with the two leucine, or 4 of them, not strong or not hydrophobic amino acid to the strong amino acid, the hydrophobic amino acid, all leucine like this. So this kind of a strategy we applied, and by something is wrong. Okay, so by doing that, in a previous study, we could get the EPO concentration, transient transfected EPO production was increased, like a 2.7-fold increase when you change hydrophobicity in signal peptide. So this time we would like to focus on interferon beta. Interferon beta is very difficult to produce. The concentration is very low, and it is very unstable protein. So it's very difficult protein. So right now, this time, we try with the interferon beta. So here, this is the signal peptide of interferon beta. Most of the the secretional protein has their native signal peptide. So interferon beta is a secretional protein, so it also has its native interferon beta signal peptide, which is this one. So, and when you look at the hydrophobic region, it's very similar to IL-2 we already done with previous research. So we follow the same approach. We apply same approach to interferon beta signal peptide. So we change glutamine to leucine and the QIA to 3 leucine as we done in previous research. And then we see the result of this approach. So this is introduction, and then I will explain the material method. For this research, we used the 4 different cell lines, HEC293 cells and CHO cells. One is HEC293A, which is adherent cells, and HEC293F cell is a suspension cell. And we have 2 CHO cell lines, CHO-K1 for adherent and CHO-S, XP-CHO-S for suspension cells. Because we try this, because what kind of a cell line is good for interferon beta production, we need to decide. So we have to use for all of these 4 cell lines to make to do this research. For adherent cells, we use lipopectamine 2000 for HEC293A and CHO-K1. We follow this process. And for suspension cells, we use the polyethylenemine or XP-fectamine for XP-CHO cells. So by using this kind of transfection protocol, we could get the transient transfection of our vector developed, expression vector developed. And to make a stable cell line in the final, because we want to make a final cell line that produce stably, not transient. So we need to make a establish stable cell line. So we did 2 different kind of selection protocol. First one is Geoshin. So selection 1, phase 1, 2, and we use MTX selection protocol. So we follow the separate the selection process, and followed by the limited deleting method to have a single cell clone. And then we actually developed sandwich ELISA method to analyze the interferon beta concentration. And most of the material like expression vector, we use pcDNA 3.1, which one is commercially available. So there's no secret material. We use the commercially available product, and we used already known information to develop, to increase the productivity of the cells we produce. So result. So first one, we changed the hydrophobic region of interferon beta synapeptide. So it actually increased the hydrophobicity of the hydrophobic region like this. For example, here we change the amino acid like this, and then the results like this. This one is interferon beta synapeptide wild type. It's a native one. When we change it, the hydrophobicity by replace the non-hydrophobic amino acid to hydrophobic leucine, we could get this kind of result. And of course, when we use no signal peptide, there is no protein secretion. So that's pretty much acceptable. However, the data shows that the opposite to our expectation, interferon beta productivity was not successful. So what's going to happen? So, but we have a good experience with the IL-2 signal peptide. So we decide to, okay, maybe the interferon beta signal peptide is not that strong. So we decide to use the IL-2 signal peptide. So we went back to the IL-2 signal peptide that we already succeeded in EPO. So we did same approach. And then this one is, number one is a wild type interferon beta signal peptide. And the number two is a wild type IL-2 signal peptide. And then we, tested different, combination of the hydrophobicity fortified signal peptide of IL-2. And then it did not increase much, but still we could get the 1.5, 1.84 increase compared to the native interferon beta signal peptide. So, okay, we could get almost twofold increase. Is that enough? Actually, if it is a bioprocessing optimization process, 1.8-fold increase in productivity is pretty much acceptable and very successful. However, we are talking about the cell line development and vector development. So we could not satisfy with this kind of small number. So what else we can do? And then we learned that the Kojak sequence is very important to get a high productivity in vector engineering. And the Kojak sequence is the GCC RCC sequence. So R is a purine sequence, and so it can be adenine or guanine. So ACC or GCC, ACC or GCC sequence before the initiation codon. So if you put this sequence in there, Kojak sequence, we learned that you can get higher productivity. So we decide to apply this sequence in our vector. So this is the result. So when we do experiment with the HEC293 adherent cells, we could get, this is the number one is IL-2 modified, SCIA changed to 4Ls. So this is a modified IL-2 signal peptide. When we put the Kojak sequence in there, we could get about 3.5-fold increase, but just by adding the Kojak sequence, it's good. And compared to the wild type, the interferon beta native signal peptide, we could, this number is about 6.5-fold increase. So this is good. And in the Hectonized F cells, this is suspension cell, we did similar experiment, and then we could get the 3.5-fold increase. And then actually Hectonized F, the suspension cell, has a more productivity compared to the two Hectonized A cell line. So compared to these two, we could get 3.2-fold. So compared to the initial interferon beta native signal peptide in Hectonized A, we could get 21-fold increase in Kozak modified IL-2 signal peptide produced in Hectonized F cells. So this is good. We could get 20-fold increase. So compared to the previous one, 1.5, 1.8-fold increase, we could get the pretty much increase in productivity. So this is good. And also we did in CHO cells, because CHO cell is mostly widely used in industry, biopharmaceutical industry. This is also important. So we could get good increase, 9-fold increase with the Kozak sequence, and the 3-fold increase in Kozak sequence, especially in CHO S cells, and this one is CHO K1. And then the concentration different, CHO S cell line, we could get the between these two, the difference is about 104-fold, more than 100-fold. So if we think about from the native interferon beta signal peptide, it is more than 1,000-fold actually. When we use XP CHO S cell line with Kozak and modified IL-2 signal peptide, we could get 1,000-fold increase before and after. So this is amazing. And then, okay, then we try to find is there any other way to increase the productivity. We already have a modified IL-2 signal peptide. So we want to change a little bit more because we have in here, this is our modified signal peptide. We have 4 extra leucine, and originally there are 3 leucine here, and then we changed it 4 amino acid to leucine. So we have 7 leucine in serial. So so we want to see if we change the number of leucine in this hydrophobic region, what's going to happen. We just curious about that. So we changed that. So this one is our modified signal peptide with the Kozak sequence, and then we changed the number of leucine. This one is 5 and 6, 7, 8, 9. So we changed the leucine number, and that, but we could not get the enough, productivity as we have already. So it seems like the productivity, produced by our hydrophobic region modified IL-2 signal peptide is optimal, very close to optimal condition. And actually this one, number 4, has a 7 leucine, and our the modified one also 7 leucine, but difference is a codon different. So codon is a little bit optimized already in our signal peptide, but CTZ might not be best in this occasion. So that's the another thing. We could get the better productivity in our signal peptide. Okay, and then we actually, this is the hydrophobic region. That's why we have interest. But there's another reason. This is a basic reason, and we cannot change all of this. But there is one polar amino acid, so glutamine here, and that here is a methionine. So we try to see, because the barrier between this is not clear. So we decide to change the glutamine to leucine to see the effect. And also we decide to change it both two to leucine like this. But the results show that if we change the M or M2 to leucine, we could not get the good productivity compared to the original one. So we should not touch anything about this basic region, and we should keep the size of the hydrophobic region as same as we have. So seems like we have a optimized the condition of the signal peptide already. And the next step is to make a stable cell line. So we start with let's see, start with the geosine selection system. We did with 400 microgram per milliliter of geosine, and we could get some cells survived already. And then we did the second phase selection system. And then after that, we could get, this one is signal Kozak sequence and signal peptide optimized, modified system, but it's a transient gene expression, transient transfection. And after we make a stable, selection system, second phase, we could get this one. And then when we compare these two, there's almost a 12-fold increase compared to the transient transfection and the stable pool, not a single clone. It's a stable pool cells. We could get a 12-fold increase. So we select this pool to isolate single clone. So we did limited dilution cloning procedure. And then we finalized the three, most productive clones, 3 of them, and then we finally cultivate them and then compare the productivity. So we have 3 different clones, so Z1, Z2, G3 clone, and then we could see the G3 clone shows the highest productivity, and like this number, and this number is compared to the transient gene expression system, this clone gives us a 35-fold increase in productivity. So actually, this the increase in productivity is more than 1,000, because we already know that when we use the native interferon beta signal peptide, this one is already 1,000, and then we have extra 35-fold increase. So it's pretty much quantum jump from the beginning to the current stage. And also we did the MTX methotrexate selection phase, and we could get the very similar productivity like this. So conclusion. So we, to develop the highly productive cell line, we have to touch the signal peptide vector engineering. From interferon beta wild type, we use IL-2 wild type, and we change the signal peptide in hydrophobicity, and then we put the Kozak sequence. And when we did the IL-2 signal peptide, we could get 1.5-fold increase, and then 1.8-fold increase when we modify the hydrophobicity. And when we add the Kozak sequence, we could get the 6.5-fold. And then use HCK E93F cell, 293F cell line, we could get the 21-fold. And then also when we did the same procedure with the CHO cells, we could get the 104-fold increase in productivity. So this is pretty much high productivity compared to the signal peptide from interferon beta. So also when we do the stable gene expression, that means we can establish a stable cell line, cell clone. We could get the 35-fold increase from transient gene expression. We could get the 35-fold increase, and also when we use the MTX selection system, we could get 28-fold increase in productivity. So today I show that how we successfully increase the productivity in cell line engineering. Especially we touch the signal peptide, we change or modify the hydrophobicity, and we put some the important sequence, Kozak sequence there, and then after we finalize the selection system, we all together we could get the more than 1,000-fold increase compared to the non-treated original native signal peptide applied cell culture system. So this kind of thing, I don't know, but maybe some of this finding we got from here can be applied to the developed cell lines producing recombinant therapeutic cells, therapeutic proteins, which is a kind of major part of biopharmaceutical industry in these days. So this is the end of my talk. Thank you very much for your attention.
Thank you, Professor Dugjeo, for a very great presentation. And let us give a round of applause to Professor Jugde, and you can also give reaction.
Thank you.
Thank you, Professor. Ladies and gentlemen, we will continue to our next agenda, which is plenary session with speakers, Professor Zihua Jiang from Department of Animal Sciences, Washington State University, USA, and also Mr. Zahir Ali, PhD, Center for Desert Agriculture, King Abdullah University of Science and Technology. This session will be moderated by Professor Siti Subandia. Now, before we begin, allow me to read a brief profile of Professor Siti. Professor Siti Subandia earned her doctoral degree in plant pathology, the United Graduate School of Agricultural Science, Gifu University, Japan, master degree in plant pathology, Faculty of Agriculture, University of Queensland, Australia, bachelor degree in phytopathology, Faculty of Agriculture, University of Gajah Mada, Indonesia. She is currently a professor in Faculty of Agriculture, University of Gajah Mada, where previously she was also a faculty of member in 1983 to 2017. She was honorary professor in the university, she is currently the honorary professor in the University of Queensland, Australia, guest professor in Gifu University, Japan, and visiting professor in Shizuoka University, Japan. Her research interests are in the area of diagnosis and management of plant diseases and molecular plant protection, and also agriculture biotechnology. Now, without further ado, Professor Siti, time is yours to lead the session.
Okay, thank you very much, Ibu Wiwit. A very nice introduction of me. And good morning, good afternoon, or good evening for everybody in this meeting. I hope everyone is stay healthy, and we have already having better condition. The pandemic already declining. Yeah. Okay. Thank you very much for this opportunity for me to lead this talk. And the first speaker will be Professor Zihua Jiang from USA. Yeah. Can I read the CV of Professor Jiang? Okay. Professor Zihua Jiang is a full professor at the Department of Animal Science, Washington State University, as a faculty member of the University of Guadeloupe in 1997 to 2002. and he was doing postdoctoral at Roslin Institute in UK, 1994 until 1997, and then as a faculty member at Nanjing Agricultural University in China since 1986 to 1997. So his research interest is focusing on the studying of the origin of phenotypes using state-of-the-art genomic and transcriptomic approaches in combination with bioinformatics and cell embryo biology. So it's very interesting field. So Professor Jiahua Jiang, sorry, you have 30 minutes to present your research, and we are looking forward for your great talk. Okay, thank you, please.
Can you hear me?
Yes.
Okay, wonderful. Just to let you know, we have kind of the heavy wind outside, or you hear some kind of background noise. Sorry about that. First of all, thank you so much, Professor Dr. Subantia. Hopefully I can pronounce your name correctly.
Just call me Siti.
Oh, okay, Siti.
Siti is easy.
Okay,
Yeah.
Professor Dr. Siti, for your nice introduction. I would also take this opportunity to thank the conference organizer, Dr. Bao, for inviting me to give a talk today. My topic today is biodiversity. Does RNA diversity matter? As you know, biodiversity has been a hot topic in biology for more than 35 years. Aims to study variety and variability of life on Earth. As you can see here, the research field mainly focus on three areas: genetic diversity, species, ecosystems. No doubt, the number of species on Earth is enormous, while the ecosystems are very, very complicated. So therefore, my talk today would focus on genetic diversity, particularly on RNA diversity.
The committee haven't presented the PowerPoint.
So can you see everything clear?
No,
okay.
We can't see it.
Can you see my slides?
I cannot see in my laptop.
This is clear in my.
Oh, sorry. What's wrong with
Can you see my Can you see my slides?
Yes.
Yes, we can.
Oh, sorry. I cannot
Okay.
see in my. Oh, sorry.
Oh, sorry about that. Okay, the question here is why RNA diversity? First, we can simply classify our human genome, for example, into the exonic, intronic, and intragenic regions. Based on ENCODE studies, it estimated that only 7% our human genome is silent. So that means that the remaining 93% of our human genome are actively transcribed, and associated with at least one primary transcript. As you can see here, that includes 1.2% expressed from protein coding exons, 39% derived from introns, 3 UTRs, and also 34% mapped outside of the protein coding genes. That's kind of one thing we need to know. Second, in addition to protein coding genes, you can see here there are several types of non-coding genes, such as snRNAs, RNAs, tRNAs, long non-coding RNAs, microRNAs, and pRNAs, for example. Third, you can see here a gene often use more than one transcript. And for example, alternative polyadenylation can occur at the tandem 3' UTR sites, alternative terminal exons, undronic sites, and exonic coding sequence size. So what's the consequence if you look at these, these alternative transcripts? For the first top two, you can see these two transcripts just have different lengths of 3 UTR regions, and they do code for the same proteins. That means no protein change for first two alternative transcripts. However, you can usually there are lots of regulatory elements residing in the 3' UTR regions. For this case, for example, and the short version of transcripts may lose the microRNA size, so the transcript should stay much longer than longer one. So from here, you can see, so-called quantitative effects when two transfers are used in the process. So again, if we look at bottom 3 alternative transcripts, they may code for different proteins. So these proteins may have a different property, such we'll see so-called qualitative effects on gene function. In some case, protein coding gene can be converted into non-coding gene. As you know, obviously, non-coding gene will execute epigenetic effects. So anyway, so you can see here when a gene uses different isoforms, it may functions quantitatively, qualitatively, or epigenetically. So anyway, so gene overall use of alternative transcripts makes a finite genome capable to coordinate an infinite phenome. Okay. Okay, somehow I got something here I could get fixed right away. So you can see this slide shows you a typical structure about mature RNA. So in this case, if you would like to profile a gene, everything from the 5' end to 3' end. So you can use RNA-seq, conventional RNA-seq, or iso-seq. However, if you would explore how alternative transcription start size or alternative polyadenylation size are used in response to different conditions. So you need to use either 5' end sequencing or 3' end sequencing methods. So here, I just show you our methods to profile alternative polyadenylation sites. That's called whole transform termini site sequencing, or just WTTS thing. As you can see here, our WTS-seq methods uses total RNA as starting materials. The assay involves fragmentation, poly-A RNA enrichment, first strand cDNA synthesis by reverse transcription, and the second strand cDNA synthesis by PCR. Actually, I want to tell you a few unique features about our WTTS-seq methods. So you can see here, actually, our WTTS-seq methods as both 5' adapter and 3' adapter simultaneously by during reverse transcription process. That's number one. Number two, as you can see here, poly-A RNA fragment are enriched using oligo GT25 bits, and also poly-A cDNA, okay, poly-A plus cDNA molecules are enriched by using RNAi and H treatment. Third one, where you can see our primers here, adapters. So basically, we use so-called poly-A anchored primers to do PCR. So that's avoiding any PCR amplification detour. Lastly, we try to avoid any over amplification. So basically, that helps us to maximize transcriptome coverage. Now, let me tell you a few advantages our WTTS-seq methods over the conventional RNA-seq method. So first one, you can see here, compared to RNA-seq methods, our WTTS-seq can directly measure the diversity and the dynamics of APA patterns. So you can see here we use the embryos at the stages 6, 8, 11, 15, and 28. Use of frog embryos, and the same samples derived at stage 6, 8, and 11 were also done used for RNA-seq analysis. So from the top part, you can see here you see 2 APA size. You can call them, one distal one, another one proximal one. So you can see at stage 6 and 8, distal APA site is more abundantly expressed than the proximal site. However, things are changing. So I would say, at 11 and 15, you can see the proximal site the proximal side is more abundantly expressed than the distal side. However, at the stage 28, looks like both sides are kind of balanced. However, if you look at RNA-seq, RNA-seq profiles, you cannot see any such kind of changes. Obviously, if you look at transcripts, there are pretty long ones. There are pretty, short ones. For example, so this is Xenopus trabeculus ribosomal protein like genes 34 L34 ribosomal protein L34. So this gene, okay, basically you look at express mature RNA. It has just 449 base pair, so it's pretty short. So you can see, look at the RNA-seq data. So that's kind of, obviously, that's not well presented in the data set. So basically, conventional RNA-seq is biased against 3' ends of transcripts. So you can not see clearly their expression levels. However, if we look at our WTTS-seq, and that's pretty clear, and the gene expression increases along these developmental stages. Now that in our genome, there are overlapping genes, our design is very unique, which our WTS-seq just a sequence from the polyasize. So that's kind of the strand-specific sequencing. So as you can see here, you can clearly assign reads to the overlapped genes based on the color. However, if you look at conventional RNA-seq data, and we have no clear way to assign reads to each gene. So as I said, our WTS-seq uses strand-specific sequencing, so that reads can assign to overlapping gene easily and clearly. And also, obviously, as you can see, APA size can be extended from exon regions to introns. They might be located in introns or expressed from the antisense strand. There are cases. So if you look at RNA-seq data, basically you have no way to identify them. If you look at our WTS-seq data, so that's pretty clear. For example, here is a GCP1A gene. As you can see, APA site is expressed at starting at stage 11, and that's pretty dominant at stage 28. Well, and here this slide shows you comparison of transcriptome distributions. As you can see, RNA-seq data produce pretty wider of the pretty wide wider distributions than the our WTS-seq. As a consequence, RNA-seq analysis would exaggerate the numbers of differentially expressed genes. So for RNA-seq data, you may see some false positive results. Here's the last one based on, so usually for RNA-seq data, and you need to have at least, RNA-seq requires at least 30 million reads per library. However, for our WTS-seq, usually we use, we just require 5 to 10 million reads. So overall, our technique will save some sequencing cost. Okay, now I would use a few examples to tell you some interesting story. The first one I want to show you the relationships from gene knockout, APA, and muscle mass changes. So here, as you know, MPK alpha 1 and MPK alpha 2 are two alpha subunits of adenosine monophosphate activated protein kinase, which serves as cellular sensors in regulation of many biological events. Here we investigate MPK, MPK R1 knockout and MPK alpha 2 knockout mice on muscle mass changes. So the knockout mice was produced by Violet and colleagues in 2003. If I remember correctly, the deleted exon 2 for each gene, and so these genes are located. AMPK R1 is located on chromosome 15, and AMPK R2 resides on chromosome number 4. So we used muscle samples to profile APA events. So as you can see, our analysis shows that down-regulated, you can see you can see upregulated and down-regulated genes, between two knockout mice. Actually, we use the wild type, as a control, as a control. So for the pathway analysis, so we used the either upregulated gene or downregulated gene for pathway analysis. At the end, we observed that downregulated APA size in AMPK alpha 1 knockout, okay, knockout, and are downregulated, while many genes are upregulated in alpha, MPK alpha 2 knockout mice. So at the end, you can see MPK alpha 1 knockout mouse have much smaller, muscle area, while MPK alpha 2 mice produce much larger muscle area. So kind of you can see here profiling APA size is a sensitive and a powerful approach that can be used to detail the information flows from a genome to transform to proteome and explain the altered phenotypes between knockout mice and wild type animals. So here I use frog, embryos and young males and growing females to tell you a story about development. APA.
Can you have slideshow, Professor Jan? Slideshow.
Sorry.
To have bigger view.
Okay. Yeah, thank you.
Yes, that's better.
Okay. Okay. So here you can see we use the embryos derived from, again, from 5 stages, 6, 8, 11, 15, and 28. So therefore, adults, so we have 2 stages, kind of young animals and growing animals. One question here, I put in a box here. So first question, are males and females genomically equal? That's kind of a question here. As you know, in mammals, males and females differ in sex chromosomes. Males have XY sex chromosomes, and the females have XX, sex chromosomes. As you know, in order to balance the dosage differences between sexes, one of the X chromosomes in females is inactive, but with 15 to 30% of its genes activated to counterpart the genes held by the Y chromosome in males. So I would say males and females are genomically equal. However, gender difference exists. Gender difference affects many different process. So as you can see from the top left side, so that basically tell you that's this that's kind of principal components analysis revealed kind of relationships among these samples, and based on color, you can clearly see where embryos are located, where the few young males and young females, growing males and growing females are located. Overall, you can see the APA chromosomes of young animals are more variable than growing adults. That's kind of one thing you can see there. And then you can see overall male frog APA chromosomes are more divergent than females. Yeah, you can see if we look at the 6 growing males and females, obviously 6 females are clustered, together, while you can see pretty separations among 6 male animals. Why? So here I just use one pathway to tell you the story. So if we look at the genes involved in sex differentiation, that's pretty interesting here. Do you see among 110 genes involved in sex differentiation, only 20 are common to both males and females. As you can see here, 6 specific genes are involved in male sex differentiation, while only 30 genes are specific to female sex differentiation. And I don't think I have time to go through pathways in details, but here you can see a story. You can look at a pathway, and I can, use the question here: why do females live longer than males generally? So also you can see a specific pathway for telomere maintenance. So you can see females have much better situation in telomere maintenance than males. So that I don't think you need me to explain further. So that tells a story why females lives longer than males. Well, so let me probably need to speed up a little bit. So here you can see kind of issue obesity is a huge issue in the United States. The current obesity rate is for 42% in the US. So here we look at APA events in response to high fat diet, and you can see here, high fat diet triggered more genes upregulated in hypothalamus. As a consequence, so these animals ate more food and grew more body weight. So here's is another interesting case about cannabis exposure, APA, and brain functions. As you know, marijuana use has been legalized in many states in the USA. And so based on kind of reports, and 48 million people used marijuana at least once in 2019. Okay, that's a big number. That's a big number. So we use a rat and also one-time exposure, that's acute exposure of cannabis for 10 minutes. So as you can see here, and hypothalamus responded to cannabis exposure pretty quickly. So as you can see, many genes were down-regulated in response to cannabis exposure. So if you look at the pathways, so basically cannabis exposure impairs learning, memory, mode, moment, sense, and thinking. That's pretty reflect what people feel when they use cannabis. This kind of last example, that's also a big issue in dairy. Okay, so that's try to tell you a story about pathogens, APA, and endometritis in dairy cows. Endometritis is one of the most devastating reproductive diseases in dairy cattle, resulting in annual loss of more than $50 million in the United States. So we screened dairy cows and collected samples from healthy and healthy and unhealthy cows. So basically, we look at here. We have collected 18 samples, and we could, based on APA profiles, we are able to classify them into clinically healthy animals and conditionally healthy dairy cows, clinically mild, mild endometritis cows, and clinically severe, severe endometritis case, as you can see here, and lots of pathways related to immune response are upper regulate. I want you to pay attention. So for healthy cow, you can see the pathway called psyllium moment is dominantly enriched. So in order to keep cow healthy, basically, psyllium moment function has to be normal. So anyway, these are kind of stories tell you how APA events in response to internal, external, and environmental challenges. So here is my kind of summary. And so if you look at genome annotation, so we know how many number of genes in our genomes. However, if we look at numbers of alternative transcripts, the number is much higher than number of genes. Based on our research, I can tell you that alternative transcripts rather than genes are minimal functional units expressed by genomes. So I guess use of alternative transcripts makes sense. Makes sense. If you look at so as I said, if you look at our phenome, okay, phenome, that's pretty involving, and you can see many phenomes. However, you look at our genomes, that's kind of the limited. So basically, use of alternative transcripts makes it possible for finite genome to coordinate infinite phenome. So overall, I would say alternative transcripts serve as bridges between genome and phenomes. And so if we want to understand, create, or maintain the biodiversity, so we need to pay attention to the use of alternative transcripts. The last things I would suggest here, hypothesis to be tested. Loss of iron diversity results in extinction of organisms. Thank you so much for your attention.
Thank you very much, Professor Xiang. So we have run out our time, and we will invite our second speaker, Mr. Zahir Ali, PhD. I think he's available now, and so we will have a question and answer after the second presenter finish his talk. Is it okay? Okay. So, Dr. Zahir Ali, please.
Hi.
Hello.
Can you hear me?
Yes. Okay, thank you.
Yep.
Okay, welcome, Dr. Zahir Ali. Yeah.
So, yep.
Can I
Zahir
introduce
Ali.
you? Yeah. Can I introduce you first? Okay.
Yes.
Mr. Zahir Ali, PhD. He got his PhD from Plant Molecular Biology and Biotechnology, Gyeongsang National University in South Korea, and then his master at Quaid-e Azam University, Islamabad in Pakistan, and then his PhD, his BSc was in the University of Peshawar, Pakistan. Okay, his work experience as a research scientist at KAUST, Saudi Arabia, and then postdoctoral fellow to 2013 to 2018, right? At KAUST, Saudi Arabia also, and then lecturer at Department of Biotechnology, University of Malakkat, Pakistan in 2003-2007. So he has very good experiences. So his research interest is in developing tools for plant genome engineering using specifically the CRISPR-Cas system. And similarly, he likes to explore plant potential to be used as biofactories for the production of various bioproducts, drugs of therapeutic importance. Okay, very nice. Please, Dr. Zahir Ali, you have 30 minutes for your presentation. Thank you.
Yep. Thank you. I will share my screen. Can you see my slides?
Yes, it's coming.
Yep. My name is Zahir Ali. Good morning. Here it's morning. I don't know what time is there. Anyhow, it's really a good opportunity for me, and especially Dr. Rekno. He told me to participate in this one, and I'm really thankful for this, making this event. So actually, I'm a plant genome engineering scientist. But as you know, the recent spread of the COVID, and actually the system was ready. We were using it for plants, but the system can be used for any pathogen or any virus. So we developed something also for the COVID-19. So I will present today our recent work, which is just accepted, and I will just present this work for this event. So the title is Bioscan. We call it Bioscan, a CRISPR-Cas9 based lateral flow assay for detection of pathogens. Why a Bioscan? Like, for example, as we know, the COVID-19 pandemic, it's just started like 2 years back, exactly 2 years back. And then here you can see that actually in October 2020, the cases were just 34 million. But just in 1 year, the cases become very high, and you can see like 234 millions were there in October, and confirmed it globally from this pandemic. In October, it was in October 2020, it was like 1.08, but in October 2021, it is 4.8 million. It's a huge, and still in some countries, still the lockdowns and all these things and all the lockdown measures are there, and still the people are suffering. But the main thing is like how to constrain a pandemic. So this was the call from World Health Organization, like they call it the 3T call. What is basically 3T call? It is like test, test, and test. So they just have like, they just say, we have a simple message for all countries, test, test, and test. Why test and test, test, and test? We make it a little bit more elaborate. Here you can see, like, for example, if there is a test that is affordable, fast, sensitive, and available to all the people, then you can trace. And if you trace, you can identify the cases and you can isolate the cases. And then if these are isolated and identified, then you can provide the treatment, supportive care, and in symptomatic cases. So the currently available method, which is normally used everywhere in the whole world, and it's a gold standard method, which is the qPCR method. What they do, like they just get the nasopharyngeal swab from the sample, and then they extract the RNA, and then they do the qPCR, and then they provide the results in the format of whatever. And here you can see that this is possible only in labs. It's not possible for individual. So what are the drawbacks of the RT-qPCR method? The main problem with this one, like you need a centralized lab where you need very specific designs and operations, and this is really a complex mechanism. And then you need the specialized people, like they should be trained very specifically for the qPCR run and for the result interpretation. Like the operators are not that much easy. Then the next one is like you have to purchase all the system, and as well as you have to hire the highly trained people, so which make it very costly. And normally, if you see the results are like from sample to the result outcome, there is a 24-hour gap, which is a very long time, especially in the case of pandemic like SARS-CoV-2. So what we think that a point of care, a simple test, are needed to accelerate the COVID-19 diagnostic, as we see very long lines when the people are going for the tests and all these things, even like by individual, or we see these lines everywhere when the pandemic started and when the pandemic was there. So these things, there is a huge need for point of care test to accelerate the COVID-19 diagnostic. An alternative should be there, which qPCR cannot do, because the qPCR, there is a limitation of the equipment, the labs, the people. So what we think, we want to develop a point of care diagnostic test. To start with, we look with the World Health Organization recommendation. They have an assured criteria for point of care diagnostics, which means it should be affordable to end user and affordable to health system. And S stands for like sensitive, avoid false negative, and the S specific, avoid false positives again. And user friendly. They should be simple. Like the qPCR, people cannot use by themselves. So they should be user friendly, and the people are maybe the simple clinics, they can detect the virus. It will be very easy, especially in pandemic cases. And they should be rapid and robust. Like the results you should get like 24 hours is a lot. The result should be get like within 1 hour or maximum by 2 hours. And equipment free, like there is like everywhere or every setup, or especially the developing countries, they don't have these sophisticated equipments and trained people. So the test should be equipment free, and it should be deliverable to the end user. So we are following this criteria to make a point of care diagnostic test. And if it's affordable, sensitive, specific, user friendly, and rapid, this can enhance the testing facilities for the pathogens, specifically the SARS-CoV-2. So to follow this one, recently the CRISPR-Cas system, which is immune system of the bacterial system, bacteria and archaea, this comes to the diagnostic, and this is called as like the next generation of point of care diagnostic tests. Basically, what is cluster regularly interspaced sharp palindromic repeats? This is actually the immune response of the bacteria against viruses. So what happened in the bacteria is basically like when the virus attack the bacteria, it send its genome into the to the bacterial cell. And what happened in the bacterial genome? The bacterial genome have a repetitive sequence. That's why it's called like clustered regularly interspaced. So it's like there is a cluster which is regularly interspaced. So there is specific region that are repeated again and again, and there are specific protein related to these sequences, which is the case nucleases. So what happened when the virus is there, the bacterial cell is playing a very good play with the virus. It's just take a small piece of the virus, use it as a memory and keep it in the genome. So when next time the virus come, it have a memory for this virus. So in the next step, this is the immunization against the virus. Like the bacterial cell keep a specific sequence as memory against this virus. So in the next step, when the virus come, like next time the virus attack, so here this sequence is expressed as single guide RNA, which is the memory of the virus. And this protein is produced, is sgRNA, and this protein make the complex, and it will cut the virus genome. So the virus cannot attack. So this is really a good mechanism which is present in the bacterial system and archaea system. And then this system is the targeting of the virus. And this system was recently adopted for genome engineering, because you need only simple sequence of the gRNA, and then you can cut, or then you can use this system for guiding this protein to any nucleic acid. So the system is very simple, and it can be engineered very well for genome targeting, for the virus targeting, or for genome engineering in any species, like mammalian cells, plant cells, bacterial cells, virus, whatever, for the virus resistance or pathogen resistance, and even diagnostics. So we want to take advantage of this simplicity. Like if we have a gRNA and if we have the Cas protein, we can use this for the detection of the virus as well. Not genome engineering and not virus resistance, but for the detection of the viruses. So to do that, if we look, like recently there are thousands of publications, which show like how the Cas9 system is used for genome editing. It's need very simple. The requirements are very simple. You just need a 20 nucleotide sequence of the genome, which is followed by the 3 nucleotides like NGG. This is called as the PALM sequence. And this, if you have a 20 nucleotide followed by the 3 NGG, then you can target this sequence. You just design RNA, which is complementary to this target sequence, and then your Cas9 will make a complex. And then if you have this one, then there will be a cut in the genome. And once the genome is cutted, then because there is a random mutagenesis from here, and then there will be insertion and deletion in the genome, or the gene will be knocked down. It can be used for the repair as well if you provide the repair template. It can be also used for diagnostics, because here you can bind your protein to a specific sequence of the nucleic acid. So the system is very simple and it can be adopted. But how to adopt this system for engineering of a diagnostic test? For that, we develop a mechanism which we call the BioScan, and that is a CRISPR-Cas9 based lateral flow assay for detection of pathogens. And what is BioScan? What is our hypothesis and what are the components of the BioScan? It's very simple. As we know, the lateral flow assay, which is commercially available, it have mainly 3 regions. Like, for example, it have the gold nanoparticles, which is conjugated with the antiform antibody. It have a streptavidin strip, or the streptavidin is impregnated into the lateral flow. And another line, which is called as control, is the antiform antibody. So this is the lateral flow strips, which is commonly available, commercially available, and it can be used. So we think that these commercial strips, they can be used for the SARS-CoV-2 detection very easily. But what we need a dCas9, which should be biotin leveled, because we know that biotin can interact with the streptavidin. So if this is the case, so we have the biotin and we have streptavidin. So this biotin can localize the Cas protein here at this region. And then if this is the case, then we can design a gRNA, which is complementary to the SARS-CoV-2 genome. And then the complex will be localized on the streptavidin strip. And this guide RNA we can easily make in vitro. This is just like you have a DNA sequence, then you can do the in vitro transcription. And if you produce the protein, then it can be used with the lateral flow strip. And if this is possible, you just need a simple heat block, or even water bath can be used with a specific temperature. Because we don't need any amplification, any PCR amplification. It can be coupled with the isothermal amplification. And we just need the simple pipettes, or the simple and tubes and strips. So with this whole setup, you can do very easily the test for the SARS-CoV-2 or any pathogen. So next, we produced like how to produce the BioScan reagent. So we have a system where we put our dCas9 with AV tag, and we have in the same cassette, in the same plasmid, we have another protein which is responsible for biotinylating this AV tag. So the biotin is there in the system, in the bacterial cell, and then we can purify the bio-dCas9. So to confirm, we induce the system and then we produce these proteins. So here you can see that the size of the dCas9 is like 180 kDa, and here we can see very clearly that yes, the protein is produced. But the next question is like, is it biotinylated? So we use the Western blot and using the anti-biotin antibody. So using the anti-biotin antibody, here we confirm that yes, this protein is biotinylated. So if this is the case, then we can produce the gRNA, which will guide us to the SARS-CoV-2 genome or the SARS-CoV-2 nucleic acid. So we did the in vitro transcription. Now we have all the components, like the guiding sequence to the SARS-CoV-2 genome, and we have the BioDKS9, and we have the lateral flow strip. So the whole concept or the working principle is that actually if you have the farm level primers, then you can amplify, first you can use the reverse transcription to convert the SARS-CoV-2 RNA into DNA, and then by the isothermal amplification, you can produce the DNA in 10 to 15 minutes in a single isothermal temperature, no need of any PCR. So we are using RPA method to amplify the DNA. And once this DNA is there, so we have a guide sequence to this DNA, and this is coming from the SARS-CoV. This DNA is made from the SARS-CoV, which is complementary here. So the complex will come here, and once this complex is on the DNA, in the next step, the complex will be bound with the gRNA and with the Cas9. And then we have the ends as farm level, and where we have the anti-farm antibody, which is coupled with the gold nanoparticles. So this whole complex will be made. And the next step, because we are using the lateral flow strip, so if you put this complex on the sample pad, it will flow through, and if you have the SARS-CoV-2 genome or the SARS-CoV-2 is there in the sample, it will be stuck here because of the biotin-streptavidin interaction. But if not, it will flow through. So here is just a representation of the positive results. So in positive result, you can see on the test band that the appearance of the color band is here because the fragment will be accumulated or localized on this side. And some of them, like for example, the remaining nanoparticles with the gold and the form, they will move here. But in the negative, there is no band appearance here because this complex is not there, the SARS-CoV-2 is not there, and here on the test line, you will see nothing. So to follow this one, we have a simple sketch that you just collect the saliva from a patient. It can be done with the saliva or nasopharyngeal, but saliva is much more easy and that you can just spit in a tube. And the next step, a simple RNA extraction protocol we developed. It's a 5 minutes RNA extraction protocol. And then you can incubate at 42 degrees C for RT-RPA, which will convert the RNA into DNA and also amplify the DNA at 42 degrees C. So we don't need a PCR, but we just need a simple heat block or a water bath. And here you are using the anti-farm, the farm labeled primers. So the DNA will be labeled with the farm. And then we are providing this complex. So this complex will be made in the tube, like the BioDKS9, sgRNA, and farm level DNA complex. So then this complex is applied to the sample pad, and the lateral flow will show you that you have a positive result or you have a negative result. So following this simple procedure, we try to develop our system for the SARS-CoV detection. So like this is just the general image, and here is the real result. We select two targets, like target 1 and target 2. And when we are using the specific gRNA, we can detect the target. But when we are using a non-specific gRNA, there is no target detection. Means our system is working very properly for target 1, and then we confirm with the target 2, another nucleic acid sequence. But the next question is like the different controls. So like, for example, no SARS-CoV-2, no gRNA, no protein, and also the different like SARS-CoV-2 genome and the influenza virus genome. So here you can see that only the SARS-CoV-2 is detected, but all other negative control, they are clean on the test line. So means our test is very specific only for SARS-CoV-2 because of the complementarity of the sgRNA that we are using, the specific sgRNA for SARS-CoV-2. So To confirm that this can be used for the SARS-CoV-2, we select different region. Like, for example, we want to amplify a short sequence from the SARS-CoV-2 genome. Here, this is the genome of the SARS-CoV-2, where we have ORF1A, ORF1B, and the spike protein, the envelope protein, membrane protein, and engine. So we select different region, like one in each gene. And here you can see that when we were amplifying these regions, two regions, which is like number 8, which is here from the ORFB, and engine, which is this one, the set one. Here they are amplifying, or we are detecting these regions very well. Comparatively, these other regions, they were not that much efficient. And the main reason is like the primer binding. So the primer binding is very well at these two regions, but maybe the primers are not binding, or the efficiency of the primer or amplification is not that much good. So then we just follow only with this specific set of the primer to follow it for the detection of the SARS-CoV-2. But one of the main question is like, what is the sensitivity of this test? Because the CDC, they provide a CT value that only this much virus should be detected by a point of care test. So we put different amount of the copy number, like for example, 0 copy number of the SARS-CoV-2, 0.4 copy number of the SARS-CoV-2, 2 copy number of the SARS-CoV-2, 4 copy number of the SARS-CoV-2 genome, and 20 copy, 40 copy, and 400 copy. So here you can see our system is very sensitive, and even 4 copies of the SARS-CoV-2, like generally one microliter contain more than 1,000 copies in the patients. But here you can see that our system is very sensitive, and we can detect 4 copies per microliter very easily. This is just the conversion of this figure into the graphical format. So this shows that our system is detecting it very well. Then we validate our system with the clinical sample. So we collect like up to 94 sample from the clinical patients. And here you can see that on the test line, most of these clinical patients we can detect. Only 3 samples, like sample number 54, 69, and 73, they were not detectable. And these are the negative control, are the people who were recovered from the SARS-CoV-2 and they don't have the SARS-CoV-2 anymore. Next is the question, like there are different variants of the SARS-CoV-2. So is it possible that we can detect the SARS-CoV-2 variants? But these variants, as we know, that these are just point mutants. And the total system is dependent on the gRNA sequence. So let's say, for example, alpha variants have a deletion of 2 amino acids, 69 and 70. So if you design an sgRNA for this sequence, only the mutant, the alpha mutant, but not the wild type. So this can easily detect, or you can predict the prevalence of any specific virus or specific strain present in a specific region. And you can follow the distribution or the prevalence of the specific variants. So we detect, like for example, the alpha, beta, delta, and one of them is common mutation in all of them. And then we confirm that our system can specifically detect the alpha, beta sequence and delta sequence very well. So next, we confirm that like the next one of the biggest requirement of the point of care is it should be compatible with point of care, like specifically at room temperature or 4 degrees C, because at point of care, like for airports, stadiums, you cannot use -20 and -80 for storage. So we confirm that can our system work at room temperature, and can we store the reagent for a long time at these 2 temperatures specifically? For that, we confirm that yes, the system is working. And also the quick extraction of RNA, like for example, can we extract the DNA in 5 minutes? And yes, so we here, you can see that the system, when they are stored for weeks, like for example, the components, you can see like for first week, second week, third week, and fourth week, you can see the reagents are still working and they can be used very well at room temperature. There is no need of these storage conditions. And here you can see that when we are extracting the RNA by our method in 5 minutes, the system is working. So as a conclusion, I will say that bioscan reagents can be easily produced in the lab because all the system you can make easily in any lab, in any molecular biology lab. But this is just the reagent production needs the lab. Other things don't need the lab. Bioscan is comparable in sensitivity with the commercial RT-qPCR because the sensitivity is very high. We see like 95% sensitivity. Bioscan doesn't require sophisticated lab equipments, no need of lab equipments, and it can be done with the simple point of care setup. The other thing is like Bioscan follow the World Health Organization assured criteria. It's affordable, sensitive, specific, user-friendly, rapid and robust, and it's equipment free. And you can deliver it into the users. And we are trying to develop it for like personal use, like if it's available in market, in clinics, so people can do it by themselves. No need to go to the labs. And bioscan detection assay can be performed in less than 1 hour. So like, for example, you collect the sample and then you treat the sample and then you got your results within an hour. No need to wait for like these 24 hours and also wait for the main labs. So this is our team, the bioscan team. This is Professor Magdi Mahfouz. He is our principal investigator. And Shiva, Nurhan, Ahmad, and Ten and Rashid. This is the bioscan team, and this picture was taken when everything was locked down and only these 7 people were allowed to come to the university. So I like this picture a lot. And thanks for your attention and questions.
Okay, thank you very much. Great talk. So can we have applause for Professor Ji Hua Jiang and Dr. Zahir Ali? Thank you. And now we have about 15 minutes, even 16 minutes to have some question from the audience. You can raise your hand, or you also can write your questions in the chat box. Okay. Anyone? Any questions? Hello? No question?
Hello, can I ask? Hello? Hello, can I ask question?
Yes, please.
Okay, hello, Prof. Zahir. Okay, I'm Pir Muhammad from Malaysia. So thank you for your presentations now. Okay, I just started to learn about CRISPR. Okay, so I just want to ask your your opinion based on your experience. Okay, I feel to design the gRNA is the guide DNA is one of the most critical component of a successful CRISPR. So based on your experience, what's your advice to design a good guide DNA?
Basically, like the gRNA design is not a big problem at all. It's very simple. You just need the NGG sequence, like any like AGG, GGG, CGG, or like you just need NGG sequence and then 20 nucleotide. The only thing, like all the gRNAs, they are not working equally. So you have to search out in your gene which gRNA is working better. You can select the top strand, you can select the lower strand. And the sequence is the same sequence as you are targeting. So because you will design, it will be in vitro transcribed or the system transcribed, and it will become complementary to the lower strand. So like the design is very simple. Specifically, if you are designing for Cas9, the Cas9 one is very specific and very simple compared to the Cas12 or the Cas13 and 14 systems. The Cas9 one is very simple and it's very widely used. So you just need 3 nucleotide, which is NGG sequence. 20 nucleotide, you can select any sequence. And NGG, the presence of 2 Gs in the genome, it's very common. So any target can be designed. The system is very simple. Like simplicity is there, and it's really robust. The Cas9 is really working very well.
Okay.
Thank you.
Okay. Thank you, Dr. Zahir. Anyone else?
Actually, I have a question for Dr. Zahir. And what's the cost for one test?
Okay. So actually, the good thing is like point of care. Again, there is one thing is affordability. Like it should be affordable. So like if you look at the lateral flow strip, so the lateral flow strip is the main cost. It's $3. The rest of the reagent we can produce in the lab, and like for example, once you in vitro transcribe it, you can like one in vitro transcription is enough for 3,000 samples. And one production of the the DCAS9, biotin DCAS9 is enough for 10,000. So what we are expecting, it should be under $10. So this is what we are expecting, that the people can afford by themselves, like not by the governments or not. So it should be available in clinics, just like a normal test, like a pregnancy test. So the people can just take it and use it by themselves.
Great. Okay. Any more questions? We still have enough time. Come on. Okay, I would like to have a little question for Professor Chihua Jiang. So on your APA analysis, you in that case, when we would like to use something like antisense for stopping or knock down any unfavorable gene, maybe for the pathogenesis or whatever for the pathogen, how can we have the benefit from your APA pattern?
So basically, you can use our WGD-seq to profile APA changes in response to internal, external, and universal challenges. So that's kind of the tool to look at the kind of mechanisms involving relationships from genome to phenome. Basically, if you look at kind of for biodiversity research, in the past, probably many people have focused on DNA, variation. However, RNA, variation has not paid lots of attention. So obviously, and we have not thought about to use APA to do kind of the pathogen, diagnosis. However, basically, I just show you a few examples, for disease, so we can see, but I couldn't have time to explain everything in details. However, our technique can be used to study kind of the progression of diseases. In human, like cancer is a huge issue. Obviously, cancer is not a kind of one-day event. That takes time to develop. And so like from, something, you start something there and to the kind of pretty, kind of level 4 stage something, there should be lots of changes. So basically, we can use this sensitive tool to monitor kind of the progression of diseases or defects of some disorder.
Okay, thank you. Very nice.
Yeah.
From the audience, if there is any question, we still have some time. No more? No more? For, oh yes, question for Professor Silva. How to maximize the transcriptome coverage? Primers and adapter are designed for that.
Okay, that's a great question. That's a great question. When we started to develop our methods, we have tried different primer combinations, and so I mentioned that in my talk that we use so-called poly-anchored primers to avoid PCR amplification detour. That's the thing can help us to maximize the transcriptome coverage. That's kind of one thing you can do. Next one, try to use less cycles of PCR amplification. So as you know, so for our genome, we just have two copies, have everything. However, for transcriptome analysis, things are not equal. Some genes are expressed abundantly. Some genes are rarely expressed. So if you do many cycles of PCR amplification, that will favor abundantly expressed, APA size, not a rarely, expressed APA size. So that's the thing we need to pay attention. So in terms of protocol, it can be used for plants. I will say yes. Use of alternative polyadylation is an evolutionary conserved event from animals to plants. Yeah, you can see lots of papers published using plant as materials. So yes, our assay will work on plants. So that's not a question. If you have RNA, to the RNA as materials, just use. Also depends on your kind of sequencing platform. So at the moment, our assay is based on ion torrent sequencing platform, not Illumina. So kind of tricky issue associated with the Illumina platform, because Illumina platform requires so-called diversity libraries. So look at our assay, so we have, our reads begins with several T's. So that's also, these are not a diversity library. So that's kind of a unique library. So Illumina system cannot process reads for you. So that's a problem. That's why we use ion torrent system. However, not a problem. We can adjust the primer design and see. So one day definitely we can use Illumina system to do APA profiling. So that's not a problem. Even bacteria, fungi, I guess many organisms use alternative polyadylation. So definitely these methods can be used for different organisms.
Okay, thank you. So we have still only 4 minutes left. If there is one more question, it's okay. No more? Okay. if there is no more questions, so thank you very much for Professor Xia Hua Jing and Mr. Zahir Ali, PhD, for your very interesting talk, and hopefully we can have more communication outside there. Yeah, okay, thank you very much, and give applause for
thank you.
them.
It's my pleasure. Thank you.
Thank you.
Yeah.
Bye.
Thank you.
Okay. I send it to the committee.
Okay, thank you, Professor Siti, and also Professor Zihua Jiang and Dr. Zahir Ali for a very insightful session. Ladies and gentlemen, next we'll have a photo session with all of the participants. So if you could please kindly open your camera in Zoom so we could take a picture together. Okay, so the picture will be taken by the host, and there are 5 slides. Okay, so are we ready? On the count of 3, let me help for the first slides. Give your best smile to the camera. 1, 2, 3. The next slide. 1, 2, 3. The third slide. 1, 2, 3. The fourth slide. 1, 2, 3. And the last slide. 1, 2, 3. Thank you for your participation. Thank you.
Okay.
All right. So ladies and gentlemen, we would like to remind you to please kindly fill out the attendee list of the first session that has been informed in the chat. For the next agenda is Principle and Mechanism of Biotechnology Product Session 2. We will hear presentation from Mr. Pandu from PT Bioscience with the topic Next Generation Sequencing to Advance Microbiology Study in Indonesia. Mr. Pandu, the next 30 minutes is yours. You may proceed.
Yeah. Hi everyone, thank you for the time. Okay, let me share my screen first, and then please, I want to talk in Indonesia. Okay, here. Pertama-tama, terima kasih kepada Bapak Ibu yang sudah menyempatkan hadir dalam acara ISIBIO kali ini. Izinkan saya memperkenalkan diri terlebih dahulu. Saya Safi Triani, merupakan Senior Field Application Scientist dari PT Pandu Bioscience, di mana PT Pandu Bioscience sendiri merupakan eksklusif distributor dari Next Generation Sequencing, yaitu Illumina System. Seperti yang telah disebutkan, kita akan membahas mengenai Next Generation Sequencing to Advance Microbiology Study in Indonesia. Oke, jadi di sini kita juga memiliki untuk giveaway untuk para partisipan yang hadir di dalam sini. Saya memberikan waktu kepada Bapak Ibu untuk bisa melakukan scan QR code-nya atau bisa langsung hubungi langsung di link yang sudah disediakan di layar Bapak Ibu sekalian untuk mendapatkan giveaway. Adapun giveaway-nya terdiri dari wireless charger, flash disk, and many more. Ini dia. Merupakan PT Pandu Bioscience. Untuk PT Pandu Bioscience sendiri berlokasi di Jalan Raya Pondok Gede nomor 10B, Duku Kramat Jati, di mana untuk informasi lebih lanjut mengenai PT Pandu Bioscience itu bisa diakses melalui info@bioscience.com atau di www.bioscience.com. Untuk produk dari PT Pandu Bioscience sendiri, kita merupakan eksklusif distributor dari Next Generation Sequencing, terutama di Illumina System, seperti yang sudah Bapak Ibu ketahui sebelumnya. Lalu kemudian kita selain ada Illumina, kita juga memiliki Beckman Coulter untuk terkait dari liquid handling automation, dan ada beberapa dari molecular region juga terkait dari kit ekstraksi maupun kit PCR lainnya. Dan yang terakhir itu juga kita ada 10x genomic untuk buat profiling single cell. Lalu selain itu juga kita ada beberapa brand yang kita distributor, yaitu ada Verogen untuk buat forensic NGS solution, ada Shimatsu, proteomic analysis, Major Science, General Lab Instrument, dan juga kita merupakan kerjasama dengan IDT, Archer, dan CARDX untuk LATP. Apa yang kita sediakan untuk buat solusi dari Bapak Ibu ketika ingin melakukan research terkait apalagi terhadap dari biologi molekuler. Banyak dari
produk bidang keilmuan yang sudah kita bisa cover, baik dari oncology, reproductive health, genetic disease, microbiology, agriculture, dan molecular. Yes, yang mana saja yang bisa di-cover, yaitu sudah bisa di-cover dari universitas, dari academic research center, pharmaceutical company, genome center, biotechnology, hospital, dan sampai ke government. Nah, ini dia. Jadi topik yang akan kita bahas adalah bagaimana NGS memberikan solusi untuk di bidang mikrobiologi tentunya, yaitu adalah salah satunya dengan menggunakan metode whole genome sequencing, atau kita sebut biasanya familiar dan disebut total genome organism, yaitu salah satunya adalah dengan menggunakan culture based whole genome sequencing. Nah, ini salah satu contoh yang diambil dari Journal of Clinical Microbiology pada tahun 2018. Salah satu paper yang dibuat ini adalah ketika melakukan whole genome sequencing dengan menggunakan culture base untuk buat bakteri Clostridium difficile, yang dimana untuk dari sini kita bisa melihat bahwa terdapat analisa pohon filogenetik yang mana menghasilkan adanya kesimpulan, yaitu adanya transmisi dari AMR genes atau antimicrobial resistant di antara farm animal and human melalui dari bakteri Clostridium difficile ini. Nah, untuk teknologi ini total whole genome sequencing lah yang menjadi solusi ketika Bapak Ibu ingin melakukan tracking adanya AMR genes. Nah, ketika melakukan culturing atau melakukan kultur untuk melakukan whole genome sequencing, apa sih konsiderasi untuk buat bakteri dan viral whole genome sequencing? Untuk bakteri sendiri, kita memastikan apakah kultur kita pure monoisolate atau tidak. Lalu bagaimana genome size-nya? Lalu pastikan kalau ketika kita melakukan kultur, dia 100% target organism. Lalu kita lihat di bagian virus. Ketika di bagian virus, kita lihat apakah dia berupa RNA atau DNA, apakah dia berupa single-stranded DNA atau double-stranded DNA, lalu apakah dia bentuknya host atau culture medium dari DNA atau RNA. Jadi kita bisa mengetahui dari sini, ketika kita mengetahui apakah dia pure isolate atau tidak, kita bisa melakukan metode selanjutnya untuk bisa mendapatkan hasil yang memuaskan, seperti itu. Nah, ini dia salah satu pipeline atau salah satu metode ketika Bapak Ibu ingin melakukan next generation sequencing dengan menggunakan Illumina system, yaitu kita bisa lihat di sini ada isolate dari virus, lalu kita bisa bagi, yaitu apakah virus tersebut berupa RNA atau berupa DNA. Untuk DNA, balik lagi, tadi ada single strand atau double stranded. Nah, dari sini kita bisa lihat untuk buat RNA virus, kita perlu melakukan reverse transkriptase terlebih dahulu. Setelah dapat dari si DNA-nya, lalu kita bisa melakukan preparasi. Adapun tahap preparasinya dengan menggunakan kit Illumina, yaitu bisa menggunakan Illumina DNA Prep, lalu instrumen yang digunakan pilihannya antara NextSeq atau MySeq, lalu dilakukan data analisis. Untuk buat DNA-nya juga sama. Pertama, kita bisa melakukan ekstraksi terlebih dahulu untuk bisa mendapatkan material genetik berupa DNA. Terus kemudian kita bisa melakukan preparasi seperti pada RNA, yaitu dengan menggunakan Illumina DNA Prep. Lalu instrumen yang dipilih yang ingin dilakukan apa, lalu dilakukan data analisis. Nah, ini dia library preparation atau kit yang digunakan untuk buat melakukan whole genome sequencing dengan menggunakan Illumina system, yaitu dengan kita sebut kitnya yaitu adalah Illumina DNA Prep dengan metode fragmentation atau pemotongan. Karena Illumina mengusungkan teknologi yang disebut short read sequencing atau pembacaan dengan jarak pendek seperti itu, maka dibutuhkan pemotongan terlebih dahulu. Nah, untuk itu, tahapan pemotongannya itu dilakukan dengan menggunakan enzim transposom. Illumina DNA Prep ini memiliki teknologi yang disebut sebagai bit link transposom, jadi transposomnya itu terlink dengan bits, sehingga hasil pemotongannya lebih seragam dan memudahkan para peneliti untuk melakukan data analisis untuk selanjutnya. Jadi Illumina DNA Prep bersifat versatil, robust, jadi mudah digunakan, reproduktif, dan fleksibel untuk buat berbagai jenis aplikasi yang akan digunakan. Nah, ini prosesnya, jadi berupa hasil dari genom DNA yang dihasilkan baik dari blood, saliva, atau dari genom DNA hasil ekstraksi, kemudian input DNA-nya kemudian akan dipotong, lalu setelah dipotong baru kita lakukan preparasi untuk kemudian dilakukan sequencing. Seperti itu. Nah, untuk buat berapa sih input requirement-nya ketika melakukan next generation sequencing ini, itu dibutuhkan konsentrasi sekitar 1-500 nanogram, jadi sangat fleksibel sekali ketika Bapak Ibu memiliki material DNA yang sangat kecil sampai yang besar, baik dari culture maupun unculture, seperti itu. Lalu hasilnya nanti ketika di akhir tidak perlu melakukan normalisasi atau penyetaraan konsentrasi, karena di dalam teknologinya, di dalam library preparation ini, konsentrasinya sudah disetarakan. Sehingga nanti proporsi setiap sampel ketika dilakukan sequencing itu akan seimbang. Dan hasilnya akan lebih baik. Jadi ketika melakukan studi mikrobiologi, salah satunya adalah dengan menggunakan teknologi yang disebut sebagai, atau metode disebut sebagai metagenomic. Nah, untuk metagenomic sendiri, pembaginya ada 2, di mana kita mengetahui yang disebut pertama adalah targeted sequencing dan shotgun metagenomic. Untuk targeted sequencing, itu jadi kita mengaplikasi terlebih dahulu untuk target amplikonnya, itu ada disesuaikan dengan studi yang ingin dilakukan, salah satunya adalah 12S, apakah 16S, CO1, atau 18S, dan ITS untuk fungi. Lalu untuk buat shotgun metagenomic, kita melakukan sequencing untuk seluruh DNA yang kita dapat. Jadi total genom DNA dari lingkungan yang kita ekstraksi, terus kemudian langsung kita lakukan sequencing, baru lakukan analisa untuk selanjutnya. Nah, bagaimana dengan 16S metagenomic sendiri? 16S metagenomic banyak familiar digunakan dengan menggunakan instrumen yang disebut sebagai MySeq system. Sudah ada protokol dari ilmiah sendiri untuk terkait dari 16S metagenomics itu sendiri. Nah, dilakukan sequencing dengan menggunakan 500 cycle, atau kita biasa lakukan dengan 2 kali 250 sampai 2 kali 300 cycle, di mana untuk regionnya kita ketahui untuk 16S sendiri memiliki panjang 1500 base pair, yang mana untuk regionnya, untuk wilayahnya itu kita hanya mengamplifikasi di versi 3 dari gene 16S, seperti itu. Lalu kemudian dilakukan analisa data. Untuk analisa datanya itu bisa menggunakan MySeq Reporter atau longkaran manager dari instrumen. Jadi di dalam instrumen Illumina itu sudah diinputkan dengan bioinformatics software. Bapak Ibu bisa langsung mendapatkan data ketika melakukan sequencing, atau bisa menggunakan Base Pair Sequence Hub, yang mana Base Pair Sequence Hub ini merupakan software analisa data berbasis cloud. Jadi dibutuhkan akses internet untuk buat melakukan analisa data itu sendiri. Untuk protokolnya sudah disediakan oleh Illumina, tinggal mengikuti prosedur yang ada. Nah, ini bagaimana workflow-nya ketika melakukan 16S RNA sequencing, yaitu dilakukan sampel prep terlebih dahulu untuk bisa mendapatkan total genom dari lingkungan. Lalu setelah kita mendapatkan total genom dari lingkungan, kita hanya mengamplifikasi di version 3 sampai version 4 region amplification. Jadi panjang base pair-nya hanya sekitar 300 sampai 600 base pair. Lalu kemudian baru dilakukan tahap preparasi. Nah, setelah dilakukan preparasi, indeks yang digunakan itu disebut sebagai indeks Nextera dari Illumina. Lalu dilakukan sequencing menggunakan MySeq system seperti ini penampakannya. Lalu dilakukan analisa data dengan menggunakan Base Pair Sequence Hub. Adapun softwarenya yang bisa digunakan atau aplikasinya yaitu Kraken atau 16S Metagenomic. Hasilnya ini merupakan tampilan salah satu ketika Bapak Ibu melakukan analisa sistem S metagenomic, kita bisa mendapatkan profil mikrobiota dari lingkungan atau dari sampel-sampel yang dilakukan untuk buat sistem S metagenomic. Jadi memudahkan para peneliti untuk bisa melakukan profiling bakteria, sudah bisa mendapatkan komposisi bakterianya dan sudah bisa mendapatkan dari taksonomi atau klasifikasi golongan spesiesnya seperti itu. Nah, jadi ketika kita melakukan sistem S amplikan metagenomic, kita harus mengetahui apa saja sih keterbatasan ketika kita melakukan studi seperti ini, bahwa akan ada selalu kemungkinan sequencing error di dalamnya. sehingga membutuhkan optimasi atau melakukan pengulangan ketika melakukan sequencing seperti itu. Kita ingin mengetahui apakah komposisi di dalam lingkungan tersebut sudah sesuai atau tidak dengan komposisi bakteri yang kita temukan. Lalu adanya keterbatasan dalam melihat species level identification seperti itu. Terus kita juga bisa melihat kalau misalkan abundance atau komposisi berapa persen rich yang didapatkan ketika melakukan sistem S metagenomic ini dia bersifat relatif, jadi tidak bersifat absolut. Lalu ada kemungkinan dia menghasilkan PCR bias. Jadi bagaimana sih untuk bisa melakukan sistem S atau melakukan metagenomic, seberapa dalam kita bisa melakukan sequencingnya, bagaimana saya menentukan reach ketika kita ingin melakukan sistem S ataupun shotgun metagenomic, itu tergantung dari jenis sampel yang akan kita lakukan ataupun juga dari komposisi sampelnya seperti apa. Lalu kemudian kita juga bisa melakukan hipotesis terlebih dahulu, bisa mengetahui apakah kita ingin mengetahui di lingkungan tersebut harusnya sudah ada komposisi apa saja di bakterinya di dalam lingkungan. Lalu kemudian kita juga bisa melihat apakah reach tersebut cukup atau tidak. Di sini sudah ada beberapa paper yang menstudikan mengenai berapa sih jumlah reach yang dibutuhkan ketika kita ingin melakukan sistem S maupun dari shotgun metagenomic itu sendiri. Nah, selain itu, di sini juga metode yang bisa dilakukan dengan menggunakan metagenomic adalah untuk fungi metagenomic, yaitu dengan menggunakan reagent ITS. Nah, untuk Illumina ITS protokolnya, ini merupakan pipeline yang dilakukan dari collecting extract sample, lalu dilakukan library prep reagent yang bisa digunakan itu sebenarnya bebas, bisa dari manapun. Nanti yang penting cocok untuk di mesinnya, salah satunya adalah kita juga menyediakan dari Illumina, lalu baru dilakukan organism test, terus dilakukan workflow-nya, di sini juga sudah kita sediakan protokol untuk melakukan ITS metagenomic, lalu dilakukan normalisasi atau dilakukan wet lab, lalu setelah selesai preparasi kita melakukan sequencing dari instrumen yang paling kecil itu ada ISIC sampai yang paling besar itu Novasic, lalu dilakukan report dengan menggunakan base pair sequence hub, yaitu aplikasinya dengan menggunakan
Mr. Sapriani, are you here with us? Or that it seems like the connection from Mr. Sapriani is rather bad. So perhaps another person from Pandu Bioscience, if you could please continue with the presentation, or while we're waiting for Mrs. Sapriani to join us. For Mrs. Aulia from Pandu Bioscience, are you here with us? Okay. Mrs. Safitriani, seems like she already joined us. It's still unmute, Mrs. Safitriani.
Yeah, sorry for the interruption, so I can continue my presentation.
Yes, please proceed. Thank you.
Okay. Ya, terima kasih Bapak Ibu. Mohon maaf atas gangguannya. Jadi seperti yang sudah kita jelaskan di sini sudah ada gambaran mengenai bagaimana para peneliti melakukan ITS metagenomic seperti itu. Bisa terlihat dengan menggunakan ITS metagenomic pada base pair sequence hub, kita bisa mendapatkan profil dari fungi yang sudah dihasilkan dari suatu sampel lingkungan. Lalu kita bisa melihat komposisi atau seberapa banyak fungi yang bisa yang terdeteksi ketika kita melakukan ITS metagenomic ini. Nah, ini untuk buletinnya. Jadi kita di Illumina juga memiliki application note terkait dari bagaimana profile dari fungal sequencing and classification with the ITS metagenomic, bagaimana prosesnya, terus kemudian bagaimana juga tahapannya dari awal sampai akhir, terus hasilnya seperti apa. Nah, selain itu juga dari tadi kalau misalnya kita bahas mengenai ITS dan Cysteine S, maka di sini juga ada yang disebut sebagai shotgun metagenomic. Jadi kalau misalkan untuk buat 16S dan ITS, itu kita hanya mendapatkan amplikonnya. Jadi prinsipnya kita mengamplifikasi terlebih dahulu region amplikon yang ingin kita dapatkan, terus kemudian baru kita melakukan sequencing atau NGS. Sedangkan kalau shotgun, hasil sampel dari lingkungan, kemudian kita lakukan sequencing untuk seluruh total genom di dalam lingkungan tersebut. Jadi kita misalkan dari soil atau dari tanah, kita ambil, kita ekstraksi, terus kemudian total genom dari tanah, kita langsung bisa melakukan preparasi dengan menggunakan kit dari Illumina, baru kita bisa mendapatkan seluruh informasi terkait genom-genom apa saja yang didapat di dalam suatu lingkungan tersebut. Seperti itu. Nah, ini dia keuntungan dengan menggunakan shotgun metagenomic. Keuntungannya adalah bahwa shotgun metagenomic ini culture independent metodologi, jadi kita tidak perlu melakukan culturing terlebih dahulu. Kalau misalkan saya mau ingin mengetahui bakteri apa saja yang didapat di dalam suatu lingkungan, kita harus culture terlebih dahulu, lalu kita pisahkan setiap organisme di dalamnya dengan menggunakan shotgun. Kita tidak perlu melakukan culturing. Dia juga relative abundance of microbial taxa seperti yang pada Cysteine S metagenomic ataupun ITS. Metagenotyping artinya kita juga bisa melihat ada mengidentifikasi adanya gene loss, gain atau mutation di antara population cell atau microbial species. Terus kemudian kita bisa melihat dia hipotesis free testing. Jadi kita bisa mengetahui seluruh informasi di dalam sampel tersebut, apakah mikroorganisme apa saja di dalam sampel tersebut, apakah mikroorganismenya terdapat archaea, bakteri ataupun yang lainnya gitu, apakah sudah sesuai atau tidak, lalu bisa dilakukan pathogen detection, lalu juga bisa menghasilkan kelebihan-kelebihan lainnya. Nah, ini dia untuk buat application note dari shotgun metagenomic. Untuk kit yang digunakan adalah yang disebut sebagai Next-DNA Flex atau kita sebut sebagai sekarang Illumina DNA Prep, di mana untuk Illumina DNA Prep ini bisa melakukan preparasi untuk buat shotgun metagenomic dengan cara tadi total genom yang sudah kita dapatkan langsung dilakukan pemotongan. Setelah dilakukan pemotongan, tambahkan indeks atau ditambahkan barcode, baru kita lakukan sequencing. Hasilnya itu bisa kita lihat bahwa dengan menggunakan NGS Illumina, hasilnya terdapat keakurasian data maupun klasifikasi untuk bisa melihat adanya perbedaan komposisi diversitas antar sampel di dalam sampel tanah. Jadi efisien dan komprehensif workflow untuk melakukan shotgun metagenomic dengan menggunakan Illumina system memudahkan para peneliti untuk mendapatkan informasi terkait taksonomi, diversity comparison, juga bisa memungkinkan untuk dilakukan de novo assembly. Jadi kita bisa salah satunya bisa mendapatkan total genome baik dari organisme-organisme yang terdeteksi di dalam suatu sampel lingkungan, lalu dilakukan functional profiling. Nah, untuk aplikasi-aplikasi atau software bioinformatik yang sudah kita gunakan, itu sudah ada berbagai macam dan disolusikan dalam base-based sequence hub. Nah, ini salah satunya yang kita lakukan dengan menggunakan shotgun metagenomic, yaitu adalah dengan menggunakan sampel tanah, soil metagenomic. Ini bekerja sama dengan Perkin Elmer, dilakukan fully automated DNA extraction dengan Semagic 360 instrumen, DNA soil kit, lalu dilakukan tadi menggunakan Illumina DNA Prep atau teknologi yang disebut sebagai bit-link transposon, dilakukan sequencingnya dengan menggunakan Nexus system dan analisisnya berupa Kraken. Yang kita lihat di sini, ketika Bapak Ibu melakukan shotgun metagenomic, maka visualisasi hasil akhirnya bisa kita lihat seperti ini. Jadi dia diklasifikasi, kita bisa lihat komposisinya Di sini sudah ada organisme, lalu di sini sudah bisa dibagi organisme apa saja yang terdeteksi dalam sampel tanah tersebut, seperti itu. Hasil akhir solusi dari slide saya yang ingin sampaikan adalah untuk microbiology workflow solution dari NGS itu sendiri, bahwa ketika kita melakukan studi mikroorganisme dengan menggunakan NGS, kita bisa melakukan berbagai pipeline yang dilakukan, yaitu salah satunya untuk mendapatkan apakah kita ingin melakukan total genome sequencing, apakah ingin melakukan targeted sequencing atau S DNA sequencing, atau ingin melakukan shotgun metagenomic. Tadi shotgun metagenomic jadi mengambil semua informasi di dalam dalam satu sampel, lalu kita melakukan sequencing, lalu dilakukan targeted sequencing virology untuk virus, atau dilakukan RNA sequencing. Hasil akhirnya material genetik yang kita dapatkan, terus kemudian kita lakukan amplifikasi ketika pipeline-nya berupa targeted metagenomic atau targeted amplicon. Terus kemudian setelah didapatkan target DNA yang kita inginkan, lalu dilakukan preparasi libraries, di mana untuk library preparasinya itu juga disediakan oleh Illumina. Salah satunya adalah Nextera XT ataupun Illumina DNA Prep. Ada dilakukan sequencing, instrumen paling kecil yaitu adalah ISIC, terus kemudian yang di sini yang paling populer yang digunakan di Indonesia salah satunya ada MySic system, lalu kemudian dilakukan data analisis dengan menggunakan base-pair sequence hub dengan aplikasi-aplikasi yang disesuaikan dengan metode yang digunakan. Itu dia untuk metode dari NGS itu sendiri. Adapun kita mendistribusikan backman culture. Untuk backman culture-nya merupakan produk yang digunakan untuk genomic solution. Jadi kita memprovide para peneliti untuk melakukan ekstraksi kit dan juga untuk buat purifikasi. Di sini backman culture memiliki beberapa jenis produk, yaitu salah satunya DNA isolation kit, ada RNA isolation kit, atau bisa akses di link yang sudah disediakan di layar Bapak Ibu. Adapun nanti untuk buat pemenangnya akan diinformasikan kepada tim kami. Demikian presentasi dari saya. Jika ada pertanyaan, dipersilahkan. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
I apologize for the connection problem. So for Mrs. Safitriani, is your presentation is done? Okay, it seems that you're still muted.
Oh, sorry. Yeah, I ended my presentation and thank you for the attention.
Okay, thank you, Mrs. Safitriani from Pandu Biosense. I'm sorry for the previous mistakes. Ladies and gentlemen, before continuing to the next agenda, there would be some documentary videos from our partnership that we can watch. So let us watch the video together.
Halo, kami dari PT ITS Science Indonesia. Hari ini kami akan memberikan penjelasan singkat mengenai pengujian porcine. Mengapa perlu melakukan pengujian porcine pada produk pharmaceutical? Beberapa macam bahan baku atau obat, termasuk tablet, kapsul, injeksi, dan sebagainya, dapat berasal dari produk hewani atau turunannya. Contohnya seperti gelatin, yang sering dipakai sebagai bahan utama kapsul atau stabilizer pada vaksin. Selain itu, antikoagulan seperti heparin juga dapat berasal dari porcine. Sedangkan berdasarkan Undang-Undang Nomor 33 Tahun 2014 tentang jaminan produk halal, produk yang masuk, beredar, dan diperdagangkan di wilayah Indonesia wajib bersertifikat halal. Oke, jadi bahan apa saja yang dapat mengandung porcine? Kebanyakan senyawa tersebut merupakan antikoagulan, digestive supplement seperti enzim lipase, amilase, insulin, dan sebagainya. Wah, cukup banyak ya. Tapi tenang saja, pengujian porsain ini tidak sulit kok. Ada beberapa metode yang dapat digunakan untuk mengecek kandungan porsain pada produk. Yang pertama, lateral flow device atau test pack. Metode ini cepat dan mudah, tanpa perlu banyak peralatan laboratorium yang canggih, tetapi sensitivitasnya rendah. Metode 2, ELISA. Waktu preparasinya dibutuhkan 2-3 jam. Preparasinya cukup mudah dan dapat menguji dalam jumlah banyak dalam satu kali running. Dibandingkan lateral flow device, ELISA lebih spesifik, tetapi masih memiliki batas deteksi sensitivitas yang sedang. Metode 3, real-time PCR. Metode ini adalah golden standard untuk pengujian porsain pada produk pharmaceutical dengan limit deteksi yang sangat rendah 1-5 kopi DNA. Pengujian porsain pada produk pharmaceutical sebaiknya dilakukan dengan metode PCR karena bahan baku yang diuji telah melalui proses pemurnian atau pemanasan tinggi sehingga kemungkinan yang tersisa hanya materi genetiknya saja. Selanjutnya, tahap real-time PCR ada 3 yaitu preparasi sampel, ekstraksi DNA, dan amplifikasi dengan menggunakan mesin real-time PCR. Untuk lebih jelasnya, mari melihat alat real-time PCR dari Roche, Light Cycler 96. Alat berkapasitas 96 well ini memiliki 4 channel fluorescence yang dapat digunakan sekaligus dalam satu kali pembacaan. Sebelum memulai proses PCR, profil temperatur dan protokol PCR dimasukkan ke dalam mesin. Setelah itu, tekan tombol start. Secara perlahan akan terbentuk grafik sinyal seperti ini. Sinyal ini terbentuk dari proses denaturasi, annealing, dan elongasi yang berulang, sehingga fluorofor yang terkandung di dalam master mix dapat berpendat dan tertangkap oleh sensor mesin. Sinyal yang didapat kemudian dianalisis menggunakan software Light Cycler, sehingga terlihat sampel mana yang positif atau negatif. Demikian penjelasan singkat mengenai pengujian porsain dari kami. Sekian dan terima kasih.
Halo, kali ini saya mau memperkenalkan alat laboratorium dari Syngene. Ingenious Tree. Sebelum pengenalan alat, apakah Anda pernah mengalami kesulitan saat mendokumentasikan hasil gel elektroforesis atau plot Anda di ruang gelap? Sistem dokumentasi gel secara manual terdiri dari ruang gelap, kamera, dan transluminator sederhana. Dokumentasi gel secara manual seperti ini kadang menyulitkan dan belum lagi kalau hasil foto gel yang didapat ternyata blur. Dengan Ingenious Tree, Anda dapat memilih pencahayaan sinar UV, sinar biru, atau putih sesuai dengan pewarna yang Anda gunakan. Selain itu, software Genesis akan membantu Anda memilih pengaturan cahaya dan filter yang tepat sehingga sangat memudahkan untuk mendokumentasikan gel fluorescence ethidium bromida atau cybersafe DNA atau protein gel dengan composite blue atau silver staining. Untuk penggunaan mode otomatis, Anda hanya perlu memilih tipe gel yang sesuai, kemudian software akan secara otomatis mengatur aperture, zoom, dan fokus serta posisi gel yang sesuai sehingga gambar gel terekam secara optimum. Software Genesis dilengkapi dengan database, tipe gel, dan pewarna yang tersedia di pasaran. Jadi jangan khawatir tidak bisa menemukan pengaturan yang sesuai. Sangat mudah. Hubungi tim ITS Science Indonesia untuk info lebih lanjut. Terima kasih. Stay safe.
Since 2019, PT Wadia Prima Mulia has continued to develop in meeting the needs of basic laboratories for Indonesian territories. Until now, our company has been trusted as an authorized distributor for 11 companies that produce the best laboratory equipment in the world. Our highly skilled and experienced sales team spread across Indonesia from Medan, Jakarta, Bandung, Semarang, and Surabaya. Our products. services, including application and technical support, installation and training, also service and maintenance.
In the life sciences, researchers are actively pursuing studies aimed at understanding life, the causes of disease, and achieving breakthroughs in regenerative medicine. To observe and accurately measure intracellular processes and structures, researchers need a microscope that combines sensitivity and speed. Increasingly, scientists require the capability to view cells and tissue at high resolution, as well as their larger context with macro to micro capability. All of these advanced features must be easy to use so that even novice users can acquire high-quality data quickly and simply. Olympus created the FV3000 series to satisfy these needs. This next generation of confocal laser scanning microscopes combines leading-edge imaging features with renowned Olympus optics.
Microtomy requires my full attention and dedication to make sure that I deliver the perfect slides. And with the Tissue Tech Auto section, I started getting the job done easier, faster, and with better quality. The automated block alignment makes sure precious tissue is preserved optimally. It is also a great help for making recuts, while the perfect alignment helps me produce even more slides. Trimming and sectioning are done with high precision, each section the same exact thickness. Thanks to the easy operation of the auto section, every technician in our laboratory is quickly up and running. Whatever the need, we are all confident to deliver the perfect slide each time. My name is Mark Shanti, and I'm empowered by Sakura.
All right, ladies and gentlemen, for the next, we will hear a presentation from Mrs. Sri Hayuni from Biotech Merck on end-to-end solutions for biopharmaceutical workflow. And for Mrs. Sri Hayuni, the next 30 minutes is yours. You may proceed.
Hello, good afternoon, everyone. My name is Sri Hayuni, and I'm going to share my screens first before I start my presentations. Here you go. All right. So first of all, I'm going to apologize because I'm going to deliver these presentations in Bahasa to make everyone in this room would understand and better about my my presentation. So allow me to deliver this presentation in Bahasa. Selamat siang semuanya, perkenalkan nama saya Sri Hayuni. Terima kasih untuk kesempatan yang diberikan kepada saya mewakili PT Merck Indonesia untuk mempresentasikan topik berjudul End-to-End Solution for Biopharmaceutical Workflow. Di sini presentasi ini akan saya saya coba. Just give me a second. I think I have a technical problem. Okay, just sorry. It seems like the PPT is closing. I'm going to reshare again. So sorry about this technical inconvenience. Okay. Oh, just give me a minute.
Do you want me
to
the
have
end to end? End to end.
Yeah.
Yeah.
It's about to. Okay, cool. All right, cool. That's my share screen.
Yes.
Can you see my?
Can.
Okay, thank you.
Yep.
Okay, cool. Yeah. Sorry for this inconvenience, everyone. So yes, I'm just starting my presentation again. Jadi presentasi ini akan dibawakan oleh saya Sri Hayuni M Biotech selaku Cell Culture Manager dari tim research PT Merck Chemicals and Life Science. Baik, sebelum saya memulai presentasi, saya ingin memperkenalkan sedikit mengenai portfolio PT Merck Life Science yang saat ini sangat ingin kita branding sebagai Life Science Company setelah mengakuisisi beberapa brand seperti Sigma Aldrich. Di kita sendiri ada beberapa divisi, yang pertama healthcare, kemudian performance material, dan life science. Khusus kebutuhan laboratorium Bapak Ibu, baik itu dari akademi maupun juga industrial, produk yang akan kita usung kebanyakan berasal dari industri life science bisnis di Merck sendiri, di mana di sini saya dan juga beberapa tim saya akan mendukung Bapak Ibu untuk kebutuhan laboratorium dan juga penelitiannya. Khusus yang healthcare, biasanya ini lebih ke arah obat-obatan yang sudah memiliki hak paten, dan khusus untuk performance materials, industri ini bergerak di bidang-bidang yang lebih ke arah coating, kemudian juga beberapa elektronik yang mendukung teknologi seperti OLED. Nah, untuk bisnis life science sendiri, di sini kita ada 6 brand utama yang biasanya sering didengar oleh Bapak dan Ibu. Yang pertama, yang paling utama kali ini adalah Sigma Aldrich. Jadi ini sudah diakuisisi lebih dari 3 tahun yang lalu, dan produk kita ke arah life science lebih banyak diusung dari brand Sigma Aldrich ini sendiri. Nah, kemudian di kita juga ada Miliki, di Miliki biasanya ini mensupport lab water solution, baik itu di laboratorium maupun juga industri. Kemudian juga brand yang sudah cukup lama, sudah mengakar di PT Merck sendiri ini adalah Supelco Analytical Product. Jadi biasanya memang chemical classics. Kemudian juga kita ada Millipore. Kalau Millipore ini dia sudah lama kita akuisisi under brand of Merck. Dan untuk Millipore sendiri ini produk-produknya lebih ke arah preparation and separations. Kemudian juga kalau untuk media-media bakteri sebagai kisi gitu ya, itu lebih banyak brandnya ke arah Millipore. Nah, yang terakhir ini kita ada SAFC dan Bioreliance, di mana kedua brand ini lebih ke arah mendukung raw materials untuk industri yang dilakukan oleh customer-customer kita. SAFC mungkin banyak yang berhubungan dengan biopharmaceutical juga, dan Bioreliance ini mungkin lebih ke arah biopharmaceutical yang mengarah ke biologics maupun juga vaccines yang rekombinan, di mana nanti mungkin kalau misalnya Bapak dan Ibu ada kebutuhan untuk engineer cells untuk menghasilkan protein tertentu atau antibody, monoklonal antibody, kita memiliki tim yang khusus untuk memberikan paten engineer cell line yang atau stable clone yang bisa memproduksi produk biologics ataupun biosimilar yang Bapak Ibu ingin produksi. Baik, without further ado, I will present the portfolio that we can support for the biopharmaceutical. Herein saya ingin memperkenalkan sedikit produk biopharmaceutical atau industri biopharmaceutical apa saja yang bisa kita support. Jadi dimulai dari compound atau industri yang biologics yang menghasilkan compound, ini dia small molecule, biasanya dia lebih kecil dari 1 kilodalton. Ini merupakan salah satu industri biopharmaceutical yang saat ini sedang sangat booming juga. Salah satu produk yang terkenal mungkin Lipitor dari Pfizer ya untuk high kolesterol. Kemudian juga new biopharmaceutical seperti monoklonal antibody MAB, ini dia berupa therapeutic protein. Biasanya untuk ukuran dari monoklonal antibody MAB ini sendiri berkisar dari 25 sampai 800 kilodalton. Dan ini salah satu produk yang dari Merck sendiri di life science-nya sudah luncurkan adalah Keytruda, ya ini berupa checkpoint inhibitor for cancer. Kemudian juga saat ini biopharmaceutical lebih ke arah vaksin, ya, terutama untuk SARS-CoV-2, di mana vaksin yang di-manufacture juga bukan hanya berasal dari inactivated gitu ya atau live attenuated vaccines, tapi sudah lebih ke arah mRNA, kemudian juga peptide. Bukan peptide vaccines, tapi kayak therapeutic atau rekombinan vaccines gitu, sehingga untuk bisnis-bisnis biopharmaceutical ini kita memfokuskan beberapa produk yang ingin di-highlight untuk kebutuhannya. Nah, di bisnis kita sendiri karena kita memiliki 6 brand pilar yang menjadi dasar dari bisnis ini, maka dari mulai end to end, dari mulai R&D sampai biopharmaceuticalnya kita dapat dukung Bapak dan Ibu. Nah, secara umum untuk prosesnya sendiri kita akan membagi industri biopharmaceutical ini menjadi 3 kelompok, yaitu di bagian discovery, development, dan manufacturing. Nah, untuk produk-produk yang bisa kita support mungkin saya compactkan dalam slide saya di slide yang ini dan juga slide-slide berikutnya, di mana produk-produk highlight yang pertama dari untuk stages discovery sendiri mungkin ada beberapa stages ya, biasanya berupa disease target identification, kemudian identify dan isolate target yang sesuai, kemudian candidate designs, jadi termasuk PEG glycylation dan sebagainya ini masuk ke sini. Kemudian di karakterisasi ini juga masih dalam discovery step by step, dan ini merupakan produk yang biasanya kita dukung. Jadi untuk discovery sendiri tahapan awal yang paling sangat sederhana itu adalah teknik culture cell, di mana di sini Sigma Aldrich sendiri sudah sangat sudah sangat populer, sudah sangat tua, berakar gitu untuk mensupport produk-produk yang berhubungan dengan culture cell. Jadi dimulai dari koleksi-koleksi sel lainnya. Jadi di sini kita untuk collection of the cell line itself, we have a variety of brands. For instance, for cell line we have ECAC cell line. Kemudian untuk primary cell we have cell application inks. Kemudian in millipore we usually support for primary and also stem cell. Nah, kemudian untuk teknologi terbaru juga ada induced pluripotent stem cell. Di sini kita biasanya membawakan atau mendeliver sel yang berasal dari ABISC. Di sini induced pluripotent stem cell yang sudah yang didistribusikan di sini merupakan stabil dari iPSC yang berasal dari donor-donor dan dimodifikasi dari lab-lab di berbagai belahan dunia. Nah, selain sel line, kita juga memiliki sera, media, antibiotik, growth factor, freezing medium, the cell disattachment factor yang sangat mendukung proses biopharmaceutical Bapak dan Ibu. Di sini mungkin saya highlight saja produknya, jadi dimulai dari yang FBS. Kalau FBS sendiri, Sigma Aldrich sudah mendeliver FBS yang memiliki GMP certified, kemudian juga untuk proses biopharmaceutical sendiri karena memang sangat rentan ya terhadap virus, kemudian juga other kontaminan yang bisa merubah atau mempengaruhi end product dari biologik itu sendiri, biasanya sangat rentan untuk virus, sehingga untuk FBS yang kita sarankan untuk biopharmaceutical itu adalah yang sudah gamma irradiated. Jadi kalau sudah gamma irradiated biasanya dia sudah eliminates virus-virus yang biasanya menginfeksi bovine itu sendiri. Jadi ada yang GMP manufacture dan gamma irradiated khusus untuk industri biopharmaceutical. Nah, kemudian juga karena industri biopharmaceutical ini nantinya akan dipakai oleh manusia, tentunya di Indonesia sangat concern dengan isu halal. Nah, untuk cell disassociation factornya sendiri biasanya menggunakan trypsin, di mana trypsin tersebut mengandung kandungan porsin, sehingga untuk tahapan awalnya kita sarankan tidak menggunakan trypsin gitu, pencacah sel yang berasal dari porsin. Di sini produk kita sendiri ada yang disebut sebagai trypzean. Jadi trypzean ini merupakan trypsin yang diambil dari gene pankreas bovine yang diekspresikan di jagung gitu. Untuk hasil disattachment-nya sendiri bila dibandingkan dengan kompetitor, trypsin memiliki kekuatan cell disattachment atau enzim yang lebih kuat dibandingkan dengan other competitor, sehingga sangat convenience untuk kebutuhan biopharmaceutical Bapak dan Ibu. Nah, terakhir untuk cell freezing mediumnya sendiri, tentunya proses freezing merupakan hal yang paling krusial ya untuk di industri biopharmaceutical. Nah, di sini kita biasanya mensarankan cryostore. Cryostore ini adalah jenis cryopreservasi yang sudah ready to use, sehingga nanti pada perlu mix lagi ya serum, kemudian medianya untuk memfreezing selnya. Di cryostore ini sendiri dia sudah ready to use, dan rate of survival sel ketika dibangunkan jauh lebih tinggi bila dibandingkan dengan kompetitor atau produk lainnya. Nah, untuk produk-produk ini juga media, kemudian juga cell disassociation factor, kemudian cell freezing mediumnya juga ada yang serum free. Jadi kalau misalnya Bapak dan Ibu ingin melakukan ingin melakukan production biopharmaceutical product yang bebas dari animal komponen, dari awal kita sebenarnya sudah ada beberapa produk yang mendukung serum free environment untuk perkembangan kultur pertama atau stabil sel yang ingin Bapak Ibu kembangkan di laboratorium. Nah, selanjutnya tidak kalah penting juga adalah filtranya. Di sini kita bisa bawa millipore, di sini ada Stericup, kemudian juga beberapa syringe filtrasi yang biasanya digunakan dalam praktik kultur sel untuk menghindari kontaminasi. Terakhir, untuk labware alliance-nya sendiri, dari kita memiliki brand partner premium dari Corning, kemudian juga dari brand sendiri, di mana kedua brand ini bisa mendukung konsistensi hasil dan juga pyrogenik atau endotoxin free untuk alat-alat kultur atau consumable yang biasa Bapak Ibu gunakan di laboratorium masing-masing. Nah, selanjutnya biasanya selain dimulai in vitronya dari kultur sel, mungkin juga untuk studi tertentu akan ada asai yang lebih advance berupa 3D cell culture. Jadi bukan sekarang ini apalagi yang lagi happening bukan hanya cell culture yang biasa yang ditempelkan di plastik, tapi juga ada yang 3D cell culture. Nah, kita juga sangat support untuk kebutuhan Bapak dan Ibu mengenai 3D cell culture, di mana seperti yang kita ketahui 3D cell culture membuat asai-asai atau studi yang Bapak Ibu lakukan untuk sel, misalnya untuk penyerapan obat atau mungkin untuk bahkan untuk menghasilkan organ seperti kolon atau alveolus gitu ya untuk SARS-CoV-2. Nah, ini biasanya didukung dengan 3D cell culture technology. Nah, di kita ada 2 produk unggulan yang biasa kita sering sarankan gitu ya untuk studi disease modeling. Yang tertarik untuk 3D, yang pertama hydrogel. Jadi kalau di kita itu ada hydrogel yang sering digunakan itu adalah yang max gel sama yang ECM gel. Jadi berdasarkan dari testimoni beberapa customer yang sudah menggunakan ini dengan menggunakan max gel dan ECM gel ini dapat meningkatkan proliferasi dari selnya sendiri, juga hasilnya sangat konsisten dan sangat bagus untuk studi disease modeling yang 3D. Selain hydrogel, kita juga memiliki 3D organoids portfolio di mana Bapak dan Ibu bisa memimik organ gitu, jadi mengembangkan sel yang biasanya berasal dari induced pluripotent stem cell untuk dikembangkan menjadi organ. Jadi kalau yang ini adalah gambar 3D organoid berupa kolon, sehingga ketika Bapak dan Ibu berhasil mengembangkannya, kalau untuk studi absorption obat gitu, dia lebih memimik in vivo dibandingkan dengan hanya 2D cell culture teknik yang biasa dikembangkan di lab. Nah, selanjutnya dalam proses discovery juga nanti akan ada teknik di mana kita harus memodify selnya atau ya gene editing. Di sini saya ingin highlight gene editing kita berupa CRISPR di Merck sendiri. CRISPR gene editing memiliki portfolio yang cukup lengkap, saya bisa infokan seperti itu. Contohnya seperti gRNA atau guide RNA, kemudian Cas9. Di guide RNA sendiri kita memiliki guide RNA yang hanya berupa protein maupun juga guide RNA yang sudah melekat pada plasmid. Selain itu kita juga memiliki sintetik gRNA atau guide RNA yang memiliki efektivitas lebih tinggi dengan gRNA yang biasa. Nah, untuk Cas9 sendiri, untuk enzimnya sendiri gitu, Cas9-nya sendiri juga sudah ada yang tercantum di dalam plasmid maupun juga sudah dalam vektor berupa lentiviral. Nah, produk unggulan dari Cas9 kita ada yang Cas9 GFP protein. Di sini adalah Cas9 yang sudah track melalui GFP, sehingga nanti ketika si Cas9 ini melekat dengan target binding yang berada di sel kita, maka dia akan menghasilkan fluorescence bahkan sebelum transfeksi, karena kalau biasanya menunggu transfeksi membutuhkan waktu yang cukup lama gitu ya, beberapa hari. Nah, ketika si Cas9 ini sudah bertemu dengan meminimalisir waktu kita untuk melihat apakah transfeksi kita ini berhasil apa enggak. Kemudian juga kita memilih Bapak dan
It seems like there's a trouble from the presenter. We're waiting for presenters perhaps to wrap up the presentation because there's 1 minute left.
Sorry, how long is the presentation for this, Mark?
1 minute left.
1 minute left. Okay.
Oh, I'm sorry, 10 minutes left.
Yeah, so sorry about the connection. I'm going to reshare my screen again. Share screen, yeah. Okay, where were we? Okay. So I'm talking about the CRISPR before. Jadi kita memiliki custom oligos. Selanjutnya untuk produk highlight CRISPR ini sendiri kita memiliki all-in-one CRISPR plasmid di mana Bapak dan Ibu sudah memiliki vektor plasmid yang di dalamnya sudah terdapat Cas9 dan juga guide RNA. Jadi ini sudah all-in-one, Bapak dan Ibu tidak perlu lagi khawatir untuk satu persatu masukin kemudian mix. Nah, di CRISPR ini sendiri apabila sudah oke nih bagian gRNA dan Cas9-nya, nanti mungkin perlu melakukan transfeksi gitu ya, transduksi. Nah, di sini saya menyarankan extreme gene 36 di sini. Mana extreme gene 36 ini merupakan reagen transfection yang khusus untuk melakukan plasmid DNA dan sangat cocok untuk aplikasi CRISPR atau Cas9 yang akan dilakukan oleh Bapak dan Ibu. Highlight produk ini sendiri adalah performance-nya yang bagus sehingga memiliki higher transfeksi dibandingkan dengan produk sejenisnya, kemudian juga low cytotoxicity, dan yang paling penting itu dia free dari animal derived component, sehingga apabila Bapak dan Ibu ingin melanjutkan ke biopharmaceutical downstream, dia jadi lebih aman untuk menggunakan extreme gene 360 ini. Nah, lanjut masih ke arah discovery yang nyerempet-nyerempet sedikit ke development. Setelah melakukan teknik berupa gene editing, tentunya kita perlu menganalisis. Nah, jadi ini ada beberapa produk analisis yang bisa kita dukung untuk kebutuhan Bapak dan Ibu. Jadi mungkin yang paling gampang kita bisa melakukan immunocytochemistry, yaitu berupa antibody ya, fluorescent antibody. Nah, antibody ini sendiri kita juga ada edisi yang jadi kalau untuk Zoom-Mac ini sendiri dia merupakan antibody yang bisa tahan di room temperatur gitu, sangat bagus. Nah, kemudian juga memiliki sangat banyak varian gitu di portfolio produknya sendiri. Dan ini merupakan rekombinan dari interspecies gitu ya, tapi bisa men-tailor atau menyesuaikan dengan kebutuhan antibody yang ingin Bapak Ibu gunakan di lab. Nah, selain itu kita juga memiliki live cell imaging reagents gitu. Jadi untuk beberapa reagen yang mendetek perubahan variabel berupa warna, kita memiliki beberapa kit reagensnya. Kemudian juga untuk analisis proteinnya atau antigennya kita bisa menggunakan SMC X-Pro dan juga Milliplex Kits untuk produk-produk Luminex. Jadi kita juga mendeliver produk-produk Luminex. Nah, selain antigen, di sini juga ada sel asik onyx, di mana ini merupakan live imaging dari sel yang biasanya kita gunakan untuk studi disease modeling. Kemudian juga ada beberapa cellular assay dan juga Scepter 3.0. Nah, cellular assay ini berupa kit ready to use, yang di sini MAK kit ready to use khusus untuk beberapa analit yang biasanya digunakan Bapak dan Ibu untuk penyakit tertentu. Jadi misalnya Bapak dan Ibu tertarik untuk sedang melakukan penelitian onkologi. Nah, kami akan dengan senang hati memberikan brosur MAK Kits karena biasanya dalam brosur ini akan berisikan komprehensif analit apa yang biasanya digunakan untuk onkologi gitu, maupun dan juga penyakit lainnya. Jadi di sini hanya gambarannya kecil saja, cuman kita memiliki beberapa penyakit yang sering diteliti oleh peneliti secara globally dan juga di Indonesia. Kemudian bahkan ada juga analit khusus untuk drug discovery ya, seperti NAHDS atau mungkin untuk melihat senyawa metabolit tertentu gitu bisa dengan menggunakan ingin lebih ke arah protein gitu ya, misalnya melakukan penelitian berupa monoklonal antibody. Di sini memiliki beberapa workflow berupa ekstraksi dan purifikasi, konsentrasi sampai analisis western blot-nya kita memiliki produknya. Untuk ekstraksi produk yang saya highlight di sini selitik kits, di mana selitik ini ekstraksinya lebih aman, lebih tanpa merusak selnya sehingga nanti hasilnya akan lebih. Kemudian untuk purifikasinya kita juga memiliki produk concentrate protein. Nah, mungkin yang saya ingin tampilkan di sini adalah Emicon state cell, di mana di sini kita bisa mengkonsentrasikan protein dalam jumlah yang lebih banyak. Dan yang terakhir untuk analisisnya itu ada western blotting ya. Kalau untuk western blotting itu ya, tapi kita punya alat berupa Snap ID2. Jadi di sini biasanya western blotting kegiatan Bapak dan Ibu bisa sampai 4 sampai 24 jam, ini bisa disingkat saja dalam waktu 30 menit gitu. Karena teknologinya bisa menggunakan vakum. Nah, terakhir untuk gel pre-cake, jadi kita ada true page di mana dalam untuk true page ini sendiri Bapak dan Ibu tidak perlu lagi membuat gel pre-cake sendiri, sudah ready to use dan diklaim 10 kali lebih stronger atau lebih kuat dibandingkan precast gel yang tersedia di market. Nah, selanjutnya juga mungkin ini highlight beberapa teknologi saja gitu yang kita punya. Kita juga punya automated 2D electrophoresis di mana di sini apabila menggunakan SDS biasa ya ini mungkin cuma bandnya hanya segini, dengan menggunakan automated 2D electrophoresis Bapak dan Ibu bisa melakukan analisis SDSP secara otomatis. Jadi tidak perlu tambah-tambah reagennya secara manual tapi langsung menggunakan alat. Dan yang terpenting dari sini adalah analisis atau band separation yang biasanya di SDS page itu hanya dalam turun ke bawah gitu dia molecular weight. Kalau di sini dia sudah menggunakan PA dan molecular weight. Jadi seandainya Bapak dan Ibu memiliki protein yang biasanya berada dalam satu band yang sama dengan menggunakan PI atau kinetik, nanti dia akan ke arah samping dulu pemisahan separationnya lalu ke arah bawah sehingga nanti untuk protein-protein yang jumlahnya sedikit atau protein yang memiliki band bersama, nanti separationnya akan kelihatan lebih jelas dengan menggunakan automated 2D electrophoresis yang kita punya. Kemudian juga untuk analisis antigen tadi sudah saya sebutkan, di sini kita bisa ada SMC X Pro, ini merupakan ultra sensitivity di mana bisa mendeteksi sensitivitas antigen sampai dengan femogram. Kemudian untuk Luminex platformnya sendiri, gitu dia menggunakan Luminex di Taylor di support Luminex dan SMC X Pro. Nah, yang terakhir untuk teknologi yang ingin detection yang ingin saya
It seems like there's a problem with Mrs. Sri Hayuni.
Plate-nya iya, sudah waktunya habis ya Mbak ya?
No, you still have 3 minutes left.
Okay,
the
thank
connection
you for
is
the confirmation.
Yes, please proceed.
Okay, thank you. Okay, so the last part I think I was just summarize. Jadi untuk manufacturing-nya sendiri kita juga memiliki banyak produk yang bisa mensupport. Jadi biasanya kalau untuk manufacturing sudah masuk ke tim Bioreliance, tapi untuk beberapa reagen kimianya, misalnya untuk vaksin maupun juga biologics lainnya gitu ya, biosimilar lainnya membutuhkan preservasi, kemudian surfactant, adjuvant, antibiotik, stabilizer, dan acidity regulators. Ini merupakan portfolio produk yang kita miliki. Silahkan bila Bapak dan Ibu tertarik bisa mendiskusikannya dengan saya sendiri ataupun tim Merck area Bapak dan Ibu untuk mendapatkan konsultasi yang lebih lanjut mengenai preservasi dan juga chemicals lainnya yang bisa mendukung produk akhir biologics Bapak Ibu kembangkan di laboratorium. Nah, mungkin sebagai highlighting saya sudah menyebutkan bahwa kita merupakan one step solution. Nah, di sini saya akan sebutkan bahwa produk Merck sendiri itu dapat dilihat dari MQ level. Jadi ini adalah quality segment dari produk kita. Biasanya di setiap produk ketika Bapak Ibu dan login ke Sigma Aldrich, di situ akan dijelaskan berapa quality levelnya. Jadi MQ 100 sampai MQ 600. Nah, MQ100 adalah komponen yang paling komponen yang paling rendah gitu, karena dia hanya melakukan pengecekan berupa basic quality gitu ya, tapi tetap menggunakan manufacturernya masih dengan menggunakan ISO 9101 gitu, 9001. Nah, ini mungkin cocok, tapi ketika Bapak dan Ibu sudah mulai melanjut ke arah biopharmaceutical atau manufacturingnya, mungkin bisa mengecek produk yang berlabel MQ 500 dan MQ 600. Jadi misalnya Bapak dan Ibu mau menggunakan sodium fosfat gitu. Sodium fosfat ada yang MQ 100, tapi kalau sodium fosfatnya sudah mau ke arah sarankan menggunakan sodium fosfat yang menggunakan MQ 600. Jadi kita memang support memiliki wide variety dari reagent chemicals berdasarkan quality segmennya sendiri. Nah, terakhir apabila Bapak dan Ibu memiliki ingin cost atau menghemat budget gitu, kita juga memiliki kemampuan untuk bulk packaging. Jadi di sini Bapak dan Ibu bisa membeli packaging atau pre-pack yang lebih besar sehingga nanti cost untuk bahan-bahan kimia yang Bapak Ibu gunakan di biopharmaceutical tersebut di-cut dengan sangat efisien. Silakan menghubungi ke mendapatkan penawaran produk untuk Bapak dan Ibu. Baik, ini saya terakhir hanya masuk ke quiz time gitu. Nah, untuk quiz time sendiri ini akan silakan menjawab pertanyaan tercepat dan mengirimkan emailnya ke saya dengan format nama, institusi, kemudian jawaban gitu. Nah, untuk pertanyaannya sendiri adalah sebutkan salah satu saja teknologi detection yang dimiliki Merck yang tadi sebutkan, walaupun dengan presentasi yang terburu-buru karena limitation waktunya. Jadi silakan Bapak dan Ibu bila memiliki jawaban untuk pertanyaan tersebut silakan kirim ke email saya. Nanti di situ untuk 5 orang tercepat akan saya berikan gimmick atau hadiah dari tim kita. Baik, saya rasa itu saja presentasi saya. That's all my presentation. So sorry about rushing the time. I have a technical problem, etc. So yes, thank you very much for your attention and hope to see you again.
Okay, thank you, Mrs. Sri Hayuni, for the presentation. Now, ladies and gentlemen, before continuing to the next agenda session with invited speakers and parallel session, we will have a break for an hour, and we will start the session at 1:30 p.m. Jakarta time. And at the end of the event, there would be announcement for 6 best speakers. So stay tuned and see you after the break. Thank you. Ladies and gentlemen, welcome back to ISIBIO 2021. In the following hours, we will have the first panel session that are divided into breakout room. But before that, there will be the session with the invited speakers. So for room 1, there will be Dr. Puspita Lisdianti. At room 2, there will be Dr. Fir Muhammad Abdul. And at room 3, there will be Professor Yong Soo Kim. Parallel session 1 will start at 2 p.m., parallel session 2 at 2:40 p.m., parallel session 3 at 4 p.m. Jakarta time. And for all presentation of parallel session will be conducted according to the rundown given by the committee. And to enter the room, you could choose the square icon, breakout rooms, and on the toolbar right below and click join of the room that you choose. And now you may enter the breakout room. Thank you.
Hello.
Yes, Mr. Yong Soo Kim.
So I can start now?
You could choose the room that you are assigned to. Professor Yong Soo Kim, you could choose the room 3.
Room 2. So how can, I see.
Room 3, if I'm
room
not mistaken.
3?
Yes.
Okay, here, room 3, okay.
To meet you, will guide you as a moderator in the session. We have invited speaker, Dr. Fir Muhammad Abdul. He's a lecturer at Biochemical Process Engineering, University Kebangsaan Malaysia. Then after this session, we continue to parallel session with 3 presenters. Before start our session, please allow me read Dr. Fir CV. Dr. Fir Muhammad Abdul, his field area in aerobic digestion, biofuel, biohydrogen, biomass pretreatment, biomass to biofuel, biomaterial, dark fermentation, environmental microbiology, fermentation biotechnology, lignocellulose, palm oil effluent, wastewater biological treatment. And his work experiences are since 2016 up to now, senior lecturer. Then 2016 from April to August, visiting research scholar at Feng Chia University, Taiwan. From 2014 until 2016, graduate research assistant at University Kebangsaan Malaysia. His research project area is native enriched inoculum for consolidated bioprocessing of biohydrogen production from affected treatment of oil palm empty fruit bunch, palm biomass and biohydrogen, a green scheme for biomass conversion to renewable fuel. Also, this project now is palm biomass, biohydrogen, green scheme for biomass conversion to renewable fuel. His project from 2019 until 2020 is genome engineering of locally isolated Clostridium butyricum using CRISPR for enhanced biohydrogen production from oil palm produce. On vegetation of light absorption efficacy in bacteria by photoluminating carbon dot, enhancement strategies for zero waste Cambodia with Tajuk project, life cycle analysis of zero waste innovation. Dr. Pir Mohammed, please welcome. Time is short, 30 minutes, 20 minutes for presentation, 10 minutes for Q&A. Time is short, please.
Okay, terima kasih Prof. Yani for your wonderful introduction for myself. Okay, so actually I didn't expect for you to read my entire project title. Okay, so let me introduce myself. Okay, my name is Pir Mohammed and I'm from University Kebangsaan Malaysia. Okay, and currently I'm working as a lecturer in the Department of Chemical and Process Engineering. Let me share my slides. Okay, and also I would like to, with your permission, I would like to stop my video, but I'll continue the presentation because I'm afraid there's a problem with my line. Okay, so I hope you okay with that. Thank you. So let me share my screen as well. Okay, so welcome everyone. Okay, to this wonderful conference. Okay, organized by BRAIN. Okay, in Indonesia. Okay, my name is Pir Mohammed and I'm from University Kebangsaan Malaysia. Okay, and today my title for this particular seminar or conference is the dynamicity of a biohydrogen producing microbial community from POME/ and using a next generation sequencing. So let me tell a bit about myself. Okay, practically I'm actually more like an engineer, chemical engineering background, rather than on the biotech. But I think it's because of the theme of the conference is more on the biotech, so I tried to incorporate a bit of a biotech element in my speaker talk today. Okay, so my main research activities, okay, in Malaysia usually is related to the oil palm. So oil palm is very common in Malaysia and extremely important in Indonesia. Malaysia is actually currently is the second largest producer of oil palm, but the biggest producer of oil palm in the whole world is actually Indonesia. So oil palm and palm oil is actually a very big industry both in Indonesia and in Malaysia. So it's naturally that we actually Malaysia and Indonesia spend a lot of our resources, okay, into researching how to improve this particular industry, because it's actually a large it's important for our own countries as well. Okay, so basically oil palm is not local in Malaysia or Indonesia. It comes from West Africa, right? So it's a monoecious crops, okay, means both the male and female flowers are on the same tree, okay. And why we actually cultivate this palm oil trees, okay, actually is for the FFB, the fresh fruit bunch. Okay, so usually we can harvest a fresh fruit bunch, okay, after about 3 to 4 years, okay, and each of the bunch can weigh until between 10 to 20 kg. And a normal tree can go up to 18 meters, okay, and then they can produce for about 20 to 30 years. Okay, so this is like a typical palm oil processing. Okay, so basically it's a process of a combination between mechanical, okay, thermal, okay, and also a lot of physical activities, okay, for separation. Okay, usually the process is very chemical free. Okay, and they use water mainly. Okay, water is actually to produce a steam and also to generate electricity. Okay, the palm oil mill is essentially is a green mill. Okay, where they actually using biomass as a source of their energy, okay, rather than using other sources like coal or petroleum. So it's practically a kind of a quite clean industry. Okay, and from the palm oil system, okay, usually there were two different products. Okay, one is actually a palm oil what do you call palm oil, okay, so crude palm oil. Another one is crude kernel palm oil, CPO and CPKO. And while the oil is actually important, okay, the oil only about 10% of whatever they produce, okay. The remaining 90% is actually the biomass, okay, lignocellulosic material from the palm trees, okay. So they have several classes, okay. But today we are not focusing on the solid lignocellulosic, but we will focus on the wastewater which is being produced in the palm oil industry, which we call as a pomade. Okay, pomade is actually stands for palm oil mill effluent. Okay, it's a wastewater from this particular industry. As I said earlier, okay, this industry, they use a lot of water. Okay, so water is essential to production of steam. Okay, to loosen the fruits. Okay, to digest the fruits. Okay, and then to extract the oil out. Okay, so this whole process use a lot of water. So basically, a rule of thumb. Okay, 1 ton of a fruits FFB. Okay, usually they will produce about 60% of wastewater. And this wastewater usually in form of a very thick brownish liquid, okay, and with a high level of COD, okay, high level of what we call chemical oxygen demand, okay, and the chemical oxygen demand COD can range between about widely about 40,000 up to even 100,000 in some cases. So, and because of the large amount of fresh fruit bunch usually is harvested in Indonesia, so it is also a large amount of wastewater, which is pomade is actually produced, okay? So pomade must be treated before we can actually send back to our waterways. Because the thing is, if we don't do any proper treatments, okay, the pomade will be polluting the water river system, okay. So for that reason, usually traditional industries, okay, in maybe about 20 years ago, they use a lot called ponding system, okay, where they have a series of ponds, okay, to treat this wastewater. Usually pond system, usually open ponding system is not very efficient, okay. At the same time, they lead to the greenhouse gas problem. So what happened, okay? Other than that, okay, there is also other issues, okay, related with the palm oil industry, okay. As I said, the use of water, the use of electricity usually is mitigated by using a biomass, okay. And there is what you call a release of CO2, okay, carbon dioxide, okay. And there's a lot of fibers, kernel and mesocarp, okay, and also pomade. Okay, so for this particular talk today, we will focus more on the pomade. So pomade, as I said, is a wastewater, okay, and the pomade characteristic usually is a widely range, okay, from as low as 15,000 COD can go up to 100,000. So it's like this, okay, if I go to the mill and I do a sampling, okay, today the value of a COD and tomorrow's the value of CD is not the same. So it's a bit hard to actually operate with the pomade, okay. And pomade, if not treated well, okay, if it's using a traditional method where they use open ponding, as you can see on this picture, they can cause emission of a lot of greenhouse gases, okay, like CO2, carbon dioxide, and also methane. So it is important to find a solution, okay, to treat this particular pomade well, okay. So a typical mill, okay, they transition, okay, from open ponding to anaerobic digestion system. So anaerobic digestion system is nothing new. It's a traditional method, okay. As to make it simple, okay, if we don't do anything, okay, we just leave it pomade as it is, okay, the pomade will undergo anaerobic digestion. But if you do it with a proper engineering technique, okay, you can actually enhance the efficiency of an anaerobic digestion system. But what is the benefit of anaerobic digestion? The benefit of anaerobic digestion is, okay, while doing this process, you can also produce energy, which we call as through the formation of methane. So methane is also called as almost similar to the natural gas. Okay, we can use the methane, okay, to actually use it for as a fuel. Okay, with a gas engine. Okay, we can actually run a gas turbine. Okay, we can run gas engine. Okay, to generate electricity. So anaerobic digestion is actually a common technique. Okay, which been started to be applied in a lot of palm oil industry, where they use what they call like a digestion system. Okay, like a tank. Okay, to actually collect this biogas. Okay. Now, in principle, okay, anaerobic digestion can be separated into 4 phases, which is the first one is the hydrolysis phases. Okay, and then we have a second phase, which is the acidogenesis, third phase, which is the acetogenesis, and fourth phase is the methanogenesis. Okay, the first phase is really to convert all the oligomers, okay, large sugar complex, okay, large macromolecules, okay, which is present in what we call pomade into a simpler monomers. And then this monomer will be consumed by anaerobic bacteria, okay, to produce acetic acid and also other organic acid. The organic acids will be converted to acetic acid through acetogenesis process, and finally to methane. So this is a 4th stage, okay, which is normally occur in the anaerobic digestion. So as I said earlier, okay, if we don't do anything, this process will happen, but with the right engineering technique, okay, this process rate can increase and we can produce a lot of methane, which eventually can turn profitable. But later on, with a bit more research, we found out, okay, if we can separate these 4 phases into 2 separate tanks, okay, meaning in a traditional way, okay, we use a 1 tank to do all phases, okay, semua dalam satu tangki. Tapi now, okay, we change it into a 2-phase system, okay, 2-phase system, okay, where the first tank is actually taking care of the first 2 phases, which is the hydrolysis and acidogenesis, and the second tank will actually the place for the what do you call acetogenesis and methanogenesis. Okay, so what's the benefit of doing this? Okay, by separating these 4 phases into 2 tank or 2 stage, okay, we can produce hydrogen gas. Okay, so initially in the old technology, it's a ponding system, okay, where the CO2 and methane actually emitted to the environment, polluting the environment, okay, and causing ozone depletion, all this thing, okay. And then the first generation actually using an anaerobic digestion system where we do it using in a tank, okay, to collect this methane. And now new trend is actually we can re-engineer this anaerobic digestion into 2-stage system where we can produce hydrogen first and also then followed by methane. But why hydrogen? Okay, now we are hydrogen making a splash. Okay, there's a lot of news on hydrogen on the world. Okay, especially in Japan and Korea, they're actually focusing a lot on hydrogen. Toyota, Hyundai, they're actually investing a lot of money on hydrogen. But again, why hydrogen? Okay, hydrogen has a higher specific energy content. Means what? Okay, let's say I give you a 1 kg of petrol and a 1 kg of hydrogen. Okay, a kg of hydrogen will contain more energy than petrol. So that's why it's because of higher specific energy content. And it's actually closer to a clean combustion. If you take a petrol, you burn them, you still get CO2. But this, if you take hydrogen, you burn them, you don't get CO2, you get only water. So that's why we call it a clean combustion. And at the same time, another advantage of hydrogen is, okay, we can use it in a PEM. Okay, PEM is actually a fuel cell technology where they don't use a combustion per se, but I think they use what we call electron exchange, okay, technique, okay, to produce electricity directly. Okay, so hydrogen has an advantage to be used in the PEM, okay, to what we call to power our life, okay, and the research on fuel cell has been widely occurring, okay. There are a lot of people are working on PEM, okay, but the thing is production of hydrogen still largely remain on the fossil fuel, means they use petroleum to produce hydrogen, which is not good, right? So alternatively, hydrogen from what you call palm oil or hydrogen from using a microbe is actually an alternative way of producing hydrogen, which is much more clean, okay. And also we can use a palm oil meal, okay, to produce hydrogen itself, okay. So pomade is a good source of hydrogen. So that's why we actually focus, okay, on this particular research in UKM, okay. We've been doing for the past almost about 12-13 years, okay, focusing on the conversion of pomade into hydrogen. So that's what I'm going to share some of the what we call research, okay, which we done last year. Okay, so to this audience, okay, so this is my research methodology, which is going to be discussing now. Okay, so initially we collect the pomade from the palm oil mill, neighboring palm oil mill, about 40 kilometers from our university. Okay, and then we use an anaerobic digestion system. Okay, but the thing, this one is a re-engineered system where we actually focusing on the hydrogen production only. Okay, and we try to increase the hydrogen amount, okay, per kg of POME. So that's the main goal of our research for our research team in UKM. Okay. And we work on several techniques, okay, to improve the hydrogen production. Okay, so these are our what do you call like experiment setups, okay, which we use for this particular studies. Okay, so this studies is focusing on the what do you call this the bacteria microbial community analysis. Yes. Okay, so the microcommunity, okay, using a simple bioreactor. Okay, so this is a simple UASB, okay, upflow anaerobic / blanket bioreactor, where we it's a working volume about 1.5 liter. Okay, and we run it on the thermophilic condition with the 55 degrees Celsius. And after we run the fermentation, we take the sludge out of it. Okay, where we have a rich biohydrogen producing bacteria, and then we do DNA extraction. Okay, and then we subject to next generation sequencing to know who's the player. Okay, so to really optimize the process, is also important to know who's the player inside the bioreactor. So we did NGS. Okay, and these are like the main results what we get from our biohydrogen tank. Okay, we operate the biohydrogen tank, okay, using a different HRT from 48 HRT. So we reduce it at 24 to 12, 6. We try to reduce to 3, and then it's failing, and then we come back to 6 and 12 again. So we found out, okay, the best HRT hydro retention time for our biohydrogen system is actually at the 6 hours. So at 6 hours, we can produce a quite robust what you call production of hydrogen, about 7.93 liter hydrogen per liter POME in a day. Okay, mine, okay, this one is using POME, okay, which is not being altered. That means there's no dilution. We use it as it is. We don't add any chemicals. There's no nutrients. There's no vitamins, nothing. We just use a POME, and then we just use a sludge that we enrich in the lab. Okay, and then with the know-how how to operate the bioreactor well, okay, we managed to achieve this. So this is a promising results, okay, which we can use it to what do you call to go for a larger scale. Okay, but the thing is, it's also important to know who's the player. Here indicate the biocatalyst, okay, referring to the microbial system in the thing. Okay, and this whole system being done using a non-sterile condition where there's a lot of microbes inside the system. So but before we go there, okay, we also try to quantify, okay, the metabolic analysis inside the system. We found out the highest amount of acid which is present. Hello. Okay, we found out the highest amount of metabolite is actually acetic acid, which is common in our system. Okay, having more acetic acid actually is a good, okay, it's a good indicator showing our hydrogen producing is actually in the right direction. So we try to understand who's the player inside the system. Okay, so by using a next gen sequencing sequencing system, okay, where we try to take out the sludge and then we extract the DNA. Okay, so using a sludge soil extraction DNA, okay, kit. Okay, so we managed to extract the DNA. So we got about like 10,000 kilobytes per, okay, of the DNA. Okay, and then we go through next generation sequencing, which is a standard procedure. We don't do any modification or something. We just follow whatever the supplier told us to do, because our research is not into the development of the NGS itself. Okay, we are more just a user of the NGS. So we managed to what do you call try to identify who's the biggest player over there. So out of our this thing, we found that there's roughly about 47 different OTU. Okay, the main microorganism species actually in our sludge. Okay, there are more, but I think we just focus about 47 because it's the most abundance. Okay, and we found out, okay, the biggest guy who actually biggest group of bacteria which is available here actually is belong to the what you call the class of Clostridia, Bacilli, and also Thermonoarabicter. So this is a good indication. Okay, why? Because Clostridia is actually known as a good hydrogen producing team. Thermonoarabicter is also known as a good hydrogen team, especially in the thermophilic condition in the high temperature. Basically is not so a good producer, but things we know they can, some of them can produce hydrogen. So we try to look into deeper, okay, thing, okay. So again, at the general, okay, the common one is actually dominated by Thermonoaerobacterium and also Clostridium. So both of these, we realize both.
Dr. Peer, your time 2 minutes more.
Okay, thank you. Okay, so we notice both of these actually is the biggest hydrogen producing thing. So now we know who's the person, okay, inside this thing. So we are strategizing, okay, further, okay, to how to preserve this particular team player, okay, so that we can have a consistent production of hydrogen throughout the year, okay, so which is a ongoing research at the moment, okay. So these are conclusions, okay. The hydrogen producing, okay, we got about 7.93 liter per hydrogen per per day, okay, at the 6-hour HRT. And the biggest player that we found is actually belong to the Clostridia, okay, and also Bacilli, and also Thermonobacterium. And with the phylogenetic tree, we think Thermonobacterium is actually the largest contributor for our biohydrogen production, which is fit our system, where it's an anaerobic system and at the same time it's actually a thermophilic system. So this is a good fit, okay. So I think also some people might wonder where we can actually implement hydrogen. So why these guys so passionate about biohydrogen? So basically, palm oil, again, I said, is a big industry in Malaysia, Indonesia, okay. So most palm oil mills, they rely on the Pondy system, the traditional one, okay. A bit more advanced mill, okay, they have a biodigester. So this is a typical picture of a biodigester. So you can see at the top here, it's like a balloon gas bag, okay. It's actually to collect the biogas, which is referred to the methane. So, but the thing is, with the right engineering, we can actually put the hydrogen tank, okay, before the what they call the methane tank. So this actually is the future application of what research that we are doing now. Okay, so I think that's it for my presentation today, because it's very short, so I cannot go very deep. So this is my reference. Okay, and also I want to thank, okay, all my contributors, okay, including the ISIBIO organizing team for inviting me. And before I end, I would like to invite everyone to join our conference, okay, in coming month, okay, next month, in fact, okay. If you cannot sign a paper, you can join us as a participant, just so thing. So you can learn more about hydrogen research in Asia. So we have a lot of speakers from Thailand, Indonesia, Taiwan, and China, even from Japan and Korea as well. So you can join us here. You can get the information here, or you can let me know, okay. And also I would like to invite all of you to join our talk, okay, sustainability talk series. This is a free talk. We do it monthly basis at the UK Wesley Chair. Okay, it's open for everyone. So in the coming thing, we have a investing in a sustainable future by Dr. Hajar Yatira. She's our funder. She's the one who actually give us the money from Sandabi plantation. Sandabi is also available in Indonesia as well. So if you have any question further, okay, you can contact me. So thank you so much. That's all. Thank you.
Thank you very much, Dr. Peer. Very nice and interesting presentation. Now for time discussion, Q&A, we have 8 minutes. The question, please directly to Dr. Peer.
So anyone has any questions?
Prof. Yani, I would like to ask one question.
Please.
So, Dr. Peer, very nice presentation. Thank you very much. I would like to ask about the sludge after the fermentation. How do you utilize the sludge? Can you apply it to farm soil, or do you have any other idea? Thank you very much.
Okay. At the moment, okay, the sludge we produce actually is not much. Okay, the sludge is very little. For us, the sludge is very valuable because it's actually rich in the biohydrogen producing bacteria. So we want to increase the what they call the amount of bacteria inside the system. Okay, if possible, we don't want to lose the sludge. Okay, but the thing is, we cannot forever keep the sludge in the system, because over time it will be start filling up. So based on our research, okay, we have to maintain about 30% of the sludge content inside the bioreactor. Whatever in excess, okay, we actually try to apply for what they call this as a soil conditioner to apply on the like a fertilizer, pupuk, right? So so far it's actually been showing a good what you call promising things. Okay, but we don't look into that at the moment. Okay, we don't look into the fertilizing value at the moment. Okay, but we try. Okay, and that's it. That's my answer for that.
Thank you.
Any other question, please?
Thank you. Can I ask to Dr. Pir?
Yes, boleh.
Okay. The nice presentation. Dr. Pir, I want to know if the process is period for acidification reactor and methane reactor. So your research is optimized in the acidification reactor only, or after you got the higher hydrogen and you subsequently process to methane reactor? Thank you.
Okay, thank you. So at the moment, the one I presented here is only focusing on the acidic reactor, which is for the hydrogen. So to tell the truth, okay, we take the fresh pomade and then we go to the first reactor, which is for the hydrogen producing. The effluent, okay, which is coming from the hydrogen tank, we use it for methane as well. So for this presentation, I didn't discuss about the methane part. Okay, so in a real life, we run two bioreactor, okay, together. Okay, the first bioreactor actually is focusing on the hydrogen. Okay, the second bioreactor is actually focusing on the methane. So as I said earlier, okay, we can do it in a single tank. We got only methane, but in our research, we do it in a two tank where we get hydrogen and also methane together. So thank you.
Okay, thank you very much. Due to limit time, Dr. Pir, thank you very much. So we stop our session. We move to parallel session. Please give applause to Dr. Pir. Thank you very much, Dr. Pir.
Okay, thank you so much for inviting me. Okay, and if you guys have any question, you can always email me. Thank you so much, Prof.
Welcome. So we move to parallel session. We have three presenters. The first will given by Anastasia Weni Indrianingsih. I think we share video, the committee, please.
Good morning, everyone. Thank you very much for the chance. Today, I would like to present our research about the composite of silver bacterial cellulose from cassava and its antibacterial activity. My name is Anastasia Weni Indrianingsih from BPT-PA BRIN, Yogyakarta. Bacterial cellulose, or BC, is a polysaccharide and consists of beta-1,4-glycosidic bond. It can be produced by bacteria such as Sarcina, Agrobacter, and Gluconacetobacter, which generate a wet sheet layer formation on the surface of culture medium. BC has similar properties with cellulose from plants. However, the cellulose production from plants required expensive and several steps of purifying process, such as using acid and alkali chemicals. Several advantages of BC properties are good permeability, high purity and crystallinity, good water absorption ability, and unique mechanical properties. However, BC seed itself does not have biological properties capabilities such as antibacterial or antifungal. Silver was known to have antibacterial activity and used for treatment of drug-resistant infection. Silver NBS has the antibacterial activity due to its high specific surface area. On the other hand, cassava is one of the abundant sources of local food in Indonesia. Cassava is usually used as source of carbohydrates by being processed directly and made into flour or starch. However, in the process of processing flour or starch, liquid waste is often produced which pollutes the environment. The study has been focused on the utilization of liquid waste to produce BC. The aims of this study are to evaluate the utilization of liquid waste from the processing of cassava flour and incorporate silver NPS into the PCC to produce EKPC composite. Also, to evaluate the antibacterial activity of EKPC composite that was be performed against 5 pathogens bacteria. The materials of the study, Ghiselinum bacteria and pathogenic bacteria, was obtained from Gadjah Mada University, Yogyakarta. Cassava was purchased from local market in Yogyakarta. For the PC production and EKPC composite synthesis, the cassava wastewater was obtained by blending the peeled cassava with the ratio of cassava and water used 1:4 weight per volume. And after left standing for 4 hours, the cassava water was used by separating the starch and the water. The culture medium was consisted of cassava wastewater, Ghiselinus, sucrose, and urea. The incubation was conducted for 7 days. The BC was immersed in the silver solution for 1.5 hours, washed with ethanol, followed by addition of sodium borohydrate solution, and reacted for 10 minutes. The silver and sodium borohydrate ratio was 1:10. Finally, the composite were washed using pure water, dried and stored in the refrigerator for further analysis. The characterization of the composite was conducted using the same EDX for the surface morphology and atomic element analysis, and also using the FTIR for the functional groups. The antibacterial assay was performed using Dix diffusion method. It was conducted using 5 pathogenic bacteria, namely S. aureus, S. type, Ecology, B. subtilis, and B. aeruginosa. The size of EKPC was cut in the symbologies of 6 mm in diameter. The positive control used was amoxicillin, and the negative control was pure PC. The discs were incubated at 37 degrees Celsius for 24 hours. And the antibacterial activity was evaluated by measuring the diameter of inhibition zone. This saw the physical appearance of the pure PC and the EKPC composite. The resulted PC with a diameter of 9 cm had an average weight of 9.1 grams and has thickness of 1.1 millimeter. The water content was around 96%. The process of synthesizing of EKPC composite was by impregnating silver particles to the PC matrix. The silver ions will go into the PC networks and absorbed by the PC matrix. It would be bound to the PC network probably by ionic bonds. The existence of OH group in the PC matrix will make electron-rich oxygen atom interact with silver ion. It showed the same analysis results. It was observed that pure PC and the EKPC composite consisted of fibrils. It was forming a porous network structure like finely woven, and the width size of the fibers below 100 nanometers. It was seen that the silver particles were incorporated in the PC matrix. However, some aggregation of silver NPS in the impregnation process was possibly occurred, indicated by the difference of size and shape of silver NPS. A study revealed that the agglomeration of NPS was an important parameter in toxicology studies. Several factors affect the structure of EKPC composites, such as the impregnation method and the silver concentration. This shows the results of EDX analysis. So the highest atomic composition of EKPC composite from cassava was O atom as of 50%, followed by C, Na, Ca, and silver. EKPC composite also contain O and C, since PC matrix consists of glucose monomers. This shows the FTIR analysis results of pure PC and EKPC spectra. The absorption peak around 3,470 and 3,320 indicated the OH group. Meanwhile, the absorption peak around 2009-16 and 2008-54 showed the CS stretching, and peak at 1004-27 showed the bending of CS bond. The absorption peak around 1006-51 indicated the CO groups, and furthermore, peak around 1060 indicated the COC stretching from the PC matrix. The absorption peak of PC and AKPC composite from cassava in the study was in accordance with the literature. This showed the antibacterial activity of the AKPC composites against 5 pathogenic bacteria. So the AKPC composite from cassava had the highest antibacterial activity against B. aeruginosa as of 8.2 mm, followed by B. subtilis, S. type, E. coli, and S. aureus. Pure PC had no inhibition ions against all the bacteria, while the amoxicillin, as the positive control, saw very good inhibition as of 17 to 30 cm. A literature reported that millimeter. A literature reported that the mechanism of AK in PS against bacteria was that silver would affect the bacteria cell wall, changing shape of the cell, causing the release of cytoplasmic content, and followed by the bacterial death. For the conclusions, this study presented that silver and PS was successfully incorporated into the PC networks from cassava to produce AKPC composite. The impregnation of silver into PC was confirmed by SEM and EDX analysis. The same image showed that the AKPC composite had nanofibril networks. The AKPC composite show good antibacterial activity against 5 pathogen bacteria, that is S. aureus, S. type, E. coli, B. subtilis, and P. aeruginosa. These results show that AKPC composite from cassava has a high potential in pharmaceutical or cosmetic industry. That's all of my presentation today, and thank you for your kind attention.
Thank you, Anastasia Weni. Then we move to second presenter, will given by Barliati Sarah Agustina. Please welcome Sarah Barliati Agustina.
Assalamualaikum warahmatullahi wabarakatuh. I am Barliati Sarah Agustina from the Center for Chemistry, National Research and Innovation Agency of Brain. Today, I will present you about my paper, Extraction of glutathione involved in its fermentation as co-product of second generation bioethanol from oil palm and different. Glutathione is one of byproducts of bioethanol production. It can be produced from glycosylosic biomass as bioethanol conversion. Glutathione also can be found in Saccharomyces cerevisiae from salt pass remaining fermentation blood. Glutathione also has potentially activity as antioxidant, because the ethyl group within the sustain of glutathione is a chemical agent which could be irreversibly oxidized and reduced. Most of glutathione produced by this fermentation is in the form of GSH because the intracellular GSH concentration is greater than GSSG concentration. The version of glycosylosic biomass is slightly developed for renewable energy and to switch petroleum. The main components of glycosylosic biomass are cellulose, hemicellulose, and lignin. Polysaccharides in glycosylosic biomass can be reintegrated to sugars through chemical or biological conversion, and then the sugars can be fermented to ethanol or alternative valuable chemicals. The conversion of glutathione to bioethanol carried out in 4 steps. 4 main steps. There are pretreatment, hydrolysis, sugar fermentation, and purification. The pretreatment of raw material is a step to produce certain compound, improve the efficiency of hydrolysis, and increase the yield of glucose from cellulose. And for hydrolysis, we can use several methods. It can be used in hydrolysis and fermentation process in these 2 steps. But in this study, we used separate hydrolysis and fermentation, or SHF, which are commonly used as enzymatic saccharification to hydrolyze polysaccharides first into monosaccharides and then ferment monosaccharides into ethanol by Saccharomyces cerevisiae. and after that, purification. The study was focused on isolating GSH as a byproduct of second-generation bioethanol fermentation and analyze its antioxidant activity. And next, we talk about materials and methods. As I described before, the first step to making the study is pretreatment, and after that, FB can be used for saccharification and fermentation process. And this is the saccharification and fermentation process. We use separate hydrolysis and fermentation, or SHF, glutathione extract. And after that, we characterize the AFB before and after fermentation using AOAC method, and also continued with antioxidant activity using DPPH. And this is the results. There are 4 variations for this study based on substrate loading and yeast loading. Type A has 10% of substrate loading and 1% of yeast loading. Then type B has 10% of substrate loading and 2% of yeast loading. And for type C, it has substrate loading 15% and 1% of yeast loading. And then the last one, type D, has 15% of substrate loading and 2% of yeast loading. And in the study, the results indicates that type C and D with substrate loading 15% obtain a higher concentration of ethanol production than type A and B with substrate loading 10%. Different concentration of yeast loading also affected the concentration of ethanol. At 10% of substrate loading, concentration of ethanol increased from 32.5 to 36.1. However, at 15% substrate loading, there was decreasing concentration ethanol from 63.3 and 50.7. This result may be caused by the cell number decreasing at 72 hours at 1 until 5% yeast loading. The ethanol concentration at 50% substrate loading with 1% yeast loading in the study is higher than 50% substrate loading and 2% yeast loading. And this indicate 50% substrate loading and 1% substrate loading is higher than previous study by Triwahyuni et al. 2015 and Dahnum et al. 2015, which obtained 62.8 and 47.4. But however, it was lower than previous study conducted by Sudiani et al. 2020 with 69.8 ethanol content. And then next is composition of substrate EFB and solid residue after fermentation. After fermentation, solid residue has several changes in the composition value. As you can see at the table, crude protein increases after fermentation because the nitrogen in fermenting yeast has a role in proteins and nucleic acid biosynthesis and also increases free alpha amino nitrogen. And for the calcium, it decreased after fermentation because the value of calcium and phosphorus content after fermentation may be due to the need of yeast to provide ionogenic ions because it's important for yeast fermentation performance. And for the ash, it decreased after fermentation. Ash is in line with decrease in calcium. Even yes, cerevisiae is known as facultative anaerobic. In fact, cerevisiae still needs oxygen for biosynthesis of fatty acids and sterols. And for crude lipid, content is increases after fermentation in response to enzymatic hydrolysis of fermentation. The increasing value of organic matter degradation was followed by an increase in both hydrolytic and oxidative enzyme activity. And for crude fiber, rising crude fiber value is typically followed by rising crude protein value. The presence more crude fiber in ADF and NDF fraction indicates that the fiber has a higher degree of protein binding. The reduction in moisture content seen as fermentation progress could be attributed to increasing dry matter. It is dry matter. And caused by yeast cell growth. In general, a decrease in moisture content enhances the content of nutrients. EFP in the study has higher value of moisture content than previous study by Rosley et al. 2017 with 9.51% moisture content of AFP. As content in the study is also higher than previous study conducted by Anwar et al. 2018 with 5.29% as content of AFP. And according to Ibrahim et al. 2015, ash is composed of several inorganic elements, one of which is calcium. The reason for the decrease in ash is in line with the decrease in calcium. The calcium content in the study is lower than in the previous study by Lau et al. 2007 with 0.7 calcium of AFP. Methanol is utilized as the solvent in the process of glutathione and glutathione extraction because DSH compounds can be fractionated in methanol solvent. The solid or liquid ratio affects glutathione concentration. According to Marianto 2017, optimum glutathione concentration was obtained at a ratio 1.1 to 3 solid per liquid. If the ratio of solid is higher than liquid, the value glutathione will be decreased because the solvent becomes saturated while extracting the glutathione. The highest antioxidant activity reached at 203 milligram per liter, and with the lowest ethanol content, 3.25%. And it showed that the lower ethanol content obtained, the higher antioxidant activity resulted. This research in line with research demonstrated by Brandolini et al. 2007, which performed fermentation of wine using 19 different strains of SRPC, showed that the high percentage of ethanol did not represent a high activity of antioxidant. And also, according to Rakhendran et al. 2020, glutathione content did not correlate with fermentation performance. Also, although the fermentation results obtain a high concentration of ethanol, which represents optimal fermentation performance, the antioxidant activity of glutathione extract is not affected by fermentation performance. And conclusion, following the biorefinery concept, the SHF process has both potentially to produce bioethanol as the main product and also produce residue from the fermentation process, which contains a high amount of SRPC that contains an antioxidant compound called glutathione. Methanol 100% could be used as an extracting solvent, glutathione in the remaining solid from AFP fermentation. The highest antioxidant activity of glutathione is 203.08 milligram per liter by using 10% substrate loading and 1% yeast loading. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Dr. Muhammad Syafaat.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for the opportunity given to me. Let me introduce myself. My name is Muhammad Syafaat, Resource Center for Chemistry BIN. Today, I would like to present you about our project entitled Pyrolysis of Microalgae and its Residue for Bio-oil. Over the past few years, substantial research has been carried out on fuel production from biomass. Algae has been examined as one of the most promising alternative resources to produce biofuels. One of microalgae that can be used as raw material for bio-oil is agar. Nowadays, the yield of agar made from macroalgae is only 30% dry weight. In the other word, there is a residue of 70%. It shows the potential macroalgae residues utilization as substantial renewable energy. Therefore, this paper aims to evaluate the feasibility of microalgae biomass and its residue as biofuel through pyrolysis. This table showed the chemical composition of microalgae such as carbon, hydrogen, nitrogen, sulfur, and oxygen are influenced by environmental conditions, light absorption, and also nutrient level in the sea. Temperature strongly affects the chemical composition of nutrients, CO2, and the growth rates of algae. Light intensity and light-dark cycle, as well as environmental factors, also play a significant role in algae growth. The low growth rate of microalgae may be due to the lack of availability of nutrients absorption and low light intensity. Disruption of the photosynthesis process inhibits the metabolic process of algae. Glucose and oxygen produced from photosynthesis also affect the C and O elements contained in each algae. In the respiration system, the glucose and the other compounds will react with oxygen to produce carbon dioxide, water, and chemical energy. The differences in an element is determined by bacterial activities in the nitrification process. The high ash content in algae produces a high biochar and a low bio-oil yield through secondary tar reaction. Pyrolysis products distribution can also be affected by the morphology of biomass, mainly due to the heat transfer effect. The increasing of pyrolysis temperature can accelerate the decomposition of algae and bio-oil residues. The amount of bio-oil from pyrolysis can be associated with an increasing temperature because micromolecules compound of algae will be increased by increasing temperature. The lost char could be attributed to the greater primary decomposition of microalgae at higher temperatures, or there is secondary thermal decomposition of char before discharging from the reactor. The enhancements of gas occurs due to secondary cracking of char and pyrolysis steam. Conventional pyrolysis produces more char, while microwave pyrolysis produces more gases. It's due to the presence of char as a microwave receptor that support a secondary reaction that leads to gas formation. The bio-oil has a lower oxygen content and a higher HO atomic ratio than the feedstock. The reduction of amount of oxygen indicates that a larger number of functional groups are lost during pyrolysis at higher temperature. The HC value ratio is lower than that of algae meal. This condition showed an aromatization reaction has been occurred. Heterocyclic compounds such as furans, pyrans, and pyridines are formed by Maillard reaction between sugars and amino acid radicals at high temperatures. The pyrolysis result from biomass and residue contain a lot of alcohol compounds, such as long-chain fatty lipophilic compounds. These compounds derive from pyrolysis of chlorophyll. Macromolecule of fatty acid, for example, steroid and carboxylic acid, and their derivative from pyrolysis result are still high. The result of pyrolysis using microwave showed the aromatic compound in this process is higher than pyrolysis using the other method, while aldehyde is not detected because aldehyde had been converted into aromatic compound. Many carboxylic acid and their derivative were easily identified because of ash of microalgae consists of alkali metals that effective on the pyrolysis. The algae residue that has been treated using sodium hydroxide has a relatively high acid compound. It can be concluded that the ester compound clearly decreases with increasing temperature. The quality of pyrolysis bio-oil needs to be improved through a catalytic process before being used as a transportation fuel. Most of the oxygen and nitrogen from bio-oil need to be removed to obtain high-quality biofuels. Water content in the fuel can prolong ignition delay and reduce engine combustion. Acid removal can reduce the corrosiveness of the biofuel. It's due to the conversion of acidic compound into other compounds such as ester and hydrocarbon. High viscosity could cause excessive fuel injection pressure during engine warm-up. A possible factor of the high viscosity bio-oil is the presence of oxygenated composition such as aldehyde and high molecular weight compound. The HDO process is an effective method to upgrade bio-oil through oxygen elimination, such as hydrodeoxygenation, deoxygenation, decarbonylation, and decarboxylation. There are three possible factors that can cause the decreasing of acid content. First, some acidic compounds are converted into ester by esterification reaction with alcohol. Second, certain acidic compounds are converted into hydrocarbon by decarboxylation reaction. Third, some acidic compounds may be transferred from the oil phase to the water phase. The content of C organic in solid waste were generally higher than the C organic content in biomass. It means that the solid waste extracted from agar can be used as a raw material for making bio-oil. Heating value of biomass is determined by the carbon content of the biomass or material. If nitrogen content of algae biomass were high, it cannot be used as fuel because of corrosiveness and environmental effect, like NOx gas can be generated. The conclusion, the maximum yield of bio-oil depends on several parameters such as moisture content, ash content, pyrolysis temperature, and vapor residence time. Physical properties that must be maintained as fuel are water content, total acidity, and viscosity. Residual gracilaria based on low alkali content has potential to produce more bio-oil than biomass itself. Thank you for the attention.
Thank you so much for all three presenters. Now this time for us for Q&A. The question, please direct or please type in this chat room. We still have 10 minutes for discussion.
May I have a question?
Please
of
direct.
Yani?
Yes.
Oh, okay. Thank you very much.
The question go to, please mention the name.
Yeah. Okay. Thank you very much for the time. I have several questions to Pak Safaat. From your presentations, have you conducted the experiment with the macroalgae? I mean, I didn't see. I'm sorry if I missed the presentation, but I didn't see the experimental results of your experiment when you pyrolyze macroalgae in your reaction. How about the results? And the second one, as I know, macroalgae or seaweeds contain molecules which are unique. I mean, some of molecules of seaweeds are not found in common terrestrial biomass or lignocellulose biomass, which means that most of the molecules derived in the seaweed usually have a high economic value in industry. For example, let's say Gracilaria or Echema, they have been used by industry to produce agar, which means, we know agar is very, how to say, like the price is more expensive rather than when we use it for the fuels. So how about how about your opinion about when you do, or when we use the macroalgae as a substrate for converting it into the biofuel? How about the result if we compare with that one? And the third one, when we use the residue of the residue that you use, is it how to say, like, if I'm not wrong, when people extract agar, let's say from the Gracilia, usually they mix some of material like diatomous earth, which means that the residue of the extraction process, agar extraction process from seaweeds, contains not only carbon but a mixture of solid materials, which is difficult to be converted in the pyrolysis reactor. So I just want to confirm what kind of residue that you use for this experiment. Maybe that's from me, Prof. Yani. Thank you very much.
Okay, thank you. Let me answer the question. Good question. Actually, this study only review paper. Now, in this year, we have a project entitled this paper. Actually, I don't know. I don't know about the other biomass exception. I only choose the biomass of in Indonesia. Actually, Gracilaria have a lot of amount in Indonesia, and the other industry only use Gracilaria and the other, I don't know, I forget it. So if you have question about why I choose Gracilaria, because Gracilaria is the most species in Indonesia, and the residue agar is a lot of residue agar, and I don't know about the other biomass. Let me. Okay. 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 Microalgae processing industry just able to 6,000 tons a year, and the predicted the residue is 7,000 year. So it's potential to utilize microalgae residue as substantial renewable energy. I think that that's all. The next question. Sorry, I forget the second question. Can you repeat?
I think Dr. Bayu not here. Dr. Bayu will repeat your question. Yeah, I don't see Dr. Bayu here. I think, we move to the other question to Safa.
Yeah.
Yeah, from the chat. The question from Ki Ageng Sarwono. The question is, do you have any criteria when choosing species for your sample?
Okay, actually, we choose the biomass species only di Indonesia agar the industry agar in Indonesia only Gracilaria and Carginan. So we choose that species to study in this year, but this study only review paper and not result experiment.
Okay, thank you, Safa. Any other question to the other presenter? Okay, I don't see another question. So we stop our session. Please give applause to all presenter. Thank you very much. Then I return to the committee the time.
Thank you very much, Professor Yani. Thank you very much to Dr. Peer and to all presenters at parallel session 1. Immediately after this, we'll have parallel session 2 with 5 presenters. But before we commence parallel session 2, let's enjoy a video from our presenters. Please stay tuned in this breakout room because we'll start parallel session 2 right after the video. Thank you.
Martin Chris, freeze drying system untuk penelitian, pilot project, hingga manufacturing produk Anda. Hubungi PT ITS Science Indonesia untuk informasi lebih lanjut. Martin Chris, freeze drying system untuk penelitian, pilot project, hingga manufacturing produk Anda. Hubungi PT ITS Science Indonesia untuk informasi lebih lanjut. Plant Genetic Engineering and Breeding Workflow, supported by ITS Science Indonesia. Plant genetic engineering can be done in many ways. One of them can be done as follows. First, growing bacteria for transferring the targeted genes into the plant. By using incubator farm member, you can measure the accuracy and stability of the optimum condition for bacteria to grow with simple user-friendly control button, just like this. Second, DNA extraction and isolation. With NanoDrop from brand Thermo Scientific, you can be assured the extracted DNA is free from contamination such as phenols and others. With feature called Ecology, we can count the concentration of extracted DNA easily without hassle. Super easy. Third, cloning and designing genes. Targeted genes then cloned with real-time PCR, LightCycler 96 from Roche. We can monitoring the amplification of targeted genes directly. LightCycler 96 has 4 different channels and can differentiate 4 different targeted genes simultaneously. And then, we can separate the amplicons with electrophoresis. The targeted genes on agarose gel can be documented very well with gel dot ingenious from Syngene. This gel dot can capture fluorescence signal from dyed DNA band. And then, from there we can verify the targeted gene and splice the gene into plasmid. Step 4. Transformation. Plasmid then are inserted into bacteria that we grow earlier. These bacteria will infect plant cells and insert recombinant plasmids into plants. Transformed cells then will be detected with flow cytometry. Meet Aurora from Cytec. This flow cytometry can help you to identify cells that are successfully transformed. Transformed cells with marker will be shown and counted by the software. Step 5. Cell and tissue culture.
Transformed plant cells then are cultured into agar in petri dish. The growing transformed plant cell can be captured and documented with cell imaging and multimode reader. Citation 5 from Biotech. Equipped with reliable microscopy module, Citation 5 can offer detection in fluorescence, bright field, HNA, and phase contrast applications. Citation 5 also equipped with magnificent lens up to 60 times. With Gen 5 software, image processing and analyzing are never been this powerful. We can also measure the optimum parameters for the transformed plant. Step 6. Plant breeding. Do not let your precious transformed plants die after all those steps above. Meet Confiron, a reliable climatic chamber for important applications such as growing tall plants, short plants, tissue culture, and incubation. With easy-to-use control panel and many other important features such as broad-spectrum LEDs and dimmable light. Alright, those are 6 steps of plant genetic engineering and breeding that ITS Science Indonesia can work with. Thank you for watching. Contact us on our socials for more info. Stay safe.
Halo, di video ini saya mau pamer alat canggih. Lihat deh, ini MagnaPure 96 dari Roche. Bisa buat bantu ekstraksi RNA virus COVID-19 secara otomatis. Sebanyak 96 sampel dalam waktu 2 jam. Setelah diekstraksi, sampel dapat diuji dengan metode real-time PCR menggunakan Light Cycler 96 ini. Hasilnya langsung terlihat positif atau negatif. Cepat kan? Hubungi teman-teman di ITS untuk info lebih lanjut. Bye-bye, stay safe.
Good afternoon, ladies and gentlemen. Thank you very much for your attendance. And right now we are going to start parallel session 2. For this session, Dr. Laude Muhdar Davis will be our moderator. He is a researcher from Research Center for Biology, National Research and Innovation Agency. He obtained his doctoral degree at Nara Institute of Science and Technology in the field of biological science, and he has a specialty in plant physiology. So without further ado, to Dr. Laude, the screen is yours.
Okay, thank you very much. Can you hear my voice all right?
Yes, we can
Okay,
hear you
thank you.
clearly.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. I hope you are all doing well. And I welcome you to the second session of the today's ISIBIO 2021. My name is Muhdar Davis from Research Center for Biology, BRIN, and just to make sure to all of you that this is a breakout room number 2, and in total in this parallel session we will have 5 presenters, and we will have the presentation session first, followed by Q&A. So during the presentation or the playing of the pre-recorded video, you are welcome to post your question to the chat box, and don't forget to type your name, institution, and to whom the question is directed. And I will read this question later on during the Q&A, or you can ask the question directly in the Q&A session later. So make sure your microphone is turned off during the video playing. So we will begin with the first presenter from Dr. Khairul Anam. You can start the video.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. My name is Khairul Anam from Research Center for Biotechnology. Today, I would like to present my research entitled Biomass Production through Sequestration of CO2 in Microalgae, Chlorella vulgaris. Here I show you the bathtub model of CO2 in the atmosphere, whereas the water flow, which illustrated as CO2 emission, is quite high to be released to the atmosphere compared to the removals that made the bathtub almost full. As you may know, the rapid development of the human population and uncontrolled consumption of energy through electricity, transportation, and industrial activities have been identified as the primary cause of global warming and environmental pollution. Several CO2 sequestration approaches have been introduced in order to capture CO2, for instance, reforestation, agriculture, and photosynthetic microorganisms. In this opportunity, I would like to talk about microalgae as CO2 sequestration agent. Microalgae are unicellular organisms which commonly found in aquatic areas such as in marine or fresh waters. As photosynthetic microorganisms, microalgae capture light as an energy source and convert carbon dioxide to produce organic compounds. In this research, we use Chlorella vulgaris, a green algae which will capture the CO2 gas. However, CO2 reaction with water may lower the pH medium. Because of that, we incorporated the medium with sodium hydroxide. In this research, we did not flow the CO2 gas continuously, but by adjusting the CO2 gas with sodium hydroxide to achieve optimum pH condition. Therefore, our study purpose is to reveal the suitable medium which gives the highest biomass production for Chlorella vulgaris, with CO2 gas as carbon source. Meanwhile, our objectives are to obtain the suitable medium for Chlorella vulgaris, to obtain the suitable pH for cultivation, and to obtain optimum concentration of sodium hydroxide. First of all, we try to find a suitable medium for microalgae cultivation. Chlorella vulgaris was cultivated in tub medium as stock culture, which kept for 1 month. And then Chlorella vulgaris is cultivated in a pre-culture medium with tub as the medium for 3 days with initial OD 0.1 to 0.5. As the main culture, the culture was inoculated into 4 different media, which were BG11, AF6, TAP, and F2 medium. Here, 4 media were screened to obtain the optimum condition for microalgae growth. In this experiment, microalgae was cultivated in 24 well plates and was observed for 7 days. The results showed that top medium had the highest biomass production in OD for Chlorella vulgaris. Top medium, which has the highest biomass production at the previous result, was investigated for its carbon source. Trace and acetic acid were considered as the carbon source for top medium. To understand the essential of carbon source in the medium, 4 kinds of media were made, and the growth of Chlorella vulgaris was observed. With almost the same procedure, the treatment focused on the 4 kinds of media. From this experiment, the results showed that trace is the essential carbon source for Chlorella vulgaris in top medium. Medium, which consists of macro and micronutrient of top, but without trace and acetic acid, was used for the basic compound. Meanwhile, sodium bicarbonate and sodium carbonate, which are releasing bicarbonate and carbonate ion, were used as inorganic carbon source for substituting the trace compound. About 20 ppm of sodium bicarbonate or sodium carbonate was added into a medium, and TAP was used as control. The results showed that Chlorella vulgaris could grow in media added with sodium carbonate or sodium bicarbonate. As I mentioned before, CO2 reacts with water and may lower the pH medium. Therefore, in order to use CO2 as carbon source, several media with different pH were made to understand in which pH that a medium for Chlorella vulgaris can be used for cultivation. The results showed that from pH 5 to pH 9, the growth of Chlorella vulgaris were good, with pH 7 had the highest biomass. The optimum condition for concentration of sodium hydroxide solution was investigated. Several concentrations of sodium hydroxide solution was made to find out which concentration would be the best to produce the microalgae biomass. Proportional time of 1 liter per minute of CO2 gas flow was applied to obtain pH 7 for the medium. The results showed that from 10 to 60 ppm of sodium hydroxide solution, 10, 20, 30, and 40 ppm were similar and give the best growth of Chlorella vulgaris. However, when the cultivation volume was upscaled and the method was changed, as we used 1 liter medium for the main culture and magnetic stirrer was used to homogenize the culture. So from this experiment, 60 ppm of sodium hydroxide solution was considered as the best concentration for biomass production. Here we compare the relative content of biomass production and its content from Chlorella vulgaris, which cultivated in top medium and medium with sodium hydroxide, 60 millimolar plus CO2 gas. From several analyses, we obtained that microalgae biomass content cultivated in sodium hydroxide, 60 millimolar plus CO2 gas, are lower compared to top medium. Even though lower, the medium of 60 millimolar of sodium hydroxide plus CO2 gas still reach 82% of top dry biomass weight. As summary of this research, first, top medium had the highest biomass production in OD for Chlorella vulgaris. Second, trace compound is the essential carbon source for Chlorella vulgaris in top medium. Chlorella vulgaris could grow in a medium added with sodium bicarbonate or sodium carbonate. The growth of Chlorella vulgaris were normal at pH 5 to pH 9 in top medium, and pH 7 had the highest biomass production. And fifth, medium at pH 7 with combination of CO2 gas and 60 ppm of sodium hydroxide solution showed the highest yield of biomass production. This project was funded by DIPA 2020 grant and also DIPA 2021. Thank you very much for your kind attention.
Okay, thank you very much for the presentation. We will move on to the next presenters. Eka Triwahyuni, you can play the video.
Good afternoon, ladies and gentlemen. I am Eka Triwahyuni from Research Center for Chemistry, National Research and Innovation Agency. Today, I would like to present our study about lactic acid production from oil palm empty fruit bunds using Lactobacillus delbrueckii. Lactic acid is one of important organic acid which is widely used in several sectors industry, such as in food industry as preservatives, flavors, or pH regulator. In chemical industry, lactic acid can be used as descaling agents, neutralizer, or green solvent. Lactic acid also can be used in cosmetic industry for moisturizer, skin lightening agents, anti-acne agent, or pH regulator. Lactic acid also can be used as chemical feedstock for propylene oxide, propanoic acid, ethyl lactate, dilactate, or polylactic acid, or PLA. Lactic acid can be produced from petrochemical resources by chemical synthesis method. In this process, petrochemical resources such as coal, petroleum products, or natural gas was converted to acetaldehyde, and then by addition of HCN and catalyzed, acetaldehyde can be converted to lactonitrile. Lactonitrile was hydrolyzed by sulfuric acid to produce lactic acid. However, according to the recent environmental issue and also limited nature of petrochemical resources, a more environmentally friendly production process is desired using renewable resources. Microbial fermentation is used to convert renewable resources such as carbohydrate and lignocellulosic biomass into lactic acid. Carbohydrate can be directly fermented to lactic acid using microorganisms such as lactobacilli, whereas the lignocellulosic biomass can be converted into lactic acid using four main steps of process. The first is pretreatment, and then enzymatic hydrolysis, fermentation, and the last is recovery and purification to provide optical pure L or the lactic acid. Indonesia has the highest production of palm oil, provided oil palm empty fruit bunds as roasted from oil palm industry. Thus, Indonesia has abundance of EFP that can be used as feedstock for lactic acid production. Therefore, the objective of this study is to explore the production of lactic acid from EFP through spirit hydrolysis and fermentation method using cellulolytic enzyme and Lactobacillus delbrueckii as lactic acid bacteria. The first step for this research is pretreatment of EFP. EFP was physically treatment by chopping and milling into particle size about 3 mm, and then The EFB was treated by adding 10% of sodium hydroxide solution in the chemical explosion reactor. The ratio of EFB and sodium hydroxide solution are 1:5. The process was conducted at 150 degrees Celsius for until 7 kg per centimeter per city of pressure for 30 minutes. After that, the sample was filtered and neutralized until the pH is 7, and then drying until the moisture content below 10%. The chemical component of EFB that are cellulose, hemicellulose, and lignin before and after pretreatment was analyzed using standard biomass analytical procedure from National Renewable Energy Laboratory. After pretreatment, EFB treated was used as substrate in the hydrolysis process. To carry out the hydrolysis, 50 grams per liter of EFB treated was added with 30 FPU per gram substrate of enzyme Cytec2 and 20% volume per volume of enzyme Hytec2. And also added buffer citrate until the working volume is 50 milliliter. The hydrolysis process was conducted at temperature 50 degrees Celsius, 150 RPM of agitation, pH 4.8 for 72-hour process. After that, the hydrolysis was fermented using Lactobacillus delbrueckii. 10% volume per volume of Lactobacillus delbrueckii was added into hydrolysis. In the fermentation process, variation of temperature and pH was conducted. The variation of pH are 5.5, 6, 6.5, and 7. And the variation of temperature are 55 and 60 degrees Celsius. The reducing sugar from hydrolysis process was determined using DNS assay method. And then the lactic acid in fermentation broth was analyzed using spectrophotometric method. For the result, the chemical composition of untreated and pretreated EFB can be seen in this table. Percent of cellulose compound increased from 35% into 65%. The hemicellulose compound was decreased. The hemicellulose compound decreased slightly from 18% to 10%. And the lignin as impurities can decrease from 35.80% to 14.59%. Therefore, the application of alkali explosion for pretreatment could provide accuracy of cellulose and can decrease the lignin content. The hydrolysis from the hydrolysis process, the result that is the reducing sugar of pretreated EFB after 72-hour enzymatic hydrolysis reached 26.7 grams per liter. And then after hydrolysis, the hydrolysis was fermented using Lactobacillus delbrueckii. This figure shows the lactic acid production during fermentation at 60 degrees Celsius with the variation of pH 5.5, 6, 6.5, and 7. As a result, the optimum lactic acid concentration was obtained at 72 hours of fermentation, reaching 0.83 until 0.84 grams per liter. After 96 hours, lactic acid from fermentation at pH 5.5, 6, and 6.5 were slightly increased, but concentration of lactic acid at pH 7 decreased. The highest lactic acid concentration, 0.91 grams per liter, was produced from sample at pH 5.5 after 96-hour fermentation. After optimum pH finding, the fermentation of pretreated EFB was carried out at another temperature that is 55 degrees Celsius. We can see in this figure, fermentation at 55 degrees Celsius produces higher lactic acid concentration as compared to fermentation at 60 degrees Celsius. 2.3 grams per liter can be obtained from 96 hours of fermentation. Therefore, the conclusion of this study are alkaline pretreated EFB can be converted to lactic acid by Lactobacillus delbrueckii. The temperature and pH of fermentation have an impact on the obtained lactic acid concentration. The highest lactic acid concentration was 2.3 grams per liter from fermentation condition at pH 5.5 and temperature of 55 degrees Celsius. The further studies are needed to optimize the substrate concentration, to enhance lactic acid concentration, and also to apply SSF or CBP method for lactic acid production. This is the end of my presentation. Thank you.
Thank you for the presentation by Ms. Eka. So we move to the next presenters, Dr. Yuni Setio Rahayu. So if you could please play the video.
Thank you very much, moderator. Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, it is in these opportunities, I would like to deliver our research with the title Soil Carbon and Various Land Uses of Mangrove. There is a NIPA ecosystem in Segara Anakan, Central Java. This map shows a mangrove ecosystem distribution in Indonesia, and based on the recent study, described that the largest mangrove area is in Asia, recorded about 42% of the total mangrove in the world. About 22.6% the total world mangrove ecosystem, reaching about 3.2 million hectares. And mangrove ecosystem in Indonesia are an essential ecosystem, which is important as a buffer zone and coastline protection, nutrient supply for the biota, habitat for terrestrial and marine flora and fauna, spawning and nursery ground some species of crustacea. And it is also well known that mangrove ecosystem absorb carbon very productive and efficient compared to the other ecosystem, with the highest carbon content of mangrove is in tropics, and Indonesian mangrove area store about 3.15 million metric tons of carbon, with the highest carbon storage is in soil, about 89%. And the potential carbon storage varies depending on the level of degradation, the type of land cover, mangrove formation, and the climate change. Ladies and gentlemen, it is well known that the change of mangrove areas in Indonesia is significant. In the 3 decades, the loss of mangrove forest is about 40%, mainly due to the urban development, settlement, and industrial development areas along the coastline, expansion of aquaculture and agriculture. And mangrove forest loss in Indonesia account for 42% of greenhouse gas emission due to the land degradation. It is therefore, by considering the importance of this essential ecosystem, either as carbon storage or economic resources, the assessment of soil carbon storage at the various community of the mangrove ecosystem is required to support management policy and their conservation planning. This research prioritizes describe the carbon content of the substrates in mangroves area and their surrounding area. This map shows the mangrove study site in Segara Anakan, Central Java. Segara Anakan, the largest mangrove ecosystem in southern coastline of Java, located between Java and Nusa Kambangan Island in Central Java. And the semi-permanent plot size 30 by 10 meters square divided into 3 subplots, 10 by 10 meters square were established across various mangrove ecosystem. And the plots were divided in dense mangrove with the vegetation cover more than 50%. For SM1, this is the condition of the our study site in dense mangrove, sparse mangrove with the vegetation cover less than 50%, and two planted mangrove, Nipa fructicans, dairies, and the other ecosystems, shrub, other terrestrial vegetation, and paddy field. So samples were taken by using an edge-cockham pit auger in the middle of the plot up to 1 meter depth, and soil sampling were collected from 0 until 15 centimeter, 15 to 30, 30 to 50, 50 to 100 centimeter, and more than 100 centimeter depth, with the total sample are 290. Carbon, water content, and pH were analyzed in plant ecology laboratory. Ladies and gentlemen, we are going to the result and discussion, and I will describe the result and discussion into 3 sections: 1, water content, carbon, and soil pH. This figure shows that the water content in the soil in various soil depth, and green, the color difference indicate the difference in the depth of the soil. From this figure, so that the water content in closed canopy cover in D, dairies, terrestrial in shrub, dense, sparse mangrove, planted mangrove are higher than open area. This shows that the closed canopy cover area are capable of storing higher water than in the open one. The height of water content may not only be associated with the season, but the type of the soil texture, such as clay soil can hold more water than other soils. And the location of dairies, Nipa, are closer to the sea or stream and experience tidal flooding. Therefore, the water content is higher than SMV. This is supported by the research conducted by Hemati in Peninsular Malaysia. And this gray box show the water content in each location. Carbon contents in the closed canopy cover, as shown in this figure, higher than in the open area. The color also indicate the difference in the depth of the soils. From this research, we know that the highest value of organic carbon content were found in the dense, sparse, and planted mangrove. And generally, the results show that increasing soil depth showed a gradual increase organic carbon content, as also described in Bintuni, where the carbon pool increased with soil depth, which is in agreement with the trend commonly found in tropical forests. And this high carbon content were associated with the high productivity of mangrove species in the study area, where the accumulation of fallen litter increased organic matter in the soil. Condition in which organic matter and nutrients accumulate due to rapid use by biotic system are common in tropical forests. Therefore, it is estimated that the organic matter accumulation and carbon storage are determined by the rate of litter decomposition rather than the rate of organic matter production. For soil pH in a closed canopy cover lower than in open area, as shown in this figure, and the highest pH value was obtained in the 0 until 15 centimeter soil depth for almost all location. It was observed that the pH value of the soil decreased with the increasing depth of the soil, this is the same result of MRT study in Peninsular Malaysia, Sarawak, and India. These results are consistent with location with almost open area cover or lower vegetation type. Ladies and gentlemen, in conclusion, the research of the soil carbon on various land use in Segara Anakan shown that the value of organic carbon content in the highest found in the dense and sparse mangrove. From this result also determined that the gradual increase in soil depth indicates an increase in organic carbon content. In mangrove forests, the stand pH is a determinant factor that influence the amount of organic carbon in the soil regardless of the season, and we can conclude that the carbon content in mangrove vegetation has good potential of carbon sequestration. At the highest pH value were found at a depth of 0 until 15 centimeters, showing a pattern of decreasing pH value with increasing soil depth. This is the end of my presentation. Thank you very much.
Thank you very much for the presentation. So I would like to remind you that you are welcome to type your question in the chat box so we can later on read and discuss the question. So we are inviting the fourth speaker, Miss Penny Lestari.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you for the moderator and all participants for the opportunity to us to deliver our ISIBIO-based presentation. Our presentation is about characterization adaptation of foxtail millet Setaria italica on low light intensity to expand its adaptability. Our member consists of Dr. Randa Fauzia Sharif, Professor Nuril Hidayati, and myself, Penny Lestari, MSc. All of us are members of Research Center for Biology, National Research and Innovation Agency, BRIN. Why we choose foxtail millet? Millet are a group of small seed poaceae that widely grown around the world as cereal crop. For foxtail millet itself, it's a member of seed-type plant that have adapted to adverse environments such drought and also poor nutrition in the soil. It has short life cycle too, about 3 months, and this species potential to feed, food, and energy source. For Indonesia itself, foxtail millet or jawawut have nutrition much more nutrition than rice, wheat, and maize. It is necessary to acquire public awareness about jawawut and encourage the increased consumption in the community. Why? Because by consuming jawawut in our diet, we can improving the nutritional status of Indonesian people and also reducing rice and wheat flour dependency. And last but not least, it will promoting conservation that preventing jawawut extinction in the nature. This is our compile about jawawut across the time. Jawawut is not new species in Indonesia because our ancestors have been cultivating this species and it's proven on Candi Borobudur. Jawawut have been familiar to Indonesian people as bird feed instead of human food. And LIPI start to reacquire public awareness about Jawawut start from 2000s as human food. And IPB universities start to breed this species for abiotic stress, especially for drought, but not yet touched to shade condition. Why we choose that condition? Because we consider that farmers will prefer to cultivate economical communities. So we can introduce to cultivate Jawawut by intercropping system. Based on our exploration on 2015, we found several accessions, but one of them is Buru Kuning. Buru Kuning accession have good characteristics, consists such as dense panicle with few hairs, highly favorable taste to local community, and it have short plant stature, and according to Ridwan, it's shade tolerant. But there are notes in that report that there was reduction in panicle production until 57% due to shedding, so that we do microgenetic improvement by low doses radiation. So our objective is to explore the response of the Buru Kuning mutants to the shade environment as an initial screening for the shade-tolerant Buru Kuning accession. Our method, we did the research in LIPI research station in Cibinong. We radiate the seed of Buru Kuning and plant and use and no mutant generation in this research. Our experimental design, we use nested RCBD with light intensity as main plot, and the radiation doses reached to 200 gray for subplot. And our observation focus in plant height and leaf physiological traits and also biomass were observed. This is our result that mutant was detected start from 25 gray, but morphologically, it's detected start from 100 gray. Okay, plant biomass can express the growth of the plant. We found that plant biomass was a half time shorter than control. This one, and the root development was reduced because of shade, resulting in higher SR ratio. Panicle weight of mutant was higher in its wild type on full light, and it means that mutant perform better in shade condition. Yeah, this one, a higher mutant production. Okay, by leaf properties, for leaf properties observation, we found that the gap in the observed value of leaf size, leaf greens, and the member of stomata for 25 gray and 50 gray mutants were narrower than the wild type of Buru Kuning. That means the mutant more tolerant than its wild type. So, our conclusion is no significant morphological difference between the wild-type Buru Kuning and its mutant. However, the 25 and 50 gray mutants showed an improvement adaptation to shade compared to the wild-type. Considering the level of shade that the plant can tolerate, it is recommended to use an intercropping system to expand the cultivation of Buru Kuning foxtail millet and its mutant. Okay. For all participants, we say thank you, and we send back the time to the moderator. Thank you.
Okay, thank you very much for the presentation from Ms. Penny Lestari, and we will play the last video from the last presenter of this parallel session from Mr. Muhammad Nikmatullah.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. I am Muhammad Nikmatullah from Research Center for Biology, National Research and Innovation Agency, or BRIN. Okay, I would like to present our research with the title An Ethnobotany Study on the Plant Utilized as Pesticide by Communities in Cianjur, West Java, and East Lombok, West Nusa Tenggara, in the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering 2021, or ISIBIO 8. Okay, the first is introduction. There are many negative impacts caused using synthetic pesticide, especially effect to human health. The use of pesticide also negative impact on the environment and ecosystem. Therefore, this has prompted an international agreement to restrict the use of chemical substances, such as synthetic
Hello? Is this a kind of technical problem from the operator? So the video is freezing?
Yes, we have technical issue.
All right.
Oh,
Okay.
59. Sorry. Okay, we'll play it again.
Okay. We will wait for the organizer to try to play it again.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. I am Muhammad Nikmatullah from Research Center for Biology, National Research and Innovation Agency, or BRIN. Okay, I would like to present our research with the title An Ethnobotany Study on the Plant Utilized as Pesticide by Communities in Cianjur, West Java, and East Lombok, West Nusa Tenggara, in the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering 2021, or ISIBIO 8. Okay, the first is introduction. There are many negative impacts caused using synthetic pesticide, especially effect to human health. The use of pesticide also negative impact on the environment and ecosystem. Therefore, this has prompted an international agreement to restrict the use of chemical substances, such as synthetic pesticide in the controlling pests and disease in agriculture. In terms of biodiversity, Indonesia is estimated to have at least more than 30,000 species of flower plant. Furthermore, 9,600 of those plants are not as useful plants. One of the use of this useful plant is as a biopesticide. Pesticides from plants are toxic compounds which basic ingredients come from plants to control organisms that interfere with cultivated plants. This study is expected to know plant diversity that have the potential as natural pesticide so that it can reduce the dependence on synthetic pesticide while protecting the environment and health. The ethnobotanical study was conducted by exploring useful plants, especially for biopesticide, by local communities in Sarongge Village, Cianjur Regency, West Java, and in Sembalun Village, East Lombok Regency, West Nusa Tenggara. The result, there are 10 species belonging to 9 genera from 6 plant families which have potential as pesticide. The plant part used for pesticide are odd leaves of 9 species, young leaves of 2 species, bark of 3 species, and stem of 1 species. The local communities still processes the plants to be pesticide in a simple manner, such as boiling the plant part with the water. Most of plants that were used for pesticide were species of Meliaceae. Our studies confirm that plant species of the family Meliaceae are rich sources of active botanical insecticides. Based upon growth inhibition, chronic toxicity, and anti-feeding activity. Mostly part used for pesticide is leaves because it is faster to grow and easier to obtain. 10 species have potential for natural pesticide because it is a chemical compound that can be poison and anti-feeding for insects, so that it can inhibit the growth of pests. The first extract of Boeninghaussenia albiflora leaves contain coumarin. Coumarin has biological activity as an insecticide. The roots, leaves, and flower of Austrorhynpatorium innovalium contain bioactive compound that can serve as pesticide. Next, Swietenia mahagoni contain alkaloid, flavonoid, saponin, tannin, and terpenoid. This compound can inhibit metabolism in plant species. Next, Tinospora tuberculate contain alkaloid compound which can cause death to insect. Next, surenin, and carotenoid in Tona sureni, which have the potential to inhibit the growth of pests. And then Solanum bicolor. Solanum bicolor contain alkaloid. It belongs to carcinogenic plants that is poisonous and harmful to some animals. Blumea balsamifera contain terpenoid, phenol, alcohol, ketone. This plant can be toxic to wheat pests. The seed of Mucuna prurient are free from pest infestation. Extract from other plant part can also be used to treat pests in plants. Polyphenol, alkaloid, flavonoid, and saponin in datura metal effective in controlling and killing pests. And the last, tannin in Swietenia macrophylla is effective against mites, termites, insect egg, and larvae. Alkaloid in this plant are bioactive compound that can reduce insect growth. Oke, The last conclusion, there are 10 species belonging to 9 genera from 6 plant families which have potential as pesticide. Blumea balsamifera and Datura metal are used by the 2 local communities. The plant part use old leaves, young leaves, and bark. Most of plants part use in old leaves. The use of plants as natural pesticide in a simple manner by farmer in part of local wisdom that can support development of organic farming. Okay, that's all our presentation. Thank you very much for your great attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. We have finished playing all the pre-recorded video from all the presenters. So we start the Q&A session by first reading the question that has been posted in the chat box. So I again invite all of you to participate by asking question or typing some comment for the research presented by the 5 presenters in this session. So we begin with the first question for Miss Penny Lestari. So the question is from Dr. Yuni Setio Rahayu. Jewawut has been known for a long time, but commercially it has not been developed or has not been fully utilized. What is the cause of not developing Jewawut products based on your research, and what are the prospects for the future?
Thank you for Miss Yuni for the question. Okay, actually, Jewawut are famous enough in Indonesia as bird feed, instead of human food. And based on our experience in Magelang and also in Buru, actually the market of Jewawut were widely opened because Indonesia, by tourism route in the hotels, they use Jewawut in their menu as a snack. But we found the problem is about distribution and to meet farmer with the consumer. We think that the problems because the farmer don't find the market for their product, while the consumer here, I mean the hotel, they need Jewawut and they're imported from the other country. So we think the problem is the distribution. So we try to promote Jewawut to the farmer and meet them to the consumer, but we found that the volume of consuming Jewawut in the hotel is little because they only for pariwisata. They're only for tourism. So we try to promote Jewawut by intercropping system. So the farmer can cultivate the Jewawut and produce continue. So the hotel can get the Jewawut. Yeah, I think like that. Thank you.
Okay, thank you for the answer. Maybe we could come back to Bu Yuni after maybe finishing some question here if we still have some time, so we try to have discussion for presentation for Dr. Khairul Anam. There is one question here for Dr. Khairul Anam about from Berlianti Sarah Brin. Her question is, what CO2 gas do you use, from the air or pure CO2? And what source of the light you use, and how is the lighting process? Is it every hour or only during the day?
Okay, thank you very much. Thank you, Sarah, for your nice question. In this experiment, I use pure CO2 gas, which is in compressed condition, and I add it to the media with the flow speed is 1 liter per minute. And about the light, I use tungsten lamp, 36 watt. In each experiment, I use 2 lamps, except for the 1 liter cultivation, I use 4 lamps. So in this experiment, there is no dark and light periods. The culture always in the light period. Maybe that's all. Thank you.
And what about the CO2? CO2 is from
pure gas.
CO2. Okay,
yes.
Thank you. And we go to a question for Mr. Imad from Dr. Yuni Setio Rahayu. Have these species been proven to be effective as insecticides? And can this insecticide also be used in other locations and have the same effects or effectiveness?
Okay, thank you for question from Ibu Yuni Rahayu. There are out of 10 plant species as pesticide, namely Datura metal, and then Balsamifera, and Tinospora tuberculata were recorded also in 107 plants used as pesticide in Sumatra Island. But for 7 species, it has not been proven effective. Therefore, further research need to be done for 7 species. But at the research site, has been proven effective.
Okay. So we have at least one question for 3 of our presenters today. I would like to invite all the audience to ask maybe Bu Yuni or Miss Eka. Is there a question for their presentation?
Can I, moderator?
Yes, please.
Yes, thank you. I have a question to Miss Eka. As you mentioned in your presentation that when we do the lactic acid production by enzymatic reactions, we can obtain single stereoisomer, D or L lactic acid. So how about in your process? Did you get the single stereoisomer or you get the racemic of lactic acid?
Okay, thank you for your question, Dr. Asep. From our research is still in the preliminary, so our research is still in the broad fermentation, so the result is still racemic lactic acid, so they have any impurities from the product. So we not yet doing the purification to have the lactic acid, the pure lactic acid. So it is still in the preliminary study.
Okay, maybe I have one suggestion for you because your presentation is interesting. As you mentioned before that the alkaline treatment gave a positive effect into your reactions. So I think it is more informative if you can provide the SEM and also the XRD of your material between the untreated empty EFB.
Okay.
Yeah, that one, and with the treatment. Because usually when we did the alkaline treatment, you can observe the material changes of the material, and I'm sure that it is responsible for the enzyme to be, how to say, like easy hydrolyze and easy convert the sugar into lactic acid. Maybe that is one suggestion from. Thank you.
Okay, thank you for your suggestion.
Okay, thank you very much for the question and suggestion from Dr. ISIBIO for Ms. Eka. So maybe for Dr. Yuni, I have one question for you. If I'm not mistaken, in your presentation, you also, in addition to the pristine mangrove, you also analyze the carbon content on the area where there is a new plantation or artificial plantation. So is there any difference between the site or actually I want to ask how old is the new plantation sites? Is it old or newly revegetated? Thank you.
Thank you, Pak Muda. This is very good question, and I will explain that our study site, it's the planted mangrove. It's planted in 2013, so about in 2014, so about 8 years, and the canopy is already good. The growth of the mangrove are also already good, but still that the carbon contents, it's depend on the species composition, because in the pristine mangrove more species than in the planted mangrove. Planted mangrove only 2 species, Rhizopora mucronata and apiculata, and the other in pristine mangrove it more than 5 species. So it's based on the species composition also. I think it's my question.
Okay, thank you very much. It has been a interesting and wonderful discussion, but we have a very limited time here, and I guess we need to stop first because we need to, how to say, have prayer break and then move on to the next parallel session. So I want to thank all the presenters and all the audience for their active discussion, and I'll return the time to the moderator. Thank you very much and goodbye.
Thank you.
Thank you. Thank you to Dr. Muhammad Muhdar Davis and all presenters. We'll have 10 minutes break. We'll be back at 4. We'll start the session at 4 p.m. Thank you. Good afternoon, ladies and gentlemen. Thank you very much that you still attend until this session 3. In a couple minutes, we'll have the last session of this parallel. And for the last session, session 3 will be moderated by Dr. Hairul Anam. But before we commence parallel session 3, let's enjoy a video from our partner.
Microtomy requires my full attention and dedication to make sure that I deliver the perfect slides. And with the Tissue Tech Auto section, I started getting the job done easier, faster, and with better quality. The automated block alignment makes sure precious tissue is preserved optimally. It is also a great help for making recuts, while the perfect alignment helps me produce even more slides. Trimming and sectioning are done with high precision, each section the same exact thickness. Thanks to the easy operation of the auto section, every technician in our laboratory is quickly up and running. Whatever the need, we are all confident to deliver the perfect slide each time. My name is Mark Shanti, and I'm empowered by Sakura.
Martin Chris, Freeze Drying System untuk penelitian, pilot project, hingga manufacturing produk Anda. Hubungi PT ITS Science Indonesia untuk informasi lebih lanjut. Halo, kali ini saya mau memperkenalkan alat laboratorium dari Xinjiang. Ingenious Tree. Sebelum pengenalan alat, apakah Anda pernah mengalami kesulitan saat mendokumentasikan hasil gel electrophoresis atau plot Anda di ruang gelap? Sistem dokumentasi gel secara manual terdiri dari ruang gelap, kamera, dan transluminator sederhana. Dokumentasi gel secara manual seperti ini kadang menyulitkan, dan belum lagi kalau hasil foto gel yang didapat ternyata blur. Dengan Ingenious Tree, Anda dapat memilih pencahayaan sinar UV, sinar biru, atau putih sesuai dengan pewarna yang Anda gunakan. Selain itu, software Genesis akan membantu Anda memilih pengaturan cahaya dan filter yang tepat, sehingga sangat memudahkan untuk mendokumentasikan gel fluorescent ethidium bromida atau cybersafe DNA atau protein gel dengan composite glue atau silver staining. Untuk penggunaan mode otomatis, Anda hanya perlu memilih tipe gel yang sesuai, kemudian software akan secara otomatis mengatur aperture, zoom, dan fokus serta posisi gel yang sesuai sehingga gambar gel terekam secara optimum. Software Genesis dilengkapi dengan database tipe gel dan pewarna yang tersedia di pasaran. Jadi jangan khawatir tidak bisa menemukan pengaturan yang sesuai. Sangat mudah. Hubungi tim ITS Science Indonesia untuk info lebih lanjut. Terima kasih. Stay safe.
Okay, ladies and gentlemen, we are now have the last session, parallel session 3. There will be 5 presenters, and this session will be guided by Dr. Hairul Anam. Dr. Hairul Anam, are you with us?
Okay.
Okay. Dr. Hairul Anam is a researcher of Research Center for Biotechnology, National Research and Innovation Agency. He obtained his doctoral degree at Nara Institute of Science and Technology, Japan, in the field of biological science. Without further ado, I'll give Dr. Hairul Anam to lead the session.
Okay, thank you very much. Thank you for your very nice introduction. I hope you all still have energy to attend this our last session. Okay, I will start. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon to all participants. We welcome you in room 2, parallel session 3. In this session, we have 5 presenters, and each presenter will present their topic more or less for 10 minutes. The discussion session will be followed after all the presenters finish their talk. But if you have a question during the presentation, you can type the question in the chat box. So thank you. So let us begin. For our presenter, I introduce you to Mr. Ahmad Faizi Andes. He will talk about the diversity of endophytic bacteria of Lempuyang Wangi from Enggano Island, Bengkulu Province. For Mr. Ahmad Faizi Andes, time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. My name is Ahmad Faizi Andes, and I will present my research about diversity of endophytic bacteria of Lempuyang Wangi, Zingiber 0.4 aromaticum fall from Enggano Island. Introduction. First is Enggano Island. Enggano Island is one of the outermost islands in Indonesia, located in the waters of Indian Ocean, south of the island of Sumatra. Enggano Island has a variety of flora, and one of the flora is Lempuyang Wangi. Lempuyang Wangi is a plant that are widely used as ingredients in herbal medicines. Indonesian people use the Lempuyang Wangi by boiling the rhizome and then using its boiled water. It is a less effective method because it needs a lot of biomass. There are another method that we can take the advantage of Lempuyang Wangi without having to use a lot of biomass. And the method is to utilize the endophytic bacteria that are present in Lempuyang Wangi. Endophytic bacteria is bacteria that live in plant tissues without causing disease symptoms in their host plants. According to Tan and Zhou 2001, endophytic bacteria can produce the similar biochemical compound that produced by its host. So, endophytic bacteria has a potential to replace the use of Lempuyang Wangi as medicinal ingredients. For the methods, first we do the sampling, and then we do the isolation and purification, morphological identification, gram staining, biochemical test. And after we got all of the data, especially on morphological identification, gram staining, and biochemical test, we determine the genus from its isolates with the Burgess manual as the reference. The results. After we isolate the part of Lempuyang Wangi, leaf part, stem, and rhizome, we obtain 44 isolates that you can see on the table. For the morphological identification, there are elevation, form, margin, and color. For the elevation, there are only 2 types of elevation that's shown from the 44 isolates. There are flat and convex. And form, there are just irregular and circular. For the margin, there are most of them are entire, but some of them are filamentous, lobate, and undulate. For the color, most of them are white and cream, and some of them are yellow and red. You can see from the table. After we did the morphological identification, we do the gram staining. From the 44 isolates that we obtained, 35 of them are gram-positive bacteria, and 9 isolates that marked with the red box are gram-negative bacteria. You can see from the PowerPoint. After we did the morphological identification, after we did the gram staining, we did the biochemical test. There are a lot of tests that I can say that catalase, motility, urease, citrate permease, carbohydrate fermentation, and starch hydrolysis. And then after we did the biochemical test, we determine the close related genus from its isolates. And then we found 5 isolates are close with the Micrococcus. Most of them are related or close with the Bacillus, that's shown with the red color. There's only one isolate that's shown the close relation with Amphibacillus. And 4 isolates are closely with the Azotobacter genus Azotobacter, and then 5 isolates are close with the genus Pseudomonas. And this is the graphic that shows the abundance of the genus, every genus that we found, of its plant part. As we can see from the leaf part, we only got 2 genus. There are Micrococcus and Bacillus. And for the rhizome, there are 4 genus we found, and it is the only part that we found Amphibacillus from the rhizome. And for the stem, we got 4 genus too. And we can see from the graphic, genus Bacillus is dominant in every part. So the conclusion of my research is the result of the isolation of Lempuyang Wangi, Zingiber zerombetvar, aromaticum fall, and the phytic bacteria from Enggano Island. We obtained 44 isolates of bacteria that are closely related to 5 genera, namely Micrococcus, Bacillus, Amphibacillus, Pseudomonas, and Azotobacter. And the genus Bacillus is the dominant genus in every part of the plant. Thank you.
Thank you for your nice presentation. Let us proceed to the next presenter. Here, the next presenter is Ms. Hayatul Fajri, who will talk about the leaf morphoanatomy variation between Codium variegatum. Please, Ms. Hayatul Fajri, the time is yours.
Hello everyone, Assalamualaikum warahmatullahi wabarakatuh. Today I will talk about the morphoanatomy variation between Codium variegatum. First of all, let me introduce myself. My name is Hayatul Fajri. I am from Tanjung Pura University. As we know, Codium variegatum is a popular ornamental plant from Euphorbiaceae family. This plant has high variation of leaf characteristics. The variation is in shape, size, and the color. Here in the picture, we can see that Codium variegatum is very varied. The variation is not only in the different plant, but we can see the color of leaf in a plant is different. In 2010, Deng et al. Was reported that 44 cultivars of Codium variegatum are divided into 7 clusters based on AFLP marker analysis. Then, in 2015, Widyaningsih also reported that 44 cultivars of Codium variegatum based on morphological character. The last, in 2019, Asmiawati and Purwantoro has revealed that Codium variegatum have 106 polymorphic band based on RAPD analysis. However, there is no information about the variation of Codium variegatum cultivar based on anatomical character. So, our research was to analyze the variation among Codium variegatum cultivar based on morphoanatomy character. We use egg number of Codium variegatum, namely TP1 until TP8, and all of cultivar have a different morphological character, and we take note to all of morphology and anatomy character of this egg cultivar. This is the summary of morphoanatomy leaf character of egg cultivar. The morphology character we have observed are in leaf tip, leaf base, leaf color, the type of leaf shape, the leaf age, and the variation of leaf color within cultivar. For anatomy character, we found that the egg leaf are different in midvein anatomy structure. The different character are in the presence of palisade in midvein. In TP2 and TP3 has a specific character. In this cultivar, TP2 and TP3 have palisade in midvein, but the other cultivar we cannot find the palisade in midvein. The other character we observe is hypodermis in abaxial midvein. Hypodermis in abaxial midvein cannot found in TP3 and TP4 cultivar, but the other cultivar we can find the hypodermis in abaxial of midvein. And then the hypodermis in adaxial of midvein we cannot find in TP2 cultivar, but in the other cultivar TP1, TP3, and until TP8 we can find hypodermis in adaxial midvein. The other character we observe is the number of vascular bundle. TP2 cultivar has specific character, which is this cultivar have only one set of vascular bundle, but the other cultivar have more than 3 sets of cultivar bundles. Here we can see the midvein anatomy of TP2 and TP3 cultivar. In both cultivar, we can find that palisade. This is a palisade in TP2 and palisade in TP3. And then the other character is hypodermis in abaxial midvein of TP2 and in adaxial midvein of TP3. The specific character of TP2 is we can find that only one set of vascular bundle in TP2 cultivar, and the other cultivar have more than 3 vascular bundle like this TP3. We also analyze the phenotypic relationship of egg cultivar Codium variegatum based on morphoanatomic character using enthesis. And the result is these egg cultivar are divided into 2 clusters. Here, 2 clusters of Codium variegatum based on morphoanatomy, and the coefficient similarity are 64% to 89%. This result indicated that among egg cultivar of Codium variegatum has high similarity based on the leaf character. So, the conclusion of this research is the variation among cultivar Codium variegatum was found on the number of xylem phloem bundle, the existence of palisade and hypodermis in midrange, and the form of hypodermis cell. And the A cultivar of Conium variegatum has high coefficient similarity, that is until 89%. We are thanks to Ministry of Education, Culture, Research and Technology to funding this research in schema PNBTF KIP Tanjung Pura University 20 and 21. Okay, for the last, I would say thank you for everyone and goodbye.
Thank you very much for your nice presentation. Let us proceed to the next, our presenter. Our presenter is Ms. Eva Erdayani, who will talk about identification of Typhonium flagelliform and its closely related species in Java Island using morphology and SRIP markers. To Ms. Eva Erdayani, the time is yours.
Welcome to my presentation. The title that I would like to share here is Identification of Typhonium flagelliform and its closely related species in Java Island using morphology and submarkers. Typhonium flagelliform is an herbal plant from the family of Araceae. It has been used traditionally as antihistamine to treat swelling, coughing, and mainly cancer. The plant is distributed in Indochina, native to Indonesia, especially in Java, Sumatra, and Bali. It has several local names such as Caladiticus, Tenggiling Menti, Kalamoyang, Iles, Kibabi, etc. Due to its morphological similarity, this species is frequently confused with the other Typhonium, especially with Typhonium blumei, Typhonium roxburghi, and Typhonium trilobatum. In many cases, there are plant materials might share same local names but belong to different botanical families. This has been an issue in the development of herbal medicines because the use of inauthentic plant might give consequences to the efficacy and safety of the products. In fact, adulteration and substitution of plant material has been common practices in commercial production of herbal medicines. So correct identification of plant species is critical in developing herbal medicine with standardized quality control. The objective of this study is to perform taxonomy identification of Typhonium flagelliform of different accessions in Java and its closely related species using morphology and submarkers. We have collected 15 plants, 10 from West Java, 3 from Central Java, and 2 from East Java. We did morphology identification using determination key by Nicholson and Sipadasan, and we perform molecular analysis using sequence-related amplified polymorphism or submarkers by the combination of 6 primers. We did data analysis with unweighted pair group methods using arithmetic average for the tree construction, and we did the analysis using multivariate statistical program version 3.22 software. From the morphological characterization, we could observe variation in the flowers and leaves of the plants in our collections. In conclusion, there are 2 species, Typhonium flagelliform 10 accession, Typhonium roxburghi 4 accession, and Typhonium blumei only 1 accession. Previously, Typhonium blumei was only reported in Sulawesi. So this is the first report, the presence of Typhonium blumei in Java. And from the features in SPADIC, we observed variation in the sterile zone on sterile flowers. Based on the description from Nicholson and Sipadasan, it should also be Typhonium trilobatum, but we didn't find in our collection. This is previously reported in Kalimantan, so it might be not present in Java. We also observe intraspecific variation in the spats and the leaves of Typhonium flagelliform. The color variation could be seen from purple, yellow to green color. And we also observe one feature in Kebumen accession. It has dark patches on adaxial part of its leaves. The variation also observed in Typhonium roxburghi. Two accession have trilobate leaf blades, while the other two accession with hasted blades. We did molecular characterization using strap markers, and we obtained around 86% polymorphism using the combination of 6 primers. We could see here some examples of the electrophoresis results. And we built dendrogram using the data. From the tree, we could observe 2 clusters. The first cluster consists of Typhonium roxburghi, and we see here separation of Typhonium roxburghi from Kebumen with the ones from Cibinong. The second cluster is Typhonium blume with Typhonium flagelliform, showing closer relationship between the 2 species. And in Typhonium flagelliform, we also observe a cluster of West Java species separated from East and Central Java species. Except the one from Cibinong, it is out of the groups from the other accession. It might be because this species coming from another place. In conclusions, taxonomy identification of Typhonium flagelliform and its closely related species could be performed based on morphological characteristics and submarker system. This study will be useful as one of the references for species identification in the development of standardized herbal medicines. Thank you.
Thank you very much for your nice presentation, Ms. Eva. To all participants, don't forget if you have question, you can type the question at the chat box. Thank you. For our next presenter is Mr. Iskandar Azmi Harahap that will talk about the quantitative risk assessment of dietary exposure to lead from sea cucumbers in Indonesia. To Mr. Iskandar, the time is yours.
Hello, good afternoon everyone. I'm Iskandar from National Research and Innovation Agency or PRIN. Today I would like to discuss about quantitative risk assessment of lead contamination in sea cucumber in Indonesia. If you have any questions, we will be discussed until the end of my presentation. The presentation contains 5 parts, namely introduction, objective methodology, results, and conclusion. As we know that cucumber is a nutritious food source. On the other hand, we are facing ocean environmental issues, for instance, marine debris, industrial activities that can pollute the ocean, and so on. So there is a potential hazard for marine biota, where chemical hazards more dominant than microbiological hazards. One of which is lead, a natural environmental contaminant. However, lead legislation for Echinoderma are no longer listed. Therefore, the information about this study is needed. So, this research aimed to determine quantitatively the risk of exposure of lead contamination in consuming sea cucumber in Indonesia. We move on to the method of this research. Cucumber samples were taken from Lampung, Bali, Lombok, and South Sulawesi. We used primary and secondary data from Sri Lanka and China for lead concentration because these locations are close to the Indonesian seas. Furthermore, due to limited cucumber consumption in Indonesia, we used the consumption data of fish and seafood in Indonesia, Sri Lanka, and China. In the study, there were 4 steps to determine the quantitative risk assessment of lead contamination. First, hazard identification. According to the FDA, the potential hazards in cucumber are environmental chemicals, where cucumber is deposit feeder that ingests the sediment as its primary food source. Second, hazard identification. The general population is exposed to lead via food, water, air, soil, and dust, where food is the primary exposure source. Moreover, the proportion of lead absorbed from the GI tract is about 10% in adults and 40 to 50% in infants. Third, exposure assessment calculated the hazards by measuring probabilistic using at-risk software, performing best fit distribution, and determining Monte Carlo simulation by 10,000 iterations. Fourth, exposure assessment. The study used 2 scenarios. Scenario 1 was all data that we used, and scenario 2 was only data below the limitation level by National Agency Drug and Food Control Republic of Indonesia, namely 200 microgram per kg. The result of this study, probabilistic exposure assessment, showed that in scenario 1, lead intake from cucumber consumption spanned from 0 to 0.912 microgram per kg person per day. In scenario 2, implementing a maximum lead intake level of 0.2 microgram per kg resulted in an intake of 0 to 0.288 microgram per kg person per day. Furthermore, the average lead exposure levels were 1.28 microgram per kg body weight per day in scenario 1 and 0.11 microgram per kg body weight per day in scenario 2. This study summarizes quantitative data related to lead exposure related to cucumber consumption in Indonesia. This study revealed that the average lead exposure level in scenario 2 were less than those in scenario 1, due to many countries having decreased exposure of lead successfully. This study's result also showed the implementation of limited lead levels could decrease heavy metals contamination in food to protect Indonesian human health. I expressed my gratitude that this study was performed as a part of the International Training Program in Food Safety, Quality Assurance, and Risk Assessment in 2019, organized by the Department of Food Safety and Food Quality, Faculty of Bioscience Engineering, Ghent University, Belgium. We also would like to thank the head of Research Center for Biotechnology, Indonesian Institute of Sciences, as well as the research and administration staff members for this support. Thank you for your attention. Bye.
Thank you very much for your nice presentation. Let us proceed to our last presenters. Would be Mr. Laude Muhammad Muhdar Davis. He will talk about the effect of mild and severe drought on jelly chili leaf cell growth. To Mr. Muhdar Davis, time is yours. Maybe, okay, thank you.
Good evening, everyone. My name is Muhdar Davis from the Research Center for Biology, National Research and Innovation Agency, BRIN. It is an honor for me to join this wonderful conference and share one of my research results entitled The Effect of Mild and Severe Drought on Guinea Chili Leaf Cell Growth. This work is done by myself together with my colleague Penny Lestari and Professor Wahyu Widiono from the Research Center for Biology BRIN, in collaboration with our partner Professor Muhammad Syukur and Dr. Tri Kusumaningtyas from Faculty of Agriculture, IPB University. I will begin with this introduction. Cabai rawit is irreplaceable for Indonesian cuisine. Thus, sustainability in cabai rawit production is essential, especially in light of adverse effects of climate change we are facing nowadays. These climate change impacts include abiotic stresses such as drought. And as you may know, drought is the primary constraint to plant growth and productivity. And here we study Geni, one of cabai rawit variety that belong to Capsicum annum species. Geni is one of local commercial genotypes developed from Indonesia, and its yield is around 16 tons per hectare. And according to some studies, this genotype is moderately tolerant to drought and osmotic stress. However, those studies mainly focus on yield and macro growth of parameters, and the understanding of physiological and growth response at organ and cellular level under drought condition remain to be explored. And therefore, of many local genotypes that we are working on right now, Geni is so appealing for us to study. And the aim of this research is to investigate Geni chili growth during vegetative phase prior to flowering in response to drought, with specific objectives to evaluate first, primary growth and biomass, second, organ formation and size along with its cellular growth parameters. And to do that, we prepare 1 month old young chili plants, and we subject those plants to a drought experiment. And there are two levels of drought condition we employ. First, the mild drought, and second, the severe drought that differ in their field capacity. And field capacity is the maximum water moisture that are retained by the soil matrix against gravity action. And for the control here, we use 80% field capacity, for mild drought 50%, and for severe drought 35% field capacity. This drought experiment lasts for 5 weeks at greenhouse, and we collect initial data such as plant height and leaf density. And at the end of experiment, we collect plant growth parameters such as height, leaf density, root length, and biomass. And we also collect leaf growth parameter data such as leaf blade area, average epidermal cell area, cell number, density, and stomatal index. And this is the result of the experiment. So here's some photos of the representative plant from each drought treatment condition. Here we can see that plant height was negatively affected by the drought treatment. Upon mild drought, the plant height is decreasing by 21.9%, and at severe drought, it decreased by 44.4%. However, there is no significant difference between root length in drought-treated plant compared with that in control plant. As for the biomass result, we found that Geni chili biomass noticeably dwindled in response to drought. Here, for example, we can see that the shoot biomass is clearly decreased, especially in response to severe drought treatment condition. Meanwhile, root biomass didn't differ significantly from that of control under mild and under severe drought. And if we take a look at the percentage biomass, we can see that shoot biomass is progressively reduced, while root biomass is increasing. And this result indicates that the drought effect was more pronounced on the plant shoot compared to the root biomass. And then plant biomass proportion shift towards the root portion of the biomass under drought stress condition. And next, we investigate growth at organ level, and in this case, the leaf. We focus on the leaf parameters such as leaf density and leaf blade area. This is important because leaf account for the majority of plant shoot growth. And here we can see that the leaf density actually is not significantly different before and after 5 weeks of experimental course, as well as between drought stress treatment condition. Meanwhile, The leaf size here, the leaf blade area, shows significant reduction, especially upon severe drought condition. The reduction reached up to 66.25% under this harsh condition. And this result indicate that leaf size is clearly decreased in response to drought. We then perform microscopy analysis on the abaxial epidermal layer of the leaf. And as you may already know, epidermal layer of the leaf consists of the arrangement of pavement cells and stomata guard cells. And this analysis is done to investigate the cellular growth parameters such as average cell area, cell number, cell density, and stomatal index. We found that the average leaf epidermal cell area were suffering a noticeable decrease here by both mild and severe drought. Interestingly, total cell number per leaf increased in response to mild drought, but markedly decreased in response to the severe drought. And epidermal cell density per leaf area unit increased in response to both mild and severe condition. So in general, we found that leaf epidermal cell size is negatively affected by mild and severe drought, but the reduction in the leaf cell number occurred only when the Guinea plant afflicted with severe drought condition. And it seems to be that the increase in cell number and density under mild drought condition is a compensation mechanism to maintain leaf growth and final size. And here we also show that severe drought significantly decreased stomatal index. This result indicate that under harsh drought condition, plant has less stomata or leaf pores. And from our current data, I want to highlight several points. So first, Guinea chili pepper may employ a stress avoidance mechanism to balance water loss and water utilization. For example, by reallocating growth from shoot to root. And indeed, we observe Guinea biomass proportion shift toward root fraction in response to drought. And at cellular level, the stomatal index decreased progressively as the drought become harsh. And this implies that it may be a mechanism to limit the water loss by limiting the number of leaf pores in the leaf. And the maintenance or induction of root growth allow the plants to reach for water and nutrient deeper in the soil. And accordingly, limiting the shoot production is advantageous to slow down water transpiration. And this growth refocusing can be a beneficial adaptation strategy in responding to water limiting stress. And to summarize, first we found that drought negatively affect Guinea chili shoot growth and vegetative phase biomass production. Second, drought-treated plant has reduced leaf size as a result of decreased cell area. Third, severe drought decreased both cell size and cell number. And fourth, Guinea drought response suggests an avoidance mechanism to the stress. And lastly, I want to share one of limitations in our methodology and way forward what to do in the future. And while our approach can define certain parameters such as cell area, cell number, cell density, it cannot determine the spatial temporal aspect of cell division because the data only observed at one time point. And for example, it is possible that drought inhibit the rate of leaf expansion either by slowing down cell division or by slowing down cell expansion. Or alternatively, it occurs as a result of complete inhibition of both cell division and cell elongation. And actually, there are many other combination or possible scenarios. Therefore, our initial results require future investigation involving a series or time series analysis to elucidate the dynamic of cellular growth, such as cell cycle rate, leaf cell elongation rate, and so on. And secondly, characterization of wider collection of chili genotypes using this pipeline or methodology could reveal a new insight into chili pepper tolerance or sensitivity to drought. And with that, I end my talk and would like to hear your question and feedback. Thank you very much for your attention.
Thank you very much for your very nice presentation. As the last presenter has finished his talk, Now it's time we open the discussion session. To all participants who want to ask a question, you can raise your hand to ask directly to the presenter, or you can type it in the chat box. Thank you. Here we have a question from Miss Penny Lestari to Mr. Faizi. Mr. Faizi, are you ready?
Yep, I'm here.
Yes. You said in the presentation that endophytic bacteria produce the identical metabolite as its host. My question is, the bacteria produce the metabolite by itself, or it still need the host to produce it? If it need the host, how we can harvest the target metabolite through the bacteria?
Okay, thanks for the question. According to Tanenzo 2001, endophytic bacteria will generally produce secondary metabolites similar to those produced by their host. This is due to the genetic recombination between endophytic bacteria and their host. So from the Tanenzo 2001, it says that the endophytic bacteria produce by itself because it has the genetic code that need to produce the biochemical compound. So it no need to the host anymore to produce the biochemical compound. That's my answer.
Okay, that's your answer. How about Miss Penny? Do you have any feedback or suggestion?
Yeah, thank you. It's clear enough.
Okay, thank you very much. Is there any other question for the presenters? I have a question. No, there's no. I have a question for Miss Hayatul Fajri. Is there any economical reason for you to choose this topic regarding the variation of Codium variegatum because of its popularity?
Okay, thank you for the question. Yes, of course, I have a reason why I choose to explore the variation within this puring. We know Codium variegatum as puring. This plant is very popular, and we know this plant is very it's very resistant to high light exposure, and I know and we know this plant is very varied in plant in morphological, and for this research I'm wondering how about the anatomical, and for the result I get the puring is not too variation, not too vary in anatomy, but maybe someday this result will be as the first information to us to learn about the response to high light exposure. So maybe this plant could be what we call could be the genetic resource to make to create a plant with genetic engineering, maybe to create the plant that resistant to high light exposure.
Okay, thank you for your answers. Is there any question for the presenters? Okay, maybe because of the I have to put some record for the presenter, so I have to ask all the presenters who have not asked by the question. So to Miss Eva Erdayani, I have a question for you. As I see at the chart or the chart among the Typhonium flagelliform collected from different places also give different similarity. Do you also have concern about this? To Miss Eva?
Similarity for
Yeah, because the Typhonium
you
flagellarium,
mean similarity for
yeah, different similarity, so it's not the same. It's not also not the same. It's similar but not the same. Do you also have concern about this? How about the safetiness or some maybe the effectiveness of the
yeah.
compound?
Yes, we actually we are doing the research for the phytochemistry and also the effectiveness of the extract to treat cancer. So the research also is still going on. So we are interested to know if the variation of the plants will give different effectivities also in the treatment for the cancer. Is that answering your questions?
Okay, thank you, Miss Eva. So is there any question for the presenter? Still no? So for Mr. Iskandar Harahap, as I see in your presentation, at the results slide, there is a table which explains about estimated lead, intake, and margin of exposure at mean and percentage in sea cucumber by Indonesian population. My question is, what number do you use for the Indonesian population in your this research, Mr. Iskandar?
Yes, I am.
Can you hear the? Do you hear the question? Did you hear?
Could you repeat, please?
So as I see in your presentation, at the result slide, there is a table which explains about the estimated lead, intake, and margin of exposure at mean and percentage in sea cucumber by Indonesian population. My question is, what number do you use for the Indonesian population?
Okay, wait a second. I will check. Okay, the Indonesian population that I use for this study, I use the calculation with other countries as I mentioned on the methodology method chart. Yes, that's all.
So do you put all of the population number as for the dividing your estimated lead and intake?
Yeah, I use for the calculation for population, all of them from Indonesia, Sri Lanka, and China. So I use the estimation for the population of consumption from those countries.
Oh,
yeah, but I want to check first on my calculation if you want to see how many population in exactly.
Okay, thank you very much for your answers. So
you're welcome.
Is there any question for the presenters from the participants? So to Mr. Muhdar Dafiz, I have a question for you. I was curious, is there any correlation between the water capacity or the level of the drought with the productivity and the quality of the chili as commodity?
Yes, actually, based on literature, there is like a correlation. For example, some study have shown that limited water actually increase the bioactive compound of chili, the capsaicinoid. So it is a subject of investigation also to optimize what kind of drought condition that can optimally induce the high amount of this bioactive compound, but not limiting the growth, because we know stress cause inhibition to the growth, the primary growth of the plant. But in our current study that I presented just now, we didn't do such analysis. We only study the cellular and organ growth of chili plant in response to a water limiting situation.
Okay, thank you very much for your answer, Mr. Muhdar Dafiz. Is there any other question for the presenters? If it's not, I will give back to the committee. So thank you very much for the all presenter for your very nice presentation, and see you in other session. Thank you very much. I give back to the committee.
Thank you very much, Dr. Anam, for your guidance, and thank you to all presenters for the participation. Okay, ladies and gentlemen, we still have a closing session and announcement of best presenters. So we expect you to join at the main room. You can leave room, you can just leave room, and you'll be directed to the main room. And we'll wait for the other participants to join us in the main room. Thank you.
Recording in progress.
Welcome back, ladies and gentlemen, to ISIBIO 2021. So we're waiting for other participants to join us in the main room. And after that, we will announce the best presenter of ISIBIO 2021. So please keep waiting. Thank you. Ladies and gentlemen, welcome back to the main room of ISIBIO 2021. We would like to thank all the speakers and moderators for very insightful session. Okay, so for the next, we will announce the best presenter of ISIBIO 2021. And the best presenter are chosen based on the highest score of the presenter in each room during day 1 and day 2 of parallel sessions. Now we would like to announce for the first day, the best presenters of the first day of ISIBIO 2021 are Room 1, Nikadek Erosi Undaharta. I'm sorry for the wrong spelling. And Room 2, congratulations. Room 2 is Mr. Aryo Beta Juan Silvero. Congratulations. And Room 3, the winner is Fitria Dwi Ayuning Bias. Congratulations for the winners of the best presenter of the first day of ISIBIO. Now, move on to the second day. The best presenter of the second day of ISIBIO 2021 are Room 1, Mrs. Wayuni. Congratulations. Room 2, La Ode MM Davis. Congratulations. And last but not least, Room 3, Andri Wardiana. Congratulations once again. Let us give a round of applause to the winners, and you can use the reaction feature in the Zoom to congratulate the winners. Once again, we'll say congratulations to the best presenter for delivering such remarkable presentation in ISIBIO 2021. Ladies and gentlemen, we will have closing remarks by Dr. Mastur, Chair of Indonesian Biotechnology Consortium. Please welcome Dr. Mastur.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. Distinguished invited participants, organizers, ladies and gentlemen. First, let us express our praise and gratitude to Allah the Almighty for His grace and guidance that today we can successfully finish the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering. On behalf of everyone involved in this conference, I will also express our gratitude to the Chairman of National Research and Innovation Agency, Dr. Laksana Trihandoko, for his support, direction, and opening remark. His support motivates and boosts our energy to enhance research and application of biotechnology, to support national development in all regard sector and field. Biotechnology at the front line of science and technology is required to increase productivity, quality, efficiency on biodiversity, sustainable environment management and use, medical and industrial sectors, product diversification, and new innovation. Biotechnology is also expected to overcome the climate change through mitigation and adaptation to bring great benefit to human life and environment. During these 2 days, we had a fruitful discussion and sharing information. Thank you so much for all resource persons for your excellent ideas and sharing information. For all participants, thank you so much for your support in scientific papers. We highly appreciate all sponsors and support for this international seminar, especially for BRINS and Biotechnology Enterprises Partners. KBI management and all members believe everyone gain a huge benefits from this program. Therefore, KBI management will commit to promote biotechnology through the similar program, especially during period 2021-2024. We will improve collaboration with other stakeholders, universities, research institute and bureaucrats, and industry and other biotechnology practitioners. Synergy to support research, education, and industrial practices in Indonesia. At this opportunity, we will also invite all elements, universities, research institute, government institution, industry, and business related to biotechnology to join KBI. KBI management commits to bring real benefits to all members. KBI will facilitate research, engineer, university lecturers, and students to improve their research and publication. For bureaucrats, KBI will participate in promoting regulation. For industry and business sector, you will get opportunity to advertise your product. Finally, we hope collaboration between KBI members and all stakeholders will be strengthened and intensified. Lastly, thank you Research Center for Biotechnology BRIN for great collaboration. For all participants, on behalf of the conference organizer, please accept our sincere apologies for inadequate services or any shortage during the conference. We hope this conference will bring advantage for everyone who join for our organizations and national development. Praise to God. Alhamdulillah, I close this conference. Thank you and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Masur, for officially closing the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering. We would like to give gratitude to Chairman of Research Organization for Life Sciences and Chair of Indonesian Biotechnology Consortium, all the keynote speakers, invited speakers, moderators, and presenters of the International Symposium, and not to forget the participant. Thank you for the participation in ISIBIO 2021. Don't forget to fill out the list of attendees, and the certificate will be sent by the committee through email maximum 15 working days. Tomorrow, we will also hold the 7th Indonesian Biotechnology Conference, or IBC 2021. There would be talk shows with many great speakers, so we hope you can join the event tomorrow. And on the behalf of the official committee, I would like to apologize for any inconvenience matters during the event. So see you on ISIBIO next year. Have a good day. Goodbye.
Oke, thank you Mbak Wiwit. Thank you semuanya.
Thank you very much.
Thank you.
Thank you for all. See you in next year.
Thank you semua panitia, seluruh komite. Live ya.
Iya, Bu.
Asep, Bayu, makasih ya.
Makasih.